Good morning, everyone. Today, the 6th of October, holds a very special significance for all of us at the National Institute of Immunology. It's a day of honor, one that invites us to celebrate our journey, reflect on our achievements, and look forward to the future. I, Banya, would like to invite one and all of you to the 39th Foundation Day of National Institute of Immunology. On behalf of the NII family, I extend a very world-renowned biophysicist, Dr. Rajesh Gokhale, Secretary to the Government of India, Department of Biotechnology, and Director General, Biotechnology Research and Innovation Council. At this moment, may I invite Dr. Debashish Mohanty, our Director, to present a token of our appreciation to Professor Talwar. A round of applause, please. Professor Balram. A token of our gratitude and appreciation for Professor Balram. Welcome to NII, Professor Balram. And a token of our gratitude and appreciation for Dr. Gokhale. Welcome to NII, Dr. Gokhale. Extend a very, very warm welcome to all the dignitaries, the scientists, researchers, alumni, staff, and students who have joined us and cherish the spirit of NII. Albert Einstein once said, the important thing is not to stop questioning. Curiosity has its own reason for existing. And this time, I think, captures the very essence of NII. A community of curious minds united in the quest to unravel life's most intricate design, possibly complex yet beautifully orchestrated immune system. Few institutions are fortunate to see their founding ideals come a full circle within a single generation, and fewer still have the privilege of doing so in the very presence of the individual who planted the first seed. This year's Foundation Day is especially meaningful for all of us. As we mark 39 years of NII, we also celebrate the extraordinary journey of Professor Talwar, whose vision gave birth to this institution. In his honor, we are hosting the two-day symposium, Immunology and Allied Science Research in India, Foundations, Frontiers and Future, to reflect on his legacy as well as the expanding horizons of immunological research in India. Since its inception in 1981 and formal establishment in 1986, NII stood at the forefront of understanding the immune system. Over the years, it has grown into a vibrant community of researchers, advancing research in immunology, infectious diseases, structural and chemical biology, genetics, cell signaling, cancer, all under the same roof, built on a very strong foundation of vision, knowledge, and excellence. We continue to address global health challenges with very clear purpose as well as a forward-looking spirit. Today is not just a celebration, but a moment of reflection. Where do we go from here? And to share this vision, and again, welcome all the esteemed, extended family of researchers who join us today, I request our director, Dr. Debashish Mohanty, to deliver the welcome address.
Namaskar, good morning. Professor G.P. Talwar, our founder director, Professor Balram, chief guest for today's event, Dr. Rajesh S. Gokhale, Secretary, DVT, and Director General, BRIC, all the guests and dignitaries, my faculty colleagues at NII, our students and staff of NII, our alumni and former colleagues. I welcome you all to 39th Foundation Day ceremony of National Institute of Immunology. So this year's Foundation Day is very special because our visionary founder director, Professor Talwar, just entered his 100th year on 2nd October 2025. We are very happy to note that, like every other year, Professor Talwar and his family members have joined us for the celebration of the Foundation Day of NII. I welcome them all. It is a rare occasion in Indian science when the institute is entering its 40th year. Its centenarian founder director is vigorously pursuing his passion for translational research with the ultimate objective of serving society. To celebrate this occasion, we have another stalwart of Indian science, Professor Padmanabhan Balram, who is not only a world-renowned biophysicist, but also one of the very few scientists with deep insights into areas beyond the conventional boundaries of scientific disciplines. We are also extremely happy to have Dr. Rajesh S. Gokhale, Secretary DBT, with us to celebrate this special day. He carried out his PhD under Professor Balaram, and NII, the institute Professor Talwar set up, provided him the opportunities to establish himself as a world-class researcher in tuberculosis and also lead the Department of Biotechnology of Government of India. I thank you all for your enthusiastic participation in this event. I apologize to all those who couldn't be accommodated inside this auditorium. So we have the webcasting of the event and they can join from the other seminar halls of NII. So every year we have been celebrating the Foundation Day of NII on 6th October to commemorate the day on which NII was dedicated to the nation by the then Prime Minister in 1986. However, NII was registered as a society in 1981 and it required several years of hard work by our visionary founder director, Professor Talwar. NII Society was established under the leadership of Professor Talwar when government identified basic and applied immunology as a priority area in recognition of its potential impact on human health. In the first decade of its existence, NII made major contribution in area of vaccine development, reproductive immunology, and most notable being successful development of leprosy vaccine based on Mycobacterium indicus pranae. It is pertinent to mention that utility of MIP as an immune modulator has led to the exploration of efficacy in sepsis, cancer immunotherapy, and in recent years. And from its inception, the institute was envisioned not as a conventional research establishment, but as a living ecosystem of innovation where fundamental inquiry meets translational impact. So Professor Talwar's visionary leadership also led the foundation of immunology research in India and continued to inspire generations of scientists. To honor the remarkable scientific legacy of our founder director, we are organizing a special two-day scientific symposium titled Immunology and Allied Sciences in India, Foundation, Frontiers and Futures. I am thankful to all the associates of Professor Talwar from his AIMS and NII days, our former faculty, alumni, and some of the leading researchers in the area of TB and leprosy, immunology and allied sciences, reproductive immunology, and cancer, who have enthusiastically joined this event despite very short notice. So this symposium embodies our heritage and horizon, a tribute to the pioneers who led the foundations and a call to the next generation to carry the torch forward. As we celebrate Professor Talwar's legacy and NII's evolution, we are reminded that institutions derive their strength not from buildings or budget, but from the courage of their ideas or the vision for the people. Today, this vision stands fulfilled across many frontiers of immunological science. Over the decade, NII has not merely observed the evolution of immunology in India, it has actively shaped its trajectory from pioneering the world's first contraceptive vaccine, early leprosy vaccine trials, to leading national programs on tuberculosis and emerging infectious diseases. NII has consistently translated discovery into impact. Our efforts today span infectious diseases, cancer biology, metabolic disorders, neurodegeneration, aging, nutrition, and vaccine design, reflecting the ever-growing realization that immune system provides to unifying framework for understanding health and disease. From decoding the molecular machine of Mycobacterium tuberculosis to coordinating Indian TB genome sequencing consortium, from the pioneering malaria vaccine research to contributing to the development of nasal COVID vaccine in COVAX, our scientists have stood at the forefront of public health imperative. Now, as NIE integrates into the Biotechnology Research and Innovation Council, BRIC, we enter a new era defined not just by discovery, but by scale, speed, and collaboration. The convergence of single-cell transcriptomics, AI-driven analytics, precision immunology is rewriting the rules of biomedical research. Whether in antibody engineering, immune modeling, or biomarker discovery, the tools of the future demand that we ask bigger questions and forge stronger alliances. As we embrace the technological renaissance, we continue to uphold our founding principle, excellence with responsibility. So the source of the success of NII is also evident from the fact that 8 of its faculty members have been recognized by Santi Swarup Bhatnagar Prize, the most prestigious award in Indian science. NII faculties have a significantly high share in fellowship of leading science academies in the country. 10 of our faculty members have been selected to take up leadership positions in other research institutes. That includes the Dr. Rajesh S. Gokhale, the current secretary of DBT, and many of you would know he is the lead architect of the Bio E3 policy of the government, which Honorable Prime Minister announced in his Red Fort speech last Independence Day. So, and as if you looked at the policy, you would realize the seeds were sown many years back in NII. I'm sure the NII influenced Dr. Gokhale's vision. And this one. Many of our alumni are also holding senior position in academics, industry, and government sector. That includes Dr. Rajiv Raghuvanshi, the current DCGI, and of course, he would also join, and many senior officials are joining for the panel discussion this afternoon. It is needless to mention that the success of NII is not only because of our faculties and research scholars, but also our extremely dedicated technical, administrative, and support staff. I also thank Secretary and senior officials of DBT for their generous support to NII in all our endeavors. We begin the celebration with the Foundation Day lecture, which is very special to NII fraternity. I must thank Professor Balram for accepting our invitation for this talk. When I wrote to him, I was not sure if he would be available, but his response was the most encouraging. So I quote here. So it would be my privilege and pleasure to give the lecture at NII on Foundation Day and the event to honor Professor G.P. Talwar, a truly inspirational scientist. So that speaks and all the participation. So I won't take any more time. Thank you all. We look forward to the excellent Foundation Day talk as well as Banya.
Thank you, Dr. Mohanty. May I now request Dr. Gokhale to please join us stage and share a few words with the House.
Well, namaskar and good morning to all of you. I think such a wonderful occasion that I couldn't keep myself away, not coming to this morning get together. That is doubly special for me. One, obviously, Professor G.P. Talwar enters a milestone that is very unique of its own kind. And secondly, my mentor, Professor P. Balram, who is actually, as Dr. Mohanty talked about, is giving the Foundation Day lecture, which hasn't done it for 25 years since I've been here, actually. It's a very, very special occasion for me to be able to hear. Also, it's also to find, all the NII past and present people. Actually, I can see many of you here, actually, and so wonderful to see all of you here. I think these Foundation Days are amazingly nostalgic at times, and this is even more special, actually. Let's try and recollect, how NII would have been set up, actually. In early 1970s, immunology was emerging as a frontier area that was actually linking various emerging areas like infection, cancer, autoimmunity, and India lacked a dedicated platform. And this is obviously learned through books that I have read about regarding NII's Foundation Day, because you can imagine I am almost half the age of Professor Talwar is, actually. So we're not almost there, actually, to really look at the historical point of view. But I think India lacked a dedicated national platform. And I think this NII thus provided a fantastic institution with a strong base to inculcate these ideas and fundamentals about it. And this is how NII has been trying to emerge out as a fundamental immunology institute, actually connecting with intersecting with national health priority policies. And one of the first programs that was set up was vaccination for birth control, which was the flagship program of Professor Talwar and others, and several of people at NII actually participated in this fantastic effort. I think it fills me with great joy and satisfaction to see what the outcomes are today, actually. An institution that was an idea at that time is now a thriving center of excellence with several wonderful things, leaders and scientists that have emerged out of it. And we hope that what Professor Talwar has set up actually continues to inspire and build the future of our country in a much better way than what we can think of in the future of the country. As I said, Professor Balram was my mentor, was my guide as well, and a mentor has been mentor. And I think what I have really imbibed from him is the intellectual rigor in most of the activities that he's really stood for his life. I think he's a rare combination of clarity of thoughts, intellectual depth, scientific excellence, with the moral courage, actually. And that is very, very critical and important, I think, in given circumstances, the way everything is happening and the geopolitical activities that happen. And I'm trying to see if I can imbibe some of what he's taught me over the years of 6 years of time that I have been in his lab, learning peptide and protein chemistry, biophysical chemistry, and which has far-reaching influence on various aspects of molecular design and biological functions. I think one thing that he taught to me was very, very important. Leadership is not about authority in science, actually. It's about creating people and environments where people continue to thrive. And I can tell you that leadership at DBT, right, at NII as well, with Dr. Mohanty there, actually continues to inspire many younger generations and motivate them to really take this forward, actually. And I'm very happy to see my dear friend who was also around the same time at IISc Bangalore doing his PhD, doing fantastically well for an institution. I think immune system is no longer just about defense. It connects metabolism, aging, and brain and environment. And I believe that's where the immunological bandwidth is continuously expanding. It's growing in the structure. And with the inclusion of, as Dr. Monty talked about, data and AI, I think this is going to be absolutely stunning how this will define the future course of precision healthcare, which is what all countries are trying to look forward to, develop different models. Can we develop integrative models where rather than looking average population-based data to try and understand the health of the individuals, can you do single human data building actually analysis and be able to do this? As India advanced in biotechnological forum through BioE3 policies, like one of the BioE3 policies and various other modes and activities, and as we become stronger and stronger in collaborations and putting together, putting biological compassion into our ecosystems excellence, I think we have to ensure that the role that NII plays would be a very, very transformative even further in terms of what it is doing. I personally believe this is the best time to think about the future, actually. You can imagine what people are talking about right now, building data centers below the oceans, or building data centers in the space stations, or doing biomanufacturing in space. You just imagine, and things are feasible right now. It's not too far off, actually. It's about a decade. You can see many of these things happening where you may have data centers situated in the space. The way satellites gives you the data for weather control and other activities, maybe the transmission of data will happen through there, actually. So clearly, institutions have to continuously rewire and reform and rebuild, actually. And I think DBT's institutions, I must very proud to say, are at the forefront of all the frontiers and have fantastic leadership and people who are, driving these systems and organizations. And I think this is the great time to what our minister talks about biorevolution in the country, actually, and in the world, of how this will transform everything that we eat, live, survive, or even the climate-related conditions and all. So it's all great stuff for future of biology and biotechnology and immunology. And I think that's where I would again congratulate all the NII members, all the past and present members who are here. My heartfelt congratulations on the Foundation Day from the DBT to each one of you. Thank you very much.
Thank you, Dr. Gokhale. Your words have made this even more special for us. It is now time to begin the scientific celebrations with the Foundation Day lecture, a tradition that brings together thought as well as discovery. And I invite Sonanjali, our young researcher in her 5th year of doctoral studies at NII, working on structure-based discovery of small molecules against tuberculosis, to introduce Professor Balram to the House.
Thank you, Dr. Banya. Good morning, everybody. It is an honor for me, really, to stand here and introduce to the House a man who's not only shaped Indian science, but also the way we think about it. Introduction to this audience, so these words are not so much an introduction as a celebration of his remarkable journey. Professor Balram completed his graduation in 1967 from Pune University, post-graduation in 1969 from IIT Kanpur, and he obtained his PhD from the Carnegie Mellon University, the United States of America, in the year 1972. He was a research associate at Harvard University from 1972 to 73. He served as a faculty of the Molecular Biophysics Unit, Indian Institute of Science, Bangalore, from 1973 to 2014. He was the director of ISC from 2005 to 14. He's been associated with the National Center for Biological Sciences, Bangalore, since 2017. Professor Balram's research has spanned the frontiers of molecular biophysics and chemical biology, with pioneering work in the structure, folding, and design of peptides and proteins. He's the recipient of several awards and honors, including the Padma Shri, the Padma Bhushan, and was awarded the American Peptide Society's R. Bruce Merrifield Award in 2021, and the Ernesto Scopone Prize of the Italian Peptide Society 2024. He was the editor of Current Science from 1995 to 2013, during which he authored over 300 editorials on diverse subjects related to science and scientists. He's never hesitated to speak his mind about ethics, about quality, about the culture of research, and about the responsibility scientists have to society. Most of us would know this, but Professor Balram was here during the Foundation Day in 1989 when Professor Talwar had invited him. And here we are, 36 years later, celebrating his legacy with both of them amongst us. It is a starstruck day for everybody, for me. And this one is a little special for me because when my PhD mentor joined his PhD, Professor Balram was his department head. So it feels surreal to have generations of scientists present in this room and reminds me and all of us actually of the responsibility we hold towards our society, the science of our country. So on this Foundation Day, it's a privilege to have you amongst us, sir. Please join me, everybody, in warmly welcoming Professor Balram to the stage. Thank you.
I was going to come up here and pretend that I was young. But Sunanjali sort of preempted me and took me back in time. I've been around for a very long time. And the very fact that I came and gave the Foundation Day lecture here in 1989 at Professor Talwar's invitation, I've never forgotten that. Because it was on a few occasions when I was young when I actually gave one of these lectures. And at the end of it, he gave me a silver glass, which I kept with me, there with me even today. And I've never forgotten this because there were only 3 senior scientists who invited me shortly after I had joined the Indian Institute of Science in 1973. One of them was Professor Rashima Chatterjee in Kolkata. The other was Professor Ubaid Siddiqui at the Tata Institute of Fundamental Research in Mumbai. City's changed its name since then. And the third was Professor Talwar. And today, when I've come here, I'm really come to pay a tribute to Professor Talwar, talk to you about the subjects in which I'm interested in today. But I must begin by really with this slide, where I would urge those of you who haven't read this to read a very short account, a personalized account in an interview which appeared in Nature Medicine about Professor Talwar's attempts to develop the contraceptive vaccine, the immunological approach to contraception using human chorionic gonadotropin in the 1970s and the 1980s and beyond. I've just listed 2 of Professor Talwar's papers from that period. One which appeared in 1976 in PNAS, and the other one which appeared in the Journal of Reproductive Immunology in 1982. Why have I listed these papers? This is because in 1976, the field of peptide and protein chemistry was not quite what it was. That's the field in which I worked. It turns out that in the mid-70s, probably around 1976, Professor Talwar had written a letter to one of my old teachers at IIT Kanpur, Professor D. Balasubramanian, who later became the director of the Center for Cellular and Molecular Biology, asking him for some help in understanding some features of the HCG molecule. Professor Balasubramanian at that time had unfortunately had an accident. So he wrote to Professor Talwar saying that there's a young student who's joined now the faculty of the Indian Institute of Science, and maybe you can write to him. He's setting up a laboratory. Professor Talwar was never hesitant to seek collaboration wherever it was available, except that when he wrote to me, I was completely taken aback because he was such a senior person, established, head of the Department of Biochemistry at the All India Institute of Medical Sciences, now writing to a newly minted lecturer in the Indian Institute of Science. So I was overjoyed. I said, somebody even knows me. He actually didn't know me, but he had heard from Professor Balasubramanian. So along came his student, Ramakrishnan, who arrived in my lab. I knew nothing about HCG, and I suspect that Ramakrishnan also knew relatively little about HCG at that time. But he brought along a poster, and on that poster was that sequence written there with those disulfide connectivities that you see there. So I was very happy because I was interested in making peptides. I was interested in peptide chemistry. I also realized at that time that Professor Talwar had an extraordinary ability to look ahead. He had already recruited one of the very, very few people in India at that time who was doing solid phase peptide synthesis. In fact, probably I would say the only one at that time until Shivanandaya in Bangalore University began. And that was Dr. K.S.N. Iyer. And Iyer began solid phase peptide synthesis. And at that time, this was unknown in India. It was largely unknown in the United States, and it was very difficult to do. And but they began to make peptide fragments of human chorionic gonadotropin, and it was the HCG peptides which were intended to make the first of the peptide vaccines. Peptide vaccines became important in later years, but to think of this in the mid-1970s was really far-sighted. So there we were. There were the disulfides, and when I looked at these, I was very interested in making small peptides with disulfide bonds in them. So I picked the sequence over here and began to make a peptide of it because I had a problem now. It then turned out, what I didn't realize, because my protein chemistry was not as good then as it is now, I didn't realize that the disulfide bonds were not established in HCG at that time. That came many, many years later from O.P. Val's group, in 1981. So effectively what I was making was the wrong molecules, but that led me then to another field. That is the field of redox active disulfides, which were then discovered in the thioredoxins at the same time. Today, of course, thioredoxins, glutaredoxins, thioredoxin reductase, all of them are very important molecules. But I entered redox active disulfide group only because Professor Talwar was interested in HCG. I always followed the work on HCG because it was actually an exercise in peptide chemistry, which was very difficult at that time. This was the immunobiological properties of a 53 amino acid peptide subunit of HCG. This was in many ways a technical tour de force. It was very difficult to characterize peptides in those days. Remember, there was no mass spectrometry and there was no soft ionization method for taking biological molecules into the gas phase. So characterization was difficult, materials were generally poorly characterized. It was only in later years that this remarkable cysteine knot motif was discovered in the growth factors, epidermal growth factor first, and the many other growth factors later, and then finally in HCG when Wayne Hendrickson solved the structure in 1994. You can see 20 years before the structure of HCG really came into the literature. And why I use this paper here is that this is around the time that the people whom you have in the audience today as the Secretary of the Department of Biotechnology and as the Director of the National Institute of Immunology were still students. And the result of this is you must conclude that Professor Talwar was far, far ahead of his time when he was doing this. Many of the techniques that we have today did not exist. Many of the characterization methods did not exist, but he was undeterred as far as doing the problem was concerned. And I think you will hear later on the many efforts to isolate HCG, I'm sure, from natural sources, which is the urine of pregnant women. And very, many fantastic experiments which I didn't know much about, but I heard about them from his associates. Dinkar Sahal later came and worked in my laboratory. Ramakrishnan was a friend for a while, and K.S. Nair, I always looked up to him as a peptide chemist. And it was my pleasure many years later when I became more influential to pull Dr. Iyer out of retirement and give him the Lifetime Achievement Award of the Indian Peptide Society, because he did really begin this field in India. But why was Professor Talwar doing this? This I thought I would put as the connection with what I'm going to talk about and what Professor Talwar's work really was about. Why was he trying to control the population at that time? Why were they interested in contraception? Remember that Carl Djerassi had introduced the steroid contraceptives in the 1960s, the oral contraceptive. Pregnancy is not a disease. Nevertheless, overpopulation can be considered as a disease. It affects human societies. The first problems in this were discussed in Malthus's famous essay on the principles of population, dating back all the way, this is a later edition, it dates back to 1798, where he wrote that population tends to increase geometrically, while food reserves grow arithmetically. So very soon, the world's population will outstrip the food reserve. This was discussed for a long time. Today, of course, in the 21st century, there are discussions why Malthus is still wrong, why Malthus makes for bad science policy. I think Malthus was not completely wrong if you look at all our cities today. Overpopulation is a major problem. Population control is important. And why is this so? It's not only food. Today, the Department of Biotechnology might tell you that the next great advance in biotechnology will solve the problems of food. Yes, it will solve the problems of food. But remember, how did you solve the problem of food in the first place? With the Haber synthesis of ammonia. If the Haber synthesis of ammonia had not been possible, you would not have had urea as a fertilizer. But what happens when you do this? You put carbon dioxide out into the atmosphere because even after 100 years, there is no biotechnological approach to producing ammonia from nitrogen. The result of this is that climate change has become a problem. So every problem is linked into every other problem. Remember that technological advance is not necessarily the solution to every human problem. Therefore, one must look at these issues as far as policy is concerned somewhat carefully. Of course, today you will find figures like this, that eventually the population will level and then it will go down. But when the population goes down, you create other problems. But that is because we have an economist view of growth, that we must be constantly growing. This is what takes me to the subject of my Foundation Day lecture. And here I must begin by showing you my next slide, which has both the title of my lecture and what I'm really going to talk about. I'll also warn you and ask Professor Mohanty to stop me in case I run over time. There is now, we are already a little bit late for starting, and here I am. My title has evolutionary inventions and innovations. It should not have been there, but I deliberately put it there because I want you to think of these two words, invention and innovation. Invention has not been used so far in anything that I've heard, but Dr. Mohanty already used two other words. He used the word discovery, I was listening carefully, and he used the word innovation. Today, the most widely used word in circles of science and technology is the word innovation. Now, I is there everywhere. It is there in BRIC. It is there, I found in ACSIR, where I'm the Chancellor. And I think it is only a matter of time before the Ministry of Science and Technology becomes the Ministry of Science, Technology and Innovation. Policy is already STI. Now, what is discovery? Discovery is something that human beings do. And invariably they do it by accident. Serendipity is the driving force for discovery. And you can't actually plan serendipity at all. Invention, on the other hand, is something more purposeful, where you want to solve a problem and you invent something which solves the problem. Could be the light bulb, like Edison. He illuminated our lives after having made that invention. But what is innovation? I leave you to think about what is innovation. But what I'm going to ask is the question, where did biology come about? Why do we have this enormous diversity in biology? Where did everything start? How did life come upon Earth? That is the evolutionary question that one wants to ask. The two pillars of chemistry and biology are illustrated on this slide. Mendeleev on the left, Darwin on the right. Mendeleev gave us the periodic table, Darwin gave us the ideas of natural selection, adaptation, and popularized the word evolution. We've had a century of science in the 20th century. Think about it. We're now in the 21st. We've already finished 25 years of the 21st century. What did we learn in the 20th century? This is the time in which science has made its maximum advance ever since human beings came upon Earth. We began the 20th century with the quantum. That's Planck, Einstein, and the quantum. We moved through the first 25 years when quantum mechanics was invented by the human mind. And there's Heisenberg's paper, 1925, on the uncertainty principle. Heisenberg's paper is now being celebrated in its centenary this year. Now, why are we worried about quantum mechanics in a biological institute? We are worried because Dirac made this statement that the underlying physical laws necessary for the mathematical theory of a larger part of physics and the whole of chemistry are thus completely ignored. It's the whole of chemistry that I have underlined there, highlighted. Whole of chemistry means the whole of biochemistry. But from the mid-1950s, when Avery discovered DNA as a genetic material and Watson and Crick found its chemical basis, the chemical basis of heredity is nothing but the DNA base there. Information is stored there. That's the half century where biotechnology has grown and come to the stage where it is today. The 20th century ended with Craig Venter and Francis Collins finally shaking hands, announcing the human genome. President Clinton was present. What are the defining images of chemistry and biochemistry then? The defining image of chemistry is this periodic table. I show a periodic table which is more than 100 years old in one of the Italian universities which I photographed. All the constituents of matter here. This is a chart which used to be there in every biochemistry department, the chart of cellular metabolism. Both these charts still are there in all the old universities. Nobody ever looks at them. In all the new institutions, they have been banished. Blackboards have been banished, and so have these charts. But the metabolic chart here looks like a traffic map of Delhi or Bangalore or anything, full of jams or potential for jams. You can't afford any traffic jam on these. If you have any traffic jam, you then have disease. So I call this the highways, crossroads, streets, and bylanes of cellular chemistry. And cellular chemistry is extraordinarily complicated. It is the chemistry of the cell which makes the cell alive. Darwin, of course, said this wonderful, at that time he knew very little about cell biology. There was no biochemistry, it had not been born. But he was worried more about the diversity of species that he sees. And he says that from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. In fact, this is right towards the end of the origin of species, and this is where the word evolve actually appears. Whole book, you can't find evolve very much. Right at the end, Darwin uses the word. But today, if you think about the word evolution, we associate it most closely with Darwin. But here I will quote Jacques Monod as far as the definition of life is concerned, because Monod is one of the persons who greatly influenced Professor Talwar when he spent time in France. And it turns out that he says there are living systems, no living matter. And he says nowhere are the paradoxical properties of life present except at the level of the cell, not below this. Cells are to biology what atoms are to chemistry. And if you want to know why atoms are important in biology, you might do no better than go back and look at Richard Feynman's lectures on physics, volume 1, chapter 1, where he will decide, ask a question. He said if in some cataclysm all of scientific knowledge were to be destroyed and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? The cataclysm that Feynman was worried about in the 1960s is a cataclysm that we might well worry about even today. We have the most dangerous statements being issued in the press, in the public domain, from India, from Pakistan, from Russia, from America, from all the major nuclear countries of the world. So we are closer then to a nuclear conflict than we've ever been before. When I was young, it was always discussed. Now when I'm old, I find it's come back to the fore again. Feynman says if you destroy everything this way, and you want to start all over again, you might get a head start if you knew the atomic hypothesis. He answers his own question. So the idea that matter is composed of atoms. After all, what is science? Science is the study of matter. One branch of science studies inanimate matter. The other branch of science studies animate or living matter. So whether you're physics, chemistry, biology, geology, it does not really matter. The word evolution is probably the word that every educated person should now really try and understand. It applies to the creation of matter, to the creation of the universe, the earth, geological, chemical, and biochemical evolution, the rise of unicellular organisms, and in biology, species and natural selection, and last of all, cultural evolution, which is ongoing. Cultural evolution is something that happens in human beings, and this is happening because of the cognitive evolution. This is the timetable of evolution, because you need a time scale over which you're thinking. This is a grand time scale. The Earth was born about 5.67 billion years ago. This is now reasonably well known. We've now reached this point here. That's 567 million years. 5.67 billion by the time we get here. When did life evolve? The Earth, when it was formed, and this really is a subject for physics and geology, would have been most inhospitable for biology. Look at all the equipment that you have in your laboratories. You want to grow an organism, it needs the right conditions to grow. Where did the first organism really come from? Where did all this biochemistry evolve? That's the question that you ask. It turns out that it's taken a couple of billion years. Somewhere around here is where the first living organisms really came upon Earth. They were anaerobic Earth, so they would have been anaerobic unicellular bacteria. Today also you have anaerobic unicellular bacteria which you study in so many places, sometimes even inside your gut. There was no oxygen in the atmosphere for much of this time. Nearly 3 billion years there's no oxygen. So don't worry even about anything else. When did the first animals come? Only about 500 million years ago. The worms, the first of them. When did the land plants come? Much later. So the result is that we are talking about the evolution of chemistry here, and then the evolution of biochemistry before you get microbiology. So the subjects that you need to understand this begin with geology, move through chemistry, biochemistry, and then comes microbiology. Much later, zoology and botany. And even much later, after human beings, after the cognitive evolution, after modern science began, you worry about medicine. How many years do we look back when we have all the fights that we have today? Not more than 3,000 to 4,000 years. That's what everybody is fighting about. So you can see that on this time scale, every human division is a meaningless division. Biology teaches you one thing, that we are all descended really from a common ancestor. And that common ancestor is the last universal common ancestor, or the first single-cell organism which appeared on Earth, which learned the ability to reproduce. These are the three pillars of modern biology: genetics and evolution, connected by the Watson-Crick chemistry of DNA. What are life's imperatives? Biology is a bleak subject sometimes. Life's imperatives are only two: survival and reproduction. It's important that you survive until you reproduce. That is true of all animal life on the planet, of all plant life, everything. Human beings think they are different. What are the ingredients for life? There are only two. One is information and the other is energy. If you break this down, then of course you can ask the question, did Darwin think about any of this? Darwin did think about life. What he said in 1863, it's rubbish thinking about it. And then in 1871 he said, but if, and oh what a big if we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed. The word protein had entered the scientific literature, but nobody knew what proteins were at the time when Darwin wrote this letter to his friend. So to try and begin the details of what I'm going to talk to you about, I must make a case for chemistry, because you can't have biochemistry without chemistry. I was trained in chemistry, I moved into biology, and as a result, I've spent a very long career in which I have never been accepted either by the chemists or the biologists completely. But they have found it difficult to ignore me altogether. But when I was trying to make this slide on a case for chemistry, I found that you always turn to Google When I turned to Google, I found this image over there. I'd never seen this before. I turned to this image, and there it is. It's a conical flask. It's a conical flask, which is the standard symbol of science. So I clicked on this conical flask to see whether Google allows you to click and find out what it is. And sure enough, I found this. It says, meet AI mode, a whole new way to search. So here, artificial intelligence has as its icon the conical flask. And to give it some dignity, it turns out that I dug up Emil Erlenmeyer, after whom this flask is named, the Erlenmeyer flask, and his paper appeared in 1863. The defining image of science really is this flask. You will find that even UNESCO has it on World Science Day on its poster. It's an amazing thing when all laboratories are filled with such complex equipment. The case for chemistry, best made by Arthur Kornberg many years ago when he wrote an article. By then he was old, reflective, and he said chemistry is the lingua franca of the medical and biological sciences. You can't do biology, you can't do medicine unless you understand the underlying chemistry of the living organism. Chemistry cannot be reduced to abbreviations. Genes, proteins, all kinds of factors, they are real molecules. There's no getting away from chemistry. But today I think it's very important for chemistry to be understood by everybody, because current geopolitics is really being determined by chemistry. And this is why I thought I knew that this audience would not contain many chemists. And I must make the case. If you want to understand why the world is having all the problems it does today, you must remember that the molecule fentanyl is the one which has created the problem between the United States and Canada and between the United States and Mexico. And it is also part of the problem, not the only problem, between the United States and China. Then there are these elements of Mendeleev's periodic table, which are the ones which everybody wants. Mr. Trump believes it's there in Greenland. He believes it's there in Ukraine. And today you will find that Ukraine and Greenland are two territories which everybody seems to covet. And if we find some rare earth somewhere or the other in India, then maybe, I think now Pakistan is now trying to say that there are rare earths under Pakistani soil. So chemistry is important. Mendeleev's periodic table and the rest of chemistry really determines what happens in the world. Life has complexities, best exemplified by the metabolic chart. If I now go to energy metabolism, this is what everybody hates when they study biochemistry. Glucose down to pyruvate, citric acid cycle, mitochondrial oxidative phosphorylation, glycolysis. Everybody knows the terms, nobody knows the details. And no one likes it. So citric acid cycle, for example, I found this quotation on the internet which I like very much. Said, little kids love dinosaurs, bugs, and exploring the woods. Science doesn't scare them. They find it fun until 9th grade. For me, of all the fear-inducing horrors of 9th grade biology, the monster in the pack, the highest mountain of meaningless memorization, do you remember this? The Krebs cycle. Just imagine that. But the Krebs cycle is central to cellular metabolism. It is there in the most primitive bacterium, and it is there in ours. So if we don't understand it, and we have to ask the question, where did it come from? How did these cycles evolve? Every reaction in biochemistry on a metabolic chart is a substrate being converted to a product. And any chemist will tell you that if you want to convert something into something, you must provide it some energy. This is why if you go into a chemistry lab, you'll always be cooking something. The same thing happens in the kitchen. And you heat. If you don't heat, you shine microwaves into it nowadays in slightly more fancy looking equipment. How does nature do it? Nature provides catalysts. It needs enzymes. And then it has special ways of coupling chemical reactions such that the energy of an unfavorable reaction is now offset by the energy of a favorable reaction. Thermodynamics cannot be violated, even in biology. But if you need an enzyme, you need a protein. And if you need a protein, you need a gene. And therefore you need information. So the coupling between information and energy really can be seen in this little schematic over here. I'll show you one which tells you a little bit about the complexities of biology, of how life has evolved on Earth. Every 12th class student now knows this. Everyone's taught photosynthesis. No entering PhD student in any of the institutions that I've seen is generally able to answer any sort of question on photosynthesis except to say that carbon dioxide now goes into carbohydrates. So for some magical way, plants manage to fix carbon dioxide. It is magical because we have only one energy source which we think is infinite. It is not, but the sun more or less has an energy source which is going to certainly last our lifetime and thousands of lifetimes after us, so we don't have to worry about it. This is the process of photosynthesis, carbon dioxide to carbohydrate, and in that process of photosynthesis, you can see what happens is that the energy of sunlight is used to take hydrogens from water and transfer them to the carbon atom there. So organic chemistry is born from inorganic chemistry. There are no carbon-hydrogen bonds in carbon dioxide. There are carbon-hydrogen bonds in the carbohydrates. But in this process, a remarkable reaction happens. Water converts into oxygen. That is, the hydrogens are stripped away from the water molecule, and you're left with oxygen. So you get molecular oxygen. This is the process by which, this is nature's major invention in recent times. And when I say recent times, 2.5 billion years ago. On the Earth's time scale, that's fairly recent. This is the invention of photosynthesis. This is the oxygenation of the atmosphere. But once oxygen comes, oxygen is an electron acceptor. So it can be used as an electron acceptor in mitochondrial oxidative phosphorylation. So today we all know this, that oxygen is the acceptor in mitochondrial oxidative phosphorylation, but it's really a secondary role, because you can have other electron acceptors which accept the electron. So that is respiration. Respiration is burning, and you see you've gotten back to water again. So these two cycles which you have here, they're complementary. So animal life and plant life on the planet now really recycle carbon atoms in a remarkable way. And in this process, one produces carbon dioxide, the other one produces oxygen, and both use it. So they're both in a living relationship with one another. This is water oxidation, this is oxygen reduction in respiration. Nature's sustainability is completely embodied in this equation. If there were only plants and animals, nothing else, human beings remained animals. Remember that human beings are animals even 1 million years ago. They belong to the animal kingdom, animalia. That's it. They hunted, they reproduced, and they died. The only thing which has now really advanced human beings is the development of the brain. And those of you who have read the many obituaries to Jane Goodall in the last couple of days would have immediately seen that she has in fact mentioned multiple times in many interviews that the difference between the chimpanzee brain and the human brain, the genomes, is only 1%. And it's already clearly seen there that in that 1% of the genome, chimpanzees have already acquired many qualities. They can in fact recognize something like 500 hand signs. And this is the same number of hand signs, for example, that somebody who's not able to speak or hear from birth also learns to use. So the development of the brain, the evolution of the brain, the evolution of cognition is that last step which has brought us to where we are today. All the chemistry has already been done by that time. More carbon dioxide going into the atmosphere because of human activity leads to climate change. This, of course, is the greenhouse effect, first recognized by Arrhenius a long time ago. Can we improve nature? Biotechnology always asks the question, can we improve nature? Can you imagine if you could fix carbon dioxide better, you don't you could do it much more efficiently, capture it. You can't because Rubisco, the enzyme, ribulose bisphosphate carboxylase, cannot be engineered better. This is the 2025 paper which has tried very hard to improve on evolution. Many times you can't improve on evolution. Look at this molecule here. I know that many of you don't like chemical structures, but even chemists don't like this stuff. Because this is vitamin B12 or coenzyme B12. And this is the most monstrous molecule that you can get as a natural product. And it's also the first known organometallic substance in nature. It has an unstable carbon-cobalt bond here. The reactions which coenzyme B12 catalyzes are difficult to do in any chemistry laboratory. They cannot be done anyway. So B12 must have come at a very early stage in evolution, because anaerobic bacteria have vitamin B12. Today, there's a cell-free system with 36 enzymes which lead to the biosynthesis of B12. All of them have been cloned, and you can produce B12 in minuscule amounts in the cell-free system. Fortunately for us, our bacteria produce it in much more abundant amounts. Every bit of chemistry in nature is monstrously complicated. This is the oxygen evolving complex of the photosynthetic system. And you can see that combination would horrify any inorganic chemist. The reduction center, oxygen reduction center in cytochrome oxidase is similarly complicated. It is hard to mimic these. Now you can then ask, with all this complexity, is there a common chemical model for life in the universe? These questions have been asked in the 21st century by those more imaginative chemists who worry about this. Where did all this come from? How did this evolve? It's not very easy to speculate on this, but Alice in Wonderland comes to your rescue here, because the Queen in Alice in Wonderland is asked, and she says, why sometimes I've believed as many as 6 impossible things before breakfast. So in trying to decipher, work backwards on evolutionary chemistry, sometimes we have to imagine the near impossible and hope that it was possible at some time or the other. This now is a different kind of evolution, this historical evolution. This is the timeline of biochemistry. I had begun in 1875 and ended the present day, 150 years. Immunology did not exist at that time. And it turns out it was disease which existed, infectious disease. The work of Pasteur, Koch is what drove the microbe hunters, immortalized in this book by Paul de Crève. This was the cause of infectious disease, and microbial pathogens were described in this period. Later we turned to disease, which was nutritional disease, which we were aware of. And the nutritional disease gave rise to the vitamins. They were called the vitamin hunters. But what do the vitamins do? Nobody knows what they do. It turns out they're coenzymes. But enzymes are not really known. Between 1950 and 1975, when I began, everybody was hunting enzymes. By this time, Sanger had begun the sequencing projects, and it turns out that the next 25 years were gene hunters. Now, in an advanced institution like this, one which is at the forefront of biology, I'll leave you with the question. 2025 has already arrived and about to end. If any of you have an idea of what I should put onto that slide now, what has been the major pivotal advances of the last 25 years, which match with these, which I say in generality, please do write to me. I do not know. Photosynthesis starts somewhere here, and that's when the oxygen level in the atmosphere goes up dramatically. And I'll show you a very recent paper connected to this. The first eukaryotic cells appear around the time oxygen appears. Maybe a little bit before, a little bit after, but around the same time. The idea of the transition from unicellular to multicellular organism now is the endosymbiotic theory, now attributed to Lynn Margulis and this famous paper. Look at the title, On the Origin of Mitosine Cells, 1967. Sent to 10 journals, rejected from 10 journals, eventually published in the Journal of Theoretical Biology. But this really, for students, I must say, this is the origin of the endosymbiotic theory of eukaryotic evolution. There's oxygen. I've expanded the time scale. What should have come before? The Krebs cycle should have come before. Chemiosmotic oxidative phosphorylation should have come before. Biological membranes must have come before. Complex molecule like vitamin B12 must have come before. And the interesting thing, if you look at the vitamin B12 structure and you look at the heme and chlorophyll structures, they are tetrapyroles, but it's actually vitamin B12, the more complex molecule, which came first. Heme and chlorophyll came later. They are needed only for photosynthesis and for oxygen transport in blood. So this is needed for metabolic chemistry, to carry out carbon migration reactions which cannot otherwise be carried out easily. The result of this is that the growth of oxygen in the atmosphere, the invention of photosynthesis, drives oxygenic biochemistry from this point onwards. Oxygenic biochemistry is very important. Where do you introduce oxygen atoms from the oxygen molecule itself? I show you only 3 examples. I show you cholesterol here, the collagen molecule here, and lignin here. Why collagen? Why lignin? Collagen is what makes up all your tissue. Lignin is what makes all the wood on all the trees outside. No plant or animal can grow without material. These are nature's biomaterials with which it has constructed complex life. Cholesterol is the molecule that I'll take you back to, where chemistry is useful. One oxygen atom. You can ask, where did this come from? Water or oxygen? That question was asked by his postdoctoral supervisor to Konrad Bloch. 20 years and a Nobel Prize later, he had discovered the biosynthesis. He had worked out the biosynthetic pathway for cholesterol. And the biosynthetic pathway for cholesterol and the steroids is remarkable. Squalene, a terpenoid found everywhere, suddenly in one step, after this enzyme, squalene epoxidase, gives rise to this tetracyclic terpenoid structure, which is the basis of cholesterol. Why is cholesterol important? Because cholesterol is important because the steroid hormones are formed from cholesterol. No sex if there is no steroid hormone. Membrane fluidity is controlled by cholesterol. The bile salts derive from cholesterol, and vitamin D, bone, derives from cholesterol. The cholesterol is central to biochemistry, but it requires oxygen. So oxygenic biochemistry is critically important. In collagen, it's the hydroxylation of proline which gives collagen its mechanical strength. In lignin, the entire phenolic polymerizations are oxygen-mediated polymerizations. And those of you who look at biosynthesis might look very carefully at cholesterol. Eventually, cholesterol has to be shaped for its function. And in order to be shaped for its function, methyl groups have to be converted into carbon dioxide. How do you convert a methyl group to carbon dioxide? Elementary chemistry will tell you oxidize it to an alcohol, oxidize it further to an aldehyde, oxidize it further to a carboxylic acid, and then decarboxylate it. Remove the carbon dioxide. Effectively, you burn the methyl group. This can be done only if you have enzymes, and that is where the cytochrome P450s really came from. I put these references in because if there are some students, they don't like to look at anything in the 20th century. The 19th, of course, is forbidden. So 21st, you have to go. And sometimes even by the decade. And you must put a reference to Twitter or somewhere like this sometime. But here, of course, Roald Hoffmann, a chemistry Nobel laureate, has written this, dioxygen. What makes this triplet diradical kinetically persistent? Today, when I see Professor Talwar, I see myself, we are aging. But then, when we look at you, all of you are aging. And all of you are aging at more or less the same rate. This is largely because of your exposure to oxygen. You need oxygen, but you sometimes don't need so much of it, or you can handle it only for a certain length of time, maybe a few decades. He says we can burn, and not just with passion, but of course we do not burn. That is what is meant by kinetically persistent. It's a thermodynamically very unstable molecule, but it doesn't react. That's because it's kinetically persistent. This is from a very recent paper which has just appeared in Nature, where the geophysicists and the geochemists have really worked out magnificently using fantastic techniques of isotope dating, the oxygen in the atmosphere. And the oxygen in the atmosphere, you can see, has these bursts. There's a little bit wet photosynthesis, and this is why I use the word invention. Nature invented photosynthesis. And that's driven for a while. And eukaryotes come around this time when there's a grand oxygenation event. Bacteria can't produce enough oxygen. You need the land plants also to begin coming, and trees need to grow, and then only the oxygenation in the atmosphere would reach its maximum levels. So we've reached this. But you can see eukaryotic development has taken a long time. So those of you who are interested in parasites might sometime find that some have begun evolving, splitting off from the initial eukaryotic cellular ancestors before animals and plants come there as hosts. And that's, you might ask this question, where did the coronavirus come from? Where do viruses come from? Do they belong to chemistry or they belong to biology? So there you are, the molecules for by B12, chlorophyll, complex. You can see similarities. There's anaerobic biosynthesis, there's aerobic biosynthesis now. Today, and this is a fairly recent paper on B12, every little structural feature in B12, every unit of B12 module is involved somewhere or the other in these many, many reactions of cellular metabolism. So nature's extraordinarily economical. It's taken bits and pieces everywhere to construct these and construct its metabolic pathways. Asking the question, why should we have biochemical cycles? When did biochemical cycles actually evolve? These are questions that will help us in understanding how life will evolve. But lastly, energy is most difficult to produce. And the only way you produce energy is by oxidation. And what is oxidation? Oxidation is really the process by which you strip away, in a hydrogen atom, you strip away the proton and the electron. That's what is oxidation. You separate the proton and the electron goes down the electron transport chain, the protons are pumped to one side of a membrane. That generates an electrochemical potential. And this is the idea that Peter Mitchell introduced in the 1960s and early 1970s, the so-called chemiosmotic hypothesis, where you have energy transduction, electrochemical energy, to drive the synthesis of ATP. In this, oxygen is only a passive electron acceptor. The trick is the protons. Chemiosmotic theory is not very well understood even today. And this is an assessment at the end of the 20th century on Mitchell. Mitchell received the Nobel Prize in 1978, remained a controversial character till the very end. But he says that there is no more cautionary tale in all of biology than that of Peter Mitchell and the discovery of chemiosmosis. And in this assessment it says, not since Darwin and Wallace has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, and Schrödinger. This is, in fact, one of the central ideas of biology, how cells make energy. Energy in the form of chemical energy. This is Mitchell's 1961 paper. The only figures in Mitchell's 1971 paper are these two hand-drawn figures. But what is the central idea here? The central idea is that if metabolism can result in transport across a membrane, then transport across a membrane can also result in metabolism. And thus he says, thus we might be inclined to recognize that transport and metabolism, as usually understood by chemists, may be conceived advantageously as different aspects of one and the same process of vectorial metabolism. This is, I think, one of the most fundamental ideas of trying to understand the basis of cellular energy transduction. Today we know that nature's done this with these electron transport chains. All kinds of structures are available. Electrons hopping, because the separation between these iron-sulfur clusters is exactly matches the wavelength of the electron. So you can have tunneling from one structure to the other. Albert St. George, the discoverer of vitamin C, said very nicely that life is nothing but an electron looking for a place to rest. That's what you do when you breathe. Why I harp on respiration is really this, because 3 or 4 years ago, a chief minister of one of India's states really went public and said that the cow is the only animal which breathes out oxygen. And a newspaper cartoonist then drew this wonderful cartoon. We had a shortage of oxygen in the intensive care units during the pandemic, and then he showed patients being linked up to cows to now sort of get them out of trouble. It is important that even a generally educated audience needs to have some understanding of the principles by which a life on Earth really survives, has come about, and so on. The iron-sulfur clusters that you see in proteins are exactly the same iron-sulfur clusters that you have in minerals. And these minerals are also found deep sea, where there's a lot of sulfur, there's a lot of hydrogen sulfide, high pressure, temperature. So one can begin, and people have begun looking at the transition between inorganic chemistry and organic chemistry, that early catalysis would have come long before the evolution of proteins, and the possibility that inorganic membranes can sustain electrochemical potential and lead to the synthesis of energy-rich molecules. So there we are, our common ancestor, the last universal common ancestor. Biology is a unifying subject, and you can see that human beings are not far from many other species in biology. Bronowski, in his Ascent of Man, really says, series of inventions by which man from age to age has remade his environment, the different kind of evolution, not biological but cultural. I call that brilliant sequence of cultural peaks the Ascent of Man. And that's what has happened over the last 2,000 to 3,000 years, where one invention after the other has driven technology. There are many cultural features which might drive biological evolution. The problem of lactose intolerance is one of them. I won't go into this, but many of you will know that genetic analysis now reveals that North European populations are much more tolerant to milk than, say, South African populations, or South Indian populations less tolerant to milk than North Indian populations. This is largely because of the invention of dairy farming, of the use of milk as a human food, because no mammal was conceived by biology to drink milk after weaning from the mother. It's only human beings. And the result of this is that over time, human beings have already carried out over a period of 30,000 years or so since the invention of agriculture, have already carried out an experiment which many of you carry out in the laboratory. Just like antibiotic resistance breeds resistant bacteria, what we've meant is we have bred human populations who are now lactose tolerant because they have resistance of the lactase gene and they're able to digest milk. Otherwise, our early ancestors would have died in large numbers by diarrhea, dehydration, in the days when oral rehydration was not even on the horizon. History and biology is my conclusion. Nowhere can you read this better than by Israeli historian Yuval Harari in his book *Sapiens*. He says, from the cognitive revolution onwards, he says that's the point when history declared its independence from biology. He says from this time onwards, biological theories cannot explain how we've come about. Not enough to comprehend the interaction of genes, hormones, and organisms. It is necessary to take into the account the interaction of ideas, images, and fantasies. In fact, it turns out that today, human beings are probably being driven more by the fantasies that the human brain has conjured up, rather than by hard biological imperatives. Where will this all end if I push this further? A long time, the astrophysicists know when it will end. That's when the sun dies out of energy. And when that happens, there's no more photosynthesis. That's the end. That's a long time coming. It's about 5 billion years. And Katie Mack's book is very interesting. He says definitely, of course, now thanks to science, you know the answer. The question was, will it end in fire or will it end in ice? We still really don't know, but she says definitely fire. In 5 billion years, what will happen to the sun? Because we know what will happen to the stars. But then there's the poet who says this. Some say the world will end in fire, some say in ice. From what I've tasted of desire, I hold with those who favor fire. But if it had to perish twice, I think I know enough of hate to say that destruction, ice is also great and would suffice. So I think Robert Frost should in fact have the last word. My last two concluding slides are on the subjects which Professor Talwar might be more comfortable with, biochemistry. Because when I first met him, he was the head of the Department of Biochemistry at the All India Institute of Medical Sciences. Sidney Brenner, one of the most acerbic commentators of modern times on biology, said this. He says two things disappeared in 1990. One was communism and the other was biochemistry, and that only one of these should be allowed to come back. Brenner, of course, at that time had not come to Bengal then, and to Kerala even now. But I suspect if he comes back again after some time, he might find it still thriving. He says we do not have to resurrect biochemistry because it provides the only causal understanding of biological mechanisms. And therefore, biochemistry is very important, and that's the subject to which Professor Talwar really has devoted much of his life. And last, from the man whom he considers an inspiration, Jacques Monod, says biology occupies a position among the sciences at once marginal and central. Marginal because the living world constitutes but a tiny and very special part of the universe. It does not seem likely that the study of living beings will ever uncover general laws applicable outside the biosphere. This has disappointed physicists very much. But if the ultimate aim of the whole of science is indeed, as I believe, to clarify man's relationship to the universe, then biology must be accorded a central position. That is because science itself, or the study of science, is a human invention. And therefore, animals are happy without science, or human beings have invented science. And therefore, the purpose of science then is to understand the purpose of human beings in nature. Lastly, what I want to do is only to acknowledge the two institutions where I've spent much of my career, the Indian Institute of Science at the top left, and the National Center for Biological Sciences at the bottom right, which has provided me shelter during my retirement years. Finally, I want all of you today to acknowledge that Professor Talwar not only is the founder of this institution, but he has also been an inspiration for many of us who have entered the field of modern biochemistry in the 1970s. This field in India was built up by Professor Talwar and many of his contemporaries, who built also many of the laboratories today which are our leading centers for biological research. But they've also belonged to a generation which I think was steeped. It is what I would call the post-independence generation, which was driven by a desire then to make India and Indian science really come of age, and it is they who created the platform which many of us have used. I have been greatly benefited by many of Professor Talwar's contemporaries. Today they are not here, but I honor all of them when I ask all of you to rise now and give Professor Talwar a standing ovation.
Thank you, everyone. Thank you, Professor Talwar. Thank you, Professor Balram, for that very captivating journey. I now request Dr. Monty to please join me on stage. I request Dr. Pushkar Sharma to please join me on stage. And we would like to express our gratitude to Professor Balram for that extraordinary lecture. And a round of applause for Professor Talwar, Professor Balram. We're truly fortunate to have both of us with us today. A token of our gratitude for Professor Balram. All I can say at the last is thank you for inspiring us. We will now take a very short break for tea, and we'll try to be back here by 11:45 to start the scientific symposium, and session 1 is mycobacterial diseases. As you step out, don't miss a surprise waiting for you, a visual tribute to Professor Talwar, his legacy, his life. Thank you, and meet you all sharp at 11:45, please.
Can I put my slides?
Yes, ma'am, sure.
I'm speaking in the first session, the last speaker.
Okay.
ये एक तो अच्छा यहाँ पर है
Ma'am, do you want to check it?
अच्छा ये शीतल गंधर्व है ये इनका है ये देख लेते ना सब सेशन 1 में डले हैं
And without further delay, I would like to invite the chairs of the first session on mycobacterial diseases, leprosy and tuberculosis to start the proceedings of the symposium. We are honored to have with us Dr. V.M. Katoj, former Secretary, Department of Health Research, and Professor Javed Agrawala, J.C. Bose Fellow, Department of Biomedical Engineering, IIT Ropar. And with that, I hand it over to the chairs to start the proceedings of the next session.
Good morning, everyone. I see we're running late, so Dr. Vanya Kar didn't. She wanted everything to be very brief. But nevertheless, because we are celebrating the life of Professor Talwar, what he did actually, so many areas of work he did. But the tuberculosis and leprosy were heavily impacted by his work. Most of them, they revolve around the microbiome W. I remember, I published first in 1981 on the W. 1979-80 was the work actually. I think Dr. Rajani Rani started publishing in 80s itself. Again, Dr. Bidrama Mukherjee at that time. With then Professor Talwar also after that. Dr. Kiran published in late 80s, 88, it was the paper on which included the BCG and MW both. I think my co-chair again, has been working on the leprosy since mid 80s. So again, a long story with working together. Dr. Patil also belonged to that era. He started in Jalma, came back again to head Jalma, went back to Nimhans. So I think again the story of that time only. Two people, we can't call them juniors, they're the, I think, eminent people now. Dr. Benay Nandikuri and Dr. Sheetal Gandotra. They are the people who have been, certainly generations apart from us, but they have been contributing to the biology of the mycobacteria, immunobiology also, and they are trying to dissect the pathogenesis, pathology, I think in a very significant way, even in the modern times. But Syed Hasnain represents, the two generations. He started with us, and even 30 years later, he's again batting. He's still publishing on the tuberculosis. He's publishing on several areas. I think though that work starting in mid-90s, I think that multicentric project on drug resistance, which I had the fortune to work with him. And I think that led to sea change in the work on the epidemiology of the drug-resistant TB, then on the targets for drug-resistant TB. And I think that later is a history actually. But I'm very delighted he continues to publish. And he is the first speaker today. He is talking on the Professor G.P. Talwar's National Institute of Immunology, a unique place for nurturing excellence in research and creating future leaders of science. He himself has been a leader, heading several institutions, universities. I think he will be sharing his feelings, perspective on the subject. Hassan साहब 20 मिनट ही हैं सबको आज बिल्कुल yes.
Hello, can you hear me? No. Can you hear me? Now? Yeah. Thank you so much. It's a pleasure to be here. I don't find Professor Talwar here. I wish he was here. In our times, when we were just coming back to India and all, we were told in Indian science, there are three Brahma, Vishnu, Mahesh. All of us who know what Brahma, Vishnu, Mahesh means. The universe was created by Brahma, Vishnu, Mahesh, the three eminent gods of Hindu mythology. And the Brahma is only alive today. Vishnu, Mahesh are not alive today. I'm referring to Professor Talwar, Professor Verma, and Professor founder director of CCMB, who we also know more with us. That was the time when I was recruited here, and that also I'll talk more about it in the session. I believe it's late in the evening or tomorrow, reminiscences
so I will not talk about those things today, but tomorrow. Coming back to what I'm going to talk today, I owe my introduction to tuberculosis to my good friend sitting there. It was accidental because, we had a big lab, we had a large number of people. I see some of our students here in this group here. And it's good to be here again. And I went to the then director and asked him, I need more space. I have 25, 30 people are there with me, and I've already changed labs 2 or 3 times. He says on one condition, you have done enough on baculovirus, publish very well. Anything you want to publish, you publish in JVC. Do something which the humanity can benefit, that the public at large can be. Move to tuberculosis. And if you move to tuberculosis, I'll give you a big lab and I'll also give you a big grant. I need time. He says, take your time. One week later, I met him. And I said, introduction was more our friend sitting here. And I'm very happy that this switch over from baculovirus to tuberculosis was very good because, a lot of things we did created precedent for us. For example, today, everybody does only rifampicin resistance as a surrogate for all multi-drug resistance. This work came from us. We both published this paper, it became a classic. It was a part of the grant. We also showed then he had become the DG ICMR. I told him, Katosh, we get a lot of false positives in India and false negatives for TB in India. And our epidemiology work where we used to sequence, isolate from the community. And then I told him that, look, there are a lot of reasons that we are getting negative and false negative, false positive is because the tests that are used are based on antibodies for targets which are sequenced and found in the US. Our targets bilkul alag hai. Sequences are very different. And both of us were convinced, and he being the DG ICMR and Secretary Department of Health decided that okay, no more immunology based testing of TB. That decision also came because of our work, and like that many other things happened. So today I'll talk to you about the work being done in our lab. Actually, I must tell you, it is the wrong thing to say. Work is being done in the lab of Dr. Nasreen Intisham. She is sitting here. She was with me at the NII. She was my first PhD student and continues to be with us today. And she leads the lab at I have a lab at Sharda University, which is about 45 kilometers from where I live. And I spent 2 days in Sharda and 3 days in IIT Delhi. We have 3 areas of working. One is on basic sciences, clinical sciences, which continues in a big way, and of course translational. I'll try to give a brief flavor about what all we do. We just have the Brahma of Indian science coming here. Thank you, Prof. A lot of things were said about Professor Talwar. I'm trying to make a digression here because I thought he was a very decisive person and somebody who would also allow dissent to be noted, but then nothing against you if you dissent. A lot of things I have learned from him in building CDFD and building University of Hyderabad. And one day I went to him, I said, Prof, आपने मुझे प्रोजेक्ट साइंटिस्ट दे दिया, मुझे पता ही नहीं था प्रोजेक्ट साइंटिस्ट क्या होती है. हमारे साथ लोग हैं, वो कहते हैं तुम तो प्रोजेक्ट साइंटिस्ट हो, मैं तो कोर साइंटिस्ट हूँ. तू बैठ यार, तू बैठ, अच्छा ही बात है, तू बैठ. And then he gave me the story behind it. He says, you're doing so well, don't worry, अगले साल तेरे को मैं ऐसे ही बोलते, तेरे को मैं साइंटिस्ट 4 बनाऊंगा, और उसके बाद there's no limit, no end to that. I know you. Thank you, Prof. He will never take objections to whatever even strongly we say. And I was a young rebel, dabang the उस ज़माने में, could talk to anybody in any language without worrying about consequences or worrying about the age of the person I'm talking to. But that was learning from Professor Talwar. So the 3 things that we do in our lab are essentially looking at drug repurposing, basic sciences, clinical and translation, and all of them have endpoint: drug repurposing, vaccines and drugs, and biomarkers. And what we do we found long ago that, TB bacterium is a bacterium that has evolved very differently, very differently. Most times bacteria evolve by acquiring genomes from outside and growing stronger. TB bacteria actually follows what we call at that time as reductionist evolution, from non-pathogenic to it becomes pathogenic, very pathogenic, reduces the gene size. And look at this. This is non-pathogenic. This is the opportunistic pathogen, and this is the TB bacilli. This is a general rule, except one thing, and that one thing was a family of protein known as the PPP family of protein. Proline glutamic acid, proline glutamic acid. It's a gene family that the bacteria spends 10% of its genome. Imagine such a small genome, yet 10% goes to this bacterial family, this gene family. And that was another reason why lately after that we decided to work slowly on this, because it's a very difficult family. We could succeed a lot in this. So the only exception was this family, and the opportunists are this here. This is the normal case. The only exception to this division, what we see here, are pathogens which we now know as the Mycobacterium indicus prani. This is also very incidental. Three of us, myself, Professor Tyagi, I call them Tyagi brothers, they are not brothers. One is Akhilesh Tyagi, one is Anil Tyagi, both are in biochemistry, one is in plant sciences, one in plant sciences. And we decided, Dr. Manju Sharma was there, he said, Sayed, ek bhi organism India se sequence nahi hua hai. Why don't you sequence something from India? Will it be the first one? And we thought we should do what Professor Talwar had done work on, the leprosy bacterium. The bacterium that actually has been used by him, and I will not talk more about this or other, because there are two people who have actually contributed a lot, much more, and they are going to be giving a talk right after me. But one thing I'll only tell you, we sequenced this genome completely. The first genome from India, any living genome completely sequenced from India was this genome, and we decided to name it as Mycobacterium indicus prani. Indicus because it was discovered in India by Pran Talwar as a sole mycobacterium, and prani because Pran Talwar discovered this genome, and NII because much of the work was done at NII. I'll stop and move to the next slide. I will not talk more about it because we have two eminent people who are actually done. I will, next slide please. I can change myself. So I will not talk about the work. All I can tell you is that MIP has been used, and we now know why MIP is still being considered as for a trained immunity. Sometime later, maybe during coffee break, I can talk more about what is trained immunity, but most of you will know. In fact, MIP was also being tried. There was a project submitted to the government of India during the COVID time that they'll use MIP simply because of the very high component of trained immunity that it has. And these details are published, but there are two eminent speakers. So we will not talk about MIP at all. The work has been published already. And the other person from the DBT who anchored this work is also here today. How does the bacteria like tuberculosis bacteria survive the genome reduction? It does so by looking at very intelligent strategies. Time हो गया? हम डर गए यार. मैं डर गया क्या यार? इससे जल्दी कैसे हो जाए? So one of the lectures that I invited Bashelkar to our university in Hyderabad was, and he spoke about more from less for more, and I like this phrase. He was talking about can the industries tomorrow can generate much more with very less investment for a very large number of people. I said, oh, Jesus Christ, this is what the Mycobacterium tuberculosis does with whatever little we had done by that time. So it actually does much more by resorting through large number of things. These are some of them. Epigenetic regulation, and I'll tell you a slide about what is so unique about Mycobacterium. Disordered proteins. We were all taught that proteins when we were in school or early university or college. We had a concept of protein or an enzyme. It's a fixed molecule. Keys come in the protein, and then the function is so one protein, one gene, one enzyme. People got the Nobel Prize for that. But now we know that this is not always true at all. Many times it's not true. And of course, moonlighting or multitasking. One gene can produce hundreds of functions, one gene, and that's what this bacteria does. And of course, the PPPGRS activity, all from the concept of the shell curse. So abundance of MTAses, we found that there are totally 121 methyltransferases. Unbelievable for a bacteria to have so many methyltransferases. And we all know methyltransferases are epigenetic modifiers. They will modify the protein to do whatever function that they want it to be done. They will modify the cell, modify the lipids, modify everything possible. And mycobacteria has such a large number. And many of these are uncharacterized, 60 of them. They are sub-characterized and they could be DNA or protein methyltransferases. They are also RNA transferases. We have identified 15 distinct MTAses present only in MTB complex, nowhere else. And that's what we thought it would be a good thing to focus on, trying to understand, and if we can disrupt this, and one of the vaccines that we are trying to develop at the Sharda is, we call it Sharda vaccine, is making use of attributes of methyltransferases. And this is, we have also characterized a large number of these proteins. 9 are completely essential and 10 are virulence factors. So imagine the methyltransferases having this kind of a function in Mycobacterium tuberculosis. Disorder. If you look at the bacteria, 2% disorder. Archaean 2% disorder. Bacteria, 4.2% disorder. Mycobacterium has a very high, much higher than others, almost half of eukaryotic systems, which means the proteins are not ordered at all. They are completely disordered. And that disorder allows them, I wish Professor Balram would have been here today. He may have gone out somewhere. So this disorder allows them protein to do any other function. And in fact, the unstructured region, if you look at the disorder, majority of the disorders are in the PPP protein family. So that made life a little easier for us so that we can think what could be the role of PPP proteins are. And today we have published a very large number of papers. We continue to get invitations to write editorials in Lancet and others on the PPP proteins and others on Mycobacterium. Much of this has been published. Moonlighting function, which means moonlighting is a term used in good old days. The kings and the emperors, we used to go undressed as ordinary civilians in the night and meet the common man and ask them problem kya batao. And so can they do dual function as a king and also as delivering something for a common man. Multiple functions, same person. That's what moonlighting is. And now we have evidence that mycobacterial protein, many of them have different functions. Many of these have been published by us and others. And this modifying the host signaling pathways, acting as sporin in calcium uptake. And actually today, we have evidence that these PPP protein could also be involved in drug resistance. And something which we are working very strongly on this. And regulation of autophagy and apoptosis. Cell death is a very important thing, and mycobacteria uses this very elegantly, very opportunistically. I'll give you an example of that from the model we made based on the work we have done. And of course, in many other functions such as role in glycerol uptake and the efflux pump leading to bacterial resistance, including throwing drugs outside. This is one simple example where a protein which is known as P35 and PP68, they are both operonic. And one thing mycobacteria does for this class of protein is it doesn't use separate operon. One operon will transcribe 1, 2, 3 genes all together. After transcription, they'll be processed and translated into 2 different proteins. In some cases, they can even be translated as one protein, then cleaved into 2 different proteins. This we have published quite a few papers on this. This is a classic example when the protein, the PPP is a part of the 4 proteins complex from a single operon here. P35 is disordered, PP68 is disordered. Both are disordered. Disordered, this has TH1, TH2. We all know what TH1 can do or what TH2 can do. No change at all. Both are almost the same. But if it is together, you can see a large TH2 response and much less TH1 response. Imagine, just by simply associating together, it can do this. So it has multiple functions, and this is all published, so you can go through it, doing research. Cell signaling, interactions, regulation network, cell cycle regulation, binding, and so on and so forth. Many such functions it can do. This is a model which we've just trying to already communicated to a big journal, where we are talking about PGRS functions opportunistically. We all know the role of cell death. Cell death is very important. If cell dies, nothing happens. Everything is over. Bacteria knows, and it uses the PPP proteins to decide when to commit suicide. And there are 4 important pathways to this pathway: autophagy, apoptosis, pyroptosis, and necrosis. Both have different signatures, both have different roles, and both have different. And we and others have shown identified large number of PEPP genes which either block autophagy, or which block apoptosis, or some which increase necrosis. Why increase necrosis? To promote tissue inflammation. So the same bacteria uses one class of protein, members of one class, to access one arm of the cell death pathway. Why? Because it needs a cell to stay intact and does not want the death to proceed. Other class says now you can proceed for death. This is why we call it as a functional opportunism. Very functionally opportunistic. This paper is going to come up very soon. Now moving on to the next part of my talk, the second part, the drug discovery. We have been looking at drug discoveries, and this is the latest paper that we have published about 2 months ago. It came in the journal iScience. And here we have shown that we have carried out preclinical development of Mycobacterium tuberculosis as an anti-drug TB drug. This work has moved very far now. We are hoping to get big time funding from a drug company based out of UK, and we do hope that this will be so far we have no evidence to believe that this may not work, but then you don't know. Drugs, you are lucky if your drug that you make, but so far luck seems to be favoring us. So this is an enzyme this is a drug. We call it a new molecule, and this is a corimate mutase inhibitor. This work was done when I was at the interface of NII and CDFD Hyderabad. This work was done by one of my students at that time. Now we know that this protein is very important. Shekhar Mande worked with us on this protein, and of course we crystallized this protein and others. So this paper has now opened up a lot of interest in tuberculosis. We have now moved to AI and ML and integrating AI and ML to look at high throughput sequencing of clinical isolates. This is a project that we received from the Northeast. I am very fortunate that I continue to get large number of projects, including from Government of India and also overseas, that allows us to do things to indulge in our science. And we have done prioritize SNP. We found where all the mutations are. These mutations are genes which are already linked to drug resistance. There are also mutations which are not linked to drug resistance, and that is what made us interesting. What is this new protein? And to a surprise, we find a very large number of variations in the PEPP family of genes, and that is what we are trying to find out. How is this with PEPP? Is it purely coincidental, or is it having a function also? 2 minutes? Okay. So this is much of it is published already. Moving to the third part of my talk, last part, interventions. We have talked about basic science. We have talked about now about new method to detect diagnosis. We all know how TB is diagnosed, but can India and economically weaker countries afford supply of free cartridges? Today, you have in all the primary healthcare centers, you will find heavy machines loaded free by the cartridge, and that's where the problem is. We cannot do diagnosis, and one undiagnosed person can infect 20 fresh, healthy individuals. So it's very important, and we developed at the IIT Delhi a device which we called it as CTB, Chai Drishti. This is the Hindi name we have given. We thought Chai Drishti will be a better name, but Chai Drishti I can't write here. So CTB is what we are using. And this device, for development of this device, the person who did this in our group received the Gaiety Award. And today we have carried out CTB clearing. CLR is another device, another solution that we created, clearing reagent. It allows the sputum to become absolutely clear like distilled water in a matter of minutes. So together with this, we are going to be publishing very soon field trials of 3,000 plus samples, where we find that it can be commercialized in place of fluorescent microscope, and the results are very, very interesting. So what about countries now, towards the end of my last 2 or 3 slides? What about countries with pandemics and endemics? You know, we need to look at the programmized versus personalized approaches to manage. This paper that we published together with others has perhaps been cited 400 times already. And 2 parallel pandemics, it was actually an invitation, the challenge faced by countries with COVID-19 and TB. And in this paper particularly, learning from COVID-19 to tackle TB pandemic from despair to hope. What we have done, we have tried to present a picture that in COVID, we learned a lot. How to treat a disease of the stature of COVID, quote unquote stature of COVID, to be taken care of in a matter of weeks and if not months, if not weeks, because everything was why do we have to, for a drug, let's say for TB, why do we have to wait for 10 years or 12 years or 15 years? And I hope we are able to convince the powers that be in India that we need to do this, learn from COVID to do things fast. While during that time, we looked at, because the labs are closed, so we worked on a SARS variant. The paper we published in Nature Medicine. This paper talks about something which was actually originally submitted to New England Journal. New England Journal said, yes, we'll publish it, but show it to your healthcare authorities. If they say yes, we will get this approval, and also allow us to share your data before we publish in New England Journal with the WHO. And we thought about it, हमने क्या लिखा इस पेपर में जो कि New England Journal कहता है कि आप discuss करो. Anyway, after we submitted, it was rejected within 24 hours. And we know why it was rejected, because we had typically said how this virus could have arisen. Part of the work was published in that paper. So this is, can we still hope to eliminate TB? These are all editorials in Lancet where we have been able to show something. And this is my group when Tom Blundell was visiting us in Jamia Hamdard. And this is the TB group that we have right now at Sharda Hospital. The person who does all the work, leads all the work is she. And this person who actually picks up the stories and makes the presentations to all of you is me. And we have been fortunate to get a lot of funding from different places, National Science Share, the ICMR, DHR, and now we are hoping to get others support also. Thank you so much. And if you are keen to know what we try to communicate to New England Journal that was not allowed to be communicated, read this book, which we authored during COVID time, Decoding the Pandemic.
Thank you, Syed. Thank you very much. I think wonderfully you summed up everything, and maybe the time is a really short one, but your journey of these 3 decades in TB, you summed up the glimpses. But one more character, we must you remember when you talked about Talwar, that is your fiercely independent nature. Actually, I've seen you as a CFD director, as a vice chancellor of the Hyderabad.
Tribute to Talwar.
Yes, but anyway, you remain fiercely independent, and wish you good luck for that. Any one single comment, actually, anyone?
Can I add here? We are already running very late.
Yes.
I promise anybody who wants to know questions, pose questions to me, write to me on my email ID, sehiitd@gmail.com. And I promise you, I'll return the reply. I'll reply immediately.
Thank you.
sehiitd@gmail.com. Let's move on to the next.
Thank you, Syed. Thank you very much. Thank you.
So good to be here with you. So good to be here. We have very fond memories when he was in military. Thank you so much.
So our next speaker is Dr. Kiran Katoj, and she is from Central Jalma Institute for Leprosy and other infectious diseases. Dr. Katoj, she did her MD from SMS Medical College, Gwalior, and in Jaipur, and then she was selected as a ICMR talent search student in 1975, and it's a very prestigious honor for the students who are selected as a talent search and the talent search scheme. And in 1979, she joined Jalma, and she was over there till 2013. And she also served as a director of Jalma Institute from 2009 to 13. And she has done enormous work on leprosy. and she's going to present about that. So welcome, Dr. Kiran Katoshi.
Thank you, Dr. Javed. It is a privilege to be here at NII after so many years. And when I first visited NII, it was long time back. It was in, I think, 1982 that I visited NII, and I came to know about this institute because I was too occupied with my own work that I could do this. Anyway, I thank everybody who have invited me, and especially Professor Talwar, under whose guidance I could achieve some of the things which I had planned. Thank you. So this MIP, what it is, all you have already heard about it, and there was nobody better person than Dr. Talwar as well as Dr. Hasnain explain to you about it. But this is just a brief snapshot of what are its various properties, and there are many more about it. But yes, what being a clinician I would be emphasizing on, it is available today in the market. Dr. Khamar is here. They are the people who took it forward, brought it to the market. It is available in the market, and it is used not only for leprosy and TB, for many other diseases. So I'll start off with leprosy. Being a clinician, it was something which came as both as a challenge and as an opportunity. In this, the main thing that came in leprosy was that was the time when MDT had come in, and it was taking care of most of the problems of leprosy, but not all of them. People were persisting with it. The availability of MDT was also not there. But in wherever it was available, when sitting at a tertiary care center at Jalma, we could see that there was something still remaining. Everything was not there. So what we actually expect from a treatment is that we should cure the disease. All its complications should be cured. The disease, no landmarks of the disease should be there. And also it should take care of the transmission. Somehow MDT there is no doubt about it, as a global problem it has come down, as MDT has communicated a lot, but the challenges still remain. Even in patients who have been treated with MDT, they still continue with reactions. They still continue to have so many other complications, including disabilities and anesthesia on their body, which leads to ulcers and a long time stigmatization, disabilities about it. So that has not been done, and somehow MDT, it has brought down the load as well as the stigma, but all the things have not been answered. So this was one thing which was always troubling us, and in the meantime I got an opportunity that we were asked to do a survey, an independent survey in the vaccine area, where Sir had administered this vaccine to healthy household contacts of leprosy patients. I'll come to it as we go on. But this was the background. Then when we were reading, always we were asked that because MDT was prescribed and also said that yes, this is the thing which is taking care of it. We were seeing that there was a fall in, you can see the prevalence and the new case incidence rate. It has come down, there is no doubt about it. The number of patients suffering from leprosy has come down, but then it has stagnated off. Neither the PR is following, neither the new annual case is following, and also, unfortunately, neither the disabilities are falling. They have come to a level that, okay, 4% disabilities because of leprosy or whatever, whether it is because of neurology, it is definitely because of neurology department and neurological involvement, but it could maybe because of a big nerve, small nerves, sensation loss, trophic ulcers, or so many other things. So that is not coming down. So this is why is it still there. So these were some of the challenges which prompted us that we should have something that not in the therapy itself. I'll come to immunoprophylaxis later, but in the therapy itself, something should be added. We already have 3 drugs. And now even WHO says that even for possibacillary you should go on with 3 drugs. When initially we came up that 2 drugs are not sufficient and 6 months is not sufficient, they did not agree to it. But now everybody has started seeing it, especially because of plateauing off of the number of new incident cases. So now what we really required was that we should have something which is above this MDT, which will help the organism, which will to kill the organism in the host. See, both leprosy and M. leprae and MTB, they have survived throughout mankind. They were there even in the biblical times, even in our pre-Vedic times, they have been described, and they are there even today. So what is there in this bacteria? It survives in this environment, and it still troubles us, although it is for a small proportion of the population. So when this initial thing came up, it was more for immunoprophylaxis than for immunotherapy. But we wanted to try it because in a tertiary care center, when you see so many disabled persons, you want to do something for them. So that is how we came up with this, and this was the initial dose that we had taken up, which was given us because it is a killed vaccine. We had to repeat it after 6 months. And the other thing that we took up was distilled water as a placebo. We also took up BCG because at that time there was hardly any funding where BCG was not involved. And for that, again, it was that BCG had shown its effect against tuberculosis. There were worldwide studies. Yes, it was different in different areas of the geographical regions of the world because of probably of the environmental bacteria or whatever of the host immunity or whatever you may call it. But this was how the initial study was started, in which MIP was one of the arms of this 3-arm study. And this was what we wanted to see, whether it was acceptable, because it was causing an ulcer which was healing in another 4 to 6 weeks. So it was causing some disruption, and also whether there was a clinical improvement. We started with a highly bacillated because it was these people who were suffering the most, whether it was disabilities, whether it was stigma, or whether it was anything. So it was always this, and that is why this was started off. And the ethics also was not giving us any permission that unless you show it to us in black and white, we will not give you any permission to start in all types of leprosy patients. So this is what it came up. So what was this thing that it was, I have already said it is every 6 months. It was found it was safe and well accepted. We followed these patients with treatment and after completion of treatment up to 12 years after release from treatment. And what we really did, which I wanted to say, because why it got accepted was, we did a detailed clinical examination. It was started, it was there, it was not digitalized at that time, but it was there in the files. Then the skin smear microscopy, which was done so often that time, and we did biopsy. It was not only from the local site, it was also from a distal site, from a distant part of the body, where we could see whether the bacilli was still lodged there, whether it was making the granuloma, whether it was leading to reactions, what was actually happening. So this was the style of our study. And we also did the viability assessment by the ATP estimation of those bacilli which we found in those biopsies. This definitely, the microbiology department helped us a lot, and this is what we did. So what we did in these highly bacillated, this is just a view in which you can see here I have not put BCG at all. It is just distilled water as a placebo and MW as I'm sorry, it is still MW, of course it is MIP, and you can see the fall in BI is much faster in the, it is going ahead. I'm really sorry for that. So if you see that initially this was the percent of patient who developed reactions. The earlier one was, this is the fall in BI of these patients, the bacteriological index in these patients. You see it is much faster than only MDT group. This and the next slide will show you the incidence of reactions. Immediately after giving MIP, it was found that there was some rise in reaction because it was actually challenging the host immunity. So there were probably a small incidence of reactions in the early time, but which fell off and much faster than with the MDT group. And there were no reactions after 30 months in the MIP group, whereas in these it was still present even after 36 months. And at that time the treatment was for about 24 months and could be extended for another 6 months if the patient was still positive. So this was what we saw, and this was the histopathology. If you see here, it was increased lymphocytic infiltration at the local site where the MIP was given, and also at the distal site. There were presence of epithelioid cells which were not heard of in multibacillary and highly bacillated leprosy. It was and there you could see that epithelioid cells were coming in. They were scavenging some of these cells and they were forming there. And there was rapid earlier reduction in the granuloma fraction, which was both at the local site and at the distant site. This granuloma fraction was important to actually demonstrate and also to record, because it was always believed that after MDT, when reactions occurs, it is because of dead bacilli, it is not because of life, because MDT has taken care of it. That was the common belief about it. But we knew that there were some dead bacilli could also evoke immunological phenomena and lead to nerve damage, and that is why we tried to show it both at the local as well as at the distal site. This was on the mouse foot pad on the viability of the bacteria. This was again both at the local, the biopsy from both the local and the distal site, and here you see the viability with MDT plus MIP. There was no viability, at least in the mouse foot pad, after 6 months, whereas in those patients who got only MDT, viable bacilli could be detected. That means there was growth in the mouse foot pad even after 18 months. This was the ATP estimation, that is when these bacilli multiply, they take up the ATP for themselves to multiply and destroy. So this was by the ATP estimation, which is detected by a bioluminescent phenomena, and there we could see that at the local site they had disappeared completely, but at a distant site this ATP estimation was showing that I'm sorry, okay, then I'll just rush through. This ATP was just showing that yes, there were some bacilli still present, active bacilli multiplying at a distal site, but they had totally vanished off at the local site, and this also with the time they were all getting manifested. Now this was the summary results. I'll rush through it, that there was there was decreased lymphocytic involvement even later on, rapid granuloma clearance, and none of the patients developed disability. But this was true for both the groups, those who were on treatment with only drugs and also with those who were not on treatment, who were on treatment with MIP. This is how some of these patients, all the nodules, everything responded. This is only after 6 months. After complete treatment, you could not recognize that this patient was ever a patient of multibacillary leprosy. Then this gave us the opportunity, and the ethics also come that yes, you can do it in a borderline patient. This just shows that we took all type of borderline patients in both the groups, BT, BB, BL, LL. Some of them were positive, some of them were ALB negative. Here this graph just shows that how many of them actually responded to this. There was actually, their lesions also fell, there were lesser lesions, the erythema in the lesion decreased, and what was the most important was that there was return of sensation. In some of these BT patients, they said yes, we could not feel it earlier, but now we can feel it after getting both MIP and MDT. And this was the histological improvement. As you see that the histological improvement was much more faster with the granuloma clearance in these BT patients. This is the histological picture showing that the granuloma also had gone off and there was infiltration. This was in a BT patient who was supposed to have many giant cells and some of the bacilli could be stained. I'm sorry, I cannot show you the bacilli here, but this was that in a BB patient. And even in reactions, just 5 more minutes, sir. Even in reactions, you could see that there was a decrease in the incidence of reactions, both, and this was both of RR type, that is type 1 reaction, as well as type 2 reaction. So there were no disabilities. So immunotherapy was helping in faster smear negativity, killing of viable bacilli as I have shown, clearing of dead bacilli and the products seen histologically, reducing the incidence and severity of reactions as seen by follow-up of these patients, and preventing disabilities and nerve damage, therefore reducing stigma. Same thing was seen in paucibacillary with a return of sensation in a sizable number of population. So this was the advantages, I have already said that. Now when we are using it as immunoprophylaxis, this is what when we came into direct contact with Professor Talwar as well as with DBT, and this is where we started that they had already vaccinated the people in this Ghatampur region of Kanpur, where the multibacillary cases as well as their contacts of all leprosy patients were vaccinated with MIP vaccine, again 2 doses, one at the start and one 6 months later, and these patients, and they had done the first and the second follow-up, and we were asked to do an independent survey with the third follow-up, which was about 8 to 9 years after the initial vaccination. Kanpur was chosen because it was highly endemic in leprosy and it was something which was approachable from Delhi, and so it was taken up. So these were the 4 codes that were given, where this was given both. In group 1 only contacts received the MIP, contacts of all leprosy patients received MIP. In group 2 patients it was only given to the patient, and that too only MB patient. In group 3 neither the contacts nor the patients got it, and group 4 both the contacts and the MB patients got the MIP vaccine. I'll come to it when it comes to the results. So this was what in nutshell the results of the 3 surveys. I've put forward the second survey and the third survey, the results, the number of patients and which was there. If you see that, you will say that the cumulative thing, yes, it is much lesser than that, but yes, they were occurring. This was in a population of about 22 to 23,000, and this was what we had covered and the total contacts that were given this. When only, just to summarize things, when only contacts received the vaccine, it was seen that the PI was 68% at the end of the first survey, that is 3 years post-vaccination, and by the end of the second survey, it was about 60%, and this decreased to about 33% when we did the survey, that is after 9 years. Probably being a killed vaccine, this vaccination was rendered, and also there were more new cases occurring, and they were actually transmitting the disease more rapidly. In the group in which contacts did not receive, we found it was not so this thing, and this is published
Please wind up.
in this journal.
Please wind up.
I'll just finish it. Then these results indicate that MIP being a killed vaccine, it requires a repeat vaccination. It is not a live vaccine as a BCG, and it has both its advantages and disadvantages. But this definitely shows that this takes care of all types of patients. It takes care of both reaction and relapses, and also it controls the transmission that it can be used. Now, this was, I was just trying to compare it. I'll just rush through this. In this study, they had reported, they had taken up 4, this was the South Indian study, undertaken by it was given to a general population. It was not only to contacts, it was for the general population, and the efficacy that they had reported, which is also published, they reported a very lower protection. So we compared it, and especially in ICMR this was discussed a lot, and then it was said that okay, what we had reported in Kanpur study when we had done the third evaluation, let us see if we separate the contacts and see how much was the protection of MIP in the South Indian study. So this is what they came up, and there we see that the protection was much more in the household contacts. With this, we were also by the by NTAGI, we were said, okay, now you have shown all this, now let us see what is the cost effectiveness. Supposing you put it in the program, how it will help. So we administered, this was the proposal in which we have calculated the cost effectiveness, in which MIP is given to all newly detected cases which are detected today, and all contacts are examined as per NLEP guidelines, and about 6 to 20 contacts of each newly detected cases is given MIP. And again it is given in 2 doses, 1 dose at the start and 1 6 months later. So the cost effectiveness of this has been calculated, and this is what we were the input parameters that we took. We will just gained it by the QALYs gained and by the ICER gained. This graph just shows you that this is much less than 1 times the GDP at that time. That was 2016 GDP, which it was compared with, and it is very, very cost effective. This graph shows this. So MIP was saying what advantages it provides for prophylaxis, as well as it also provides for immunotherapy, so that the treatment is better, and it has a potential of achieving a much lower ANCDR, prevent nerve damage, decrease the severity and extent of reactions. If added as an adjunct, it can be taken up. It is very, very cost effective, and it also prevents quiet nerve paralysis, which is now being reported. Patients are not coming now with disabilities with pain. They come with quite suddenly they have a loss of sensation and they develop a claw hand or a foot drop, and they come ki nahi hame to koi dard nahi hua. So that is what they say, it is a quiet nerve paralysis, and all this reduction leads to reduction in stigma. And then I'll just sum up for tuberculosis. While we were doing this study, we found that in those contacts in which leprosy we detected, we found the TB rate was very low in those patients who had received the vaccine, because we could see the scar which was present there. So we said we gave this to the DBT, and the DBT funded this study in which we...
Can you please finish up?
Yes, sir. So in this study, sir, what we saw that there was a lesser, that we could show, this is again is published in Vaccine in 2006, and where we showed that MIP was also protecting against TB in these contacts, in only those household contacts in which who were actually vaccinated for leprosy. So then we started seeing whether it was acting as an immunotherapy. We took up category 2 patients to first, because they were more difficult to treat, and especially in that Kanpur region there were many people who had defaulted and had become from category 1 had returned to category 2. This is a multicentric study. Rajni will also be talking about this. I'll just sum it up saying that this was in this the cure rate was higher in the MIP group, as well as what we could see in these very extensive TB patients, that in the MIP group especially it was more better that they became smear negative faster, and also culture negative. Not only smear negative, culture negative also faster, and this was statistically significant. And many of these patients who had multiple cavities, which was a part of an RNTCP criteria of diagnosing this disease, this was acting better as compared to them, and also in those who were more smear positive. So this, I think Rajni will be talking. The relapse part of this, yes, it was not statistically significant among in this multicentric study.
Ma'am, you are also consuming Rajni's time.
No, I'm sorry. So I will just say that besides its action as an immunomodulator, it is also acted that many people will be talking about it. And I'll end my lecture with acknowledging all my patients who were there, who were there both at the tertiary care and as well at the Ghatampur Center, all my staff members, my SAC members, my RAC members, and of course my teachers who always guided me through this. And thank you all for your patient hearing in spite of my overdoing.
So ma'am forgot to acknowledge Dr. V.M. Katosh as well, because all microbiology was done by Dr. V.M.
Yes,
Katosh.
definitely, sir.
So always, you know, wives, they fail to acknowledge their husbands. So same was the case over here as well. So you can see very well, you know, how beautiful, it's excellent data shown that it prevent deformity, it can prevent sensation loss, transmission, as well as, you know, control of leprosy. So we'll take quick one question. Okay, so if there aren't any questions, thank you very much, ma'am, for a wonderful talk.
Thank you very much.
And,
Thanks.
I could very well see that while ma'am was presenting excellent data, Professor Talwar face was glowing, it was shining. He was so excited to see his data. Okay, so our next.
Thank you, Javed. Well, next, my pleasure to invite Dr. Rajni Rani.
Thank you.
Well, it's a pleasure to invite Dr. Rajni Rani to deliver the next talk.
Sorry, I couldn't hear.
Dr. Rajni Rani, as I said in my initial remarks, she has been an old colleague of Professor Talwar, Professor Ramam Mukherjee, then Professor Talwar, who has been publishing on leprosy since 80s actually. And I think even the paper on the W also came in 1992. I remember my old stories, but I think. And she has been consistently working on that.
Okay,
after, even she went to ICMR after a superannuation from here. Again, her work, she has continued with her association with the TB work. And I think she represents, I think, really a long 45 years of association with both TB and leprosy. The work, but of course, her span of work goes far beyond vitiligo, so many other things which she had done over the period of time. Dr. Rajni Rani, please.
Thank you. Can you hear me? Am I audible? Yeah. Okay. Good afternoon, everyone. I'd first like to thank the organizers for the invitation on this auspicious day of Dr. Talwar's birthday, and I'm really very happy to be here. And what I'm going to do in the next 15 to 20 minutes is take you over 4 different trials that with me. 2 for leprosy, therapeutic and prophylactic, and then 2 for TB, Cat2 TB, therapeutic and prophylactic trials which have just concluded. So when I joined NII in 1987, we used to have Monday and Friday meetings, and then we used to present our data. And when I joined, I used to hear LL, BL, BT, BB, and I had no idea what it was because I was an immunogeneticist. So what I thought was, in this audience, there may be some people who do not know what leprosy exactly is. So leprosy, or Hansen's disease, is caused by M. leprae. Incubation period may range from 2 to 25 years. Not all people who are infected with M. leprae will develop the disease. There are several factors that compromise the immune response, like malnutrition, other illnesses, or genetic mutations that increase the risk of developing leprosy when infected. It manifests in the form of a spectrum. So this is the spectrum that I was talking. On one end of the spectrum, there are tuberculoid leprosy patients, and on the other end are the lepromatous leprosy patients. The tuberculoid leprosy patients have good cell-mediated immunity. They are paucibacillary because of their cell-mediated immunity. The disease is self-resolving, and these are lepromin positive. So if you do a skin test, you will get a positive result. However, in the lepromatous, the CMI is lacking, and they are multibacillary, there are many bacilli, and they are lepromin negative because they don't have a CMI. Humoral immune response is present throughout the spectrum, but this is not protective at all. So the problem is with the LL, the lepromatous leprosy. In between the two poles are borderline tuberculoid, borderline, and borderline lepromatous leprosy patients. So I thought for the others who don't understand this, this may be easy. I'll give you a bit of historical perspective. I've taken this from Dr. Talwar's paper. In 1970s, Professor Talwar and his group realized that in lepromatous leprosy patients there is no immune response. So something is required which would generate immune response in them. So they collected 16 cultivable atypical mycobacteria from various sources, and then they shortlisted 5 of them. And Mycobacterium W was one of them. They coded these, so W was the code. They coded these and evaluated their ability to induce blast formation of T cells from both tuberculoid as well as lepromatous patients. To confirm their closeness to M. lepri, they also made lepromin-like preparations and injected them along with lepromin in TT and LL patients to evoke M. lepri lepromin-like responses in leprosy patients. And of all these, MW appeared to be the closest, and it also induced immune response in LL patients. So the question was whether MW could convert lepromin-negative LL patients to lepromin-positive status, and yes, indeed it could. So what is MIP? So Dr. Hasnan has already explained you Mycobacterium indicus prenai, so I won't go into that. MIP is an atypical cultivable non-pathogenic mycobacteria which has been shown to have both immunoprophylactic cum immunotherapeutic effects against leprosy. Besides leprosy, MIP also has remarkable capabilities to treat tuberculosis, including CAT2, difficult to treat tuberculosis. Dr. Katoch just mentioned some of it just before me. MIP is an immunomodulator that activated T cells and cytokines, particularly interferon gamma, which played a major role in its action against cancer cells. Combination of MIP with cyclophosphamide improves anti-tumor activity. MIP is approved by the Drug Controller General of India and US FDA, and it is licensed to Kerala for availability to public in India. Now, I will talk about when I joined in 1987, 2 hospital-based trials were going on, one in Ramanoa Loya Hospital and the other in Sabdayan Hospital. So there were 156 lepromin-negative multibacillary leprosy patients and 145 matched control who were given MDT plus placebo. MDT was given for a minimum period of 2 years, and MIP was given at 3 monthly intervals up to a maximum of 8 doses. Fall in clinical and bacteriological score was significantly more rapid in MIP cases from 6 months onwards. MIP induced lepromin conversion from negative to positive, indicating that CMI has been generated in the lepromatous cases. Histopathological upgradation was seen from LL to BL, BB, BT. Many patients showed complete disappearance of dermal granulomas. In some non-responders to MDT who have taken MDT for 2 years, giving MIP just in couple of doses, they became lepromin positive. The vaccine did not precipitate neuritis. However, higher incidence of type 1 reactions were observed in LL patients due to upgradation in CMI. So several papers, more than 20 papers I think have been published, and I'm just showing a few of these. This is a favorite slide of Professor Talwar, and I have taken this from his paper. And Dr. Zahir, who was doing this trial, used to show these often. See, this is one patient. Initially, he had this type of granuloma, and after 5 doses cleared. This is initial, and this is after 2 doses. And this is again another case where you have initially hyperpigmented areas which have disappeared after 4 doses, and this person initially has this. So, and this is the most favorite slide of Dr. Talwar. He would show that initially she had this, and after 4 doses this. I think she's from Ramanoa Loya Hospital. And now 1 year after, everything is clean, and you can't even recognize that it is the same patient. So after this, immunotherapeutic trials, Dr. Talwar thought whether we could go for immunoprophylaxis. And for that, a large-scale trial was conducted in Kanpur Dehat that Dr. Kiran Katoch mentioned. So it was a population of 420,823 spread over 272 villages of Ghatampur Dehat of Kanpur. 1126 multibacillary and 3757 paucibacillary cases of leprosy were detected. 24,060 household leprosy patients were enrolled and vaccinated with either MW or placebo. Vaccine consisted of 1 × 10⁹ heat-killed MW in normal saline for the first dose, followed by 5 × 10⁸ bacilli 6 months later, and placebo consisted of 1/8 dose of tetanus toxoid. So these codes Dr. Kiran Katoch just showed you. So the leprosy patients got in P1C1 MDT plus placebo and the contacts got vaccine. P2C2 MDT plus vaccine was given to patients, placebo to the contacts. P3C3 was MDT plus placebo, here only placebo to contacts. P4C4 MDT plus vaccine and vaccine here. Villages were stratified and cluster randomized on the prevalence of leprosy and population size. Household contacts are followed up for first follow-up at 3 to 4 years, second follow-up 6 to 8 years, and the third follow-up, which was done by JALMA, 9 to 10 years. Enrollment, first and second follow-up were done by the field unit of NII, and the third follow-up was done by ICMS JALMA. So, if you see this slide, this shows the first survey. P4C4 is absolute vaccination case where both the patients and household contacts have got the vaccine. And P3C3 is absolute placebo group where both patients and the contacts have got the placebo. The protective efficacy of 68% was seen in 3 to 4 years, which reduced to 60% in 6 to 8 years and 28% in 9 to 10 years in the third survey. When vaccine was given only to the household contacts plus placebo to both, the vaccine efficacy was 68.6, which is similar almost. In 3 to 4 years, came down to 59, again similar, but it reduced to 39% in the third follow-up. When vaccine for patients only, although the difference is significant, the vaccine efficacy was quite low, 42.9 for the first follow-up, 31% for the second follow-up, and 3% for the third follow-up. After that, since Dr. Kiran Katoch had shown that when they followed up the patients in the third follow-up, they saw that the number of TB cases had reduced. So it was thought that can we try this for Cat2 TB. Now, we know that there is a good ATT and there's in primary TB which takes care, but there are defaulters, non-compliance is there, relapses are rampant, converting these patients into category 2 patients. So Cat2 includes those patients who have failed previous treatment, relapsed after treatment, or have defaulted during previous treatment. A significant number of Cat2 TB patients have been reported to develop MDR TB. Immunotherapy as an adjunct to standard ATT could be an important strategy to treat this category of patients, to prevent Cat2 patients converting into MDR. MIP shares antigens with both MLEPT and MTB and has shown to have significant protection against TB in both BCG responder as well as BCG non-responder strains of mice. So immunotherapeutic potential of MIP was evaluated in Cat2 pulmonary TB patients. The study design was a prospective randomized double-blind placebo-controlled trial. 18 to 60 years was the age of the patients. Cat2 patients included those with treatment after default, treatment failure, and treatment relapse. Vaccine schedule was patients were randomized and 5 × 10⁸ heat-killed bacilli were given in normal saline, or placebo was normal saline without bacilli, once in 2 weeks over 2 months. So there were 6 doses. First dose was two on both the arms, and then after every 15 days for 2 months they were given the rest of the 4 doses. The primary outcome measure as defined in the registered trial was the time of sputum conversion as well as the early sputum conversion in the 2 groups. The cure rate to be evaluated as primary parameter of efficacy. The relapse in patients of category 2 tubercular tuberculosis to be compared in both the groups at an interval of 6, 12, 18 and 24 months after the completion of the therapy. A total of 1553 CAT2 patients were screened, 531 screen failure and 1022 were randomized into 1:1 ratio. However, after exclusion of MDRs, only 890 patients were eligible. MIP was given to 449 and placebo was given to 441. 642 cases in MIP arm, MIP arm 32 and placebo arm 314 patients completed the full course of therapy. Now here I am showing the smear conversion and the culture conversion. If you see the yellow part, this is weeks. At 4 weeks, there was significantly higher number of people in the MIP arm who converted to whose culture negative compared to placebo arm, and this was very significant with an odds ratio of 1.86. So culture, and if you look at the smear, there is no significant difference. So in smear, we may miss some bacilli which will actually multiply in culture. So culture is more sensitive, and we see that in culture we see a very significant number of people in MIP who showed culture conversion. That means they became culture negative. If we look at the cure rate, there was no significant difference because the cure rate was seen at the end of the trial, end of the time. So naturally everybody would actually get cured. But 68.8% of all patients who were initially inducted for the treatment were cured in MIP group as compared to 64% in the
Dr. Rajni, 2 minutes.
Okay. The frequency of patients cured amongst those who completed the full course of treatment was 94.2 compared to 90.4. So there was no significant difference. However, people who were 2 to 3 drug resistant were significantly more cured in the MIP. All 16 were cured compared to 76% in the placebo arm. People who had 3 plus BI also were significantly more number were cured, and people with bilateral cavitation also more numbers were cured in MIP as compared to the placebo. So I am not going to summarize this as Dr. Katosh has already summarized, and this was published in scientific reports. And immunoprophylactic effect of MIP has been tested in tuberculosis, and I won't go into the details of this. Rationale also, I think we all know that it protects BCG resistant, and then I would like to say in Dr. Sangeeta Bhaskar's group, they have shown MIP as an immunomodulated vaccine that reduced bacterial load, improved pathology and organized granulomatous response post-infection in the MIP immunized guinea pigs. And MIP induced an early increase in protective Th1 immune response followed by decrease in the inflammatory response and increase in the immunosuppressive response. So the protocol for this has already been published, and I cannot tell you the details of this right now, the data right now, but all I can tell you is that 1,217 people, 4239 in one, 4239 in the second, and the 4239 in third group, which is MIP, Immuvac, MIP, VPM 1002 and placebo have been enrolled, and of these 252 people have developed microbiologically confirmed TB, and the details of the results will be presented sometimes later. I cannot present right now. Thank you. So I'd like to acknowledge Professor Talwar, Dr. Rama Mukherjee and Dr. Zahid were doing the trial at the time when I joined. Unfortunately, none of them are here now. Dr. Walia, Dr. S.K. Parida and Dr. Pankaj Sharma, these were the clinicians involved. Dr. Harvinder Kaur was a social worker. Dr. Indra Muleja did the mice work, and Dr. Sangeeta Bhaskar did the work on guinea pigs. And these were the collaborators from RML, Safdarjung Hospital, Institute of Pathology, Jalma, names. And then this is for the CAT2 study. I couldn't write all the names, so I've written this. Dr. Bindude was involved in, she was a catalyst actually for these trials. And Catalyst Clinical Services was the CRO, and Catalyst Pharmaceuticals provided the vaccines. Thank you.
Thank you, Dr. Rajandrani. for really summing up everything again, that's in a very focused way, and which reinforces, that it has worked in all the studies, whether done in Delhi, done in Ghatampur, done elsewhere, MW has played a part, very significant part, and I think that's beyond doubt actually. Thank you very much actually for the very nice presentation.
Thank you.
Next speaker is Professor S.A. Patil. So he's a very renowned personality, does not need any introduction. However, he has a long-standing working in the field of leprosy, tuberculosis for last 40 years. He started his career from Central German Institute for Leprosy in 1986, 85, and then continued working over there. Later on, he moved to NIMHAMS, and then again came back as a director of Central German Institute for Leprosy and other microbacterial diseases. And currently, he is working as a professor and dean of NIMHAMS Bangalore. So over to you, Professor Patil, and you have only 20 minutes.
Thank you so much, Dr. Javed. We are working in the same lab. So now this obviously tells what I am going to talk about. This is a tuberculosis, how the disease spreads, and this is a bacteria with the EM scanning. Initially, the mycobacteria was thought to be, it's a normal bacteria in the atmosphere, but some of them developed into their whatever complications of that, and they have become pathogenic, and MTB is one of them. And it is regarded as wayward son of a honorable parents, means they are non-infective parents, and this is one which has become infective. Now, tuberculosis complex, these are the organisms. Just to tell you a brief, because the government of India and the global, they are thinking to control this disease by 2030, then the disease is not in proper control. So we have to look back to our pages, what we have done, where we have to improve upon, or where we have gone wrong. So that's the reason I am going back to some of those slides. And these other organisms, Africanum, bovis, microti, these are keeping quiet all these days, but they are doing a lot of mischief over the period as a non-tuberculous mycobacteria, mimicking tuberculosis and then confusing the clinicians. The main thing of TB, that Mycobacterium tuberculosis, is its cell wall. This is the one which makes it, very robust and viable in the atmosphere for 2 to 3 months. And then with the wind and all that, it is being taken up, and then again the disease starts. Unlike the other gram-positive, gram-negative bacteria which die very, within couple of hours in the atmosphere. So this lipoarabinomenon, which is a structure of this, has a very important role even in diagnosis, and these lipids and galactans, these are the one which makes it survive for a longer period in the atmosphere and even in the body, and refractile to the drugs which we are giving. Now these risk factors are quite known to all of us, probably HIV infection, these diabetes and all that. And all that, what I would like to highlight is this nutrition. Earlier nutrition was given only 2% as a risk factor, but now this has been more than 25% as a risk factor. Professor Soumya has mentioned in her recent article that more the waste, there's a BMI, lesser the chances of succumbing to tuberculosis. So this is gaining importance, so I would like to highlight on that aspect. Is commonly transmitted by droplet nuclei which are aerosolized coughing, sneezing. The slide I showed, transmission from one to the other and number of infectious droplets, they tell about the catching of the infection, and the virulence of M. tuberculosis is the one which is. This is a picture where the global TB, India has 20% of tuberculosis, whereas Indian population, I would put it in another way, 1/5 of the global TB is there in India, whereas we have a population of 1/6. So we have higher density, we are getting bigger share of tuberculosis globally in India. The other countries are there which have a minimal this thing, and we are ahead of China. So it is very clear from here. This slide was made about 4-5 years back. That time the Chinese population was much higher than Indian population, so this is to be considered. This is a picture of India showing higher this thing, and this is the distribution over the different places. Now the global emergency, what I was talking, by 2030 we would like to contain this disease. It's still in a dilemma. Some of the hardcore workers on TB, they would say yes, it is possible, and rest of them they say it's little doubtful. Now what I would like to focus on, though I have worked on TB and leprosy, but I would like to talk on extrapulmonary tuberculosis. This is one of the neglected sort of thing among tuberculosis. Though this is about 20% of the cases of TB are extrapulmonary TB. This bacteria reaches the brain. We call it as TB meningitis, lymphadenitis, pleura, miliary tuberculosis, and cardiac tuberculosis, bones and joints, spine, and genitourinary tuberculosis. Me working in a institute of national importance on neurological disorders, I have worked extensively on these things. You can see how the bacteria looks on culture and staining, and there is a form of latent TB. You can see that the bacteria resides here, whereas in a fulminant cases, active TB, it grows like this. So these are more infective, and this is less infective. Other forms of TB are TB lymphadenitis. These are some of the rare pictures probably you may not come across. And this is a skeletal TB. You can see how the bones have been infected with this bacilli. And now genitourinary TB, intestinal TB, miliary TB, TB meningitis, the one which I would like to talk in detail, TB of the liver, and then TB peritonitis. Cutaneous TB gains more importance because of skin related activities have increased in the country, want to have a good skin and so on and so forth. TB pericarditis, how it just is a layman's picture where once you inhale the sneezing of another person who is having TB, that goes into the lungs, resides in the lungs, settles, then slowly gets into the circulatory system, breaks the blood-brain barrier, goes to the brain, and then it concentrates there, and then again distributes within the brain. And this disease is very pulmonary tuberculosis gives some time for you, because you know that by pulmonary tuberculosis nobody dies within a month or two or so on and so forth. Maybe you get time up to 1, 2, 3 years and so on. Whereas for TB meningitis, you get very little time. In this little time, initially you will feel just malaise, and then after some time you get a headache, which you will prolong it with your coffee and whatever the treatment, small tablets here and there. But then in the meantime it establishes itself, and then it goes very difficult even after diagnosing the treatment. So it's very heartening to share that about 50 to 60% of the patients we can't retrieve with this condition. To look into this, I have done these studies where you can see different class of antibodies present in that. G and M are the main classes of antibodies, and these are some of the other percentage which you have shown here. What I would like to highlight, though this study has been done long time back, I am coming back to that study for the reason that when a patient comes, you know, if you have a idea about the things happening in the hospital, or the clinician takes the sample, then he tells, okay, you come next week or something, next week he may not be able to come, or you know, he feels a little better and he may not, or for some reasons he doesn't come, and that link, whatever he has come, misses. So if we have a immediate test which is point of care test, okay, wait for half an hour and we do some of the tests, and then if it comes positive, then we can say that yes, the subsequent thing of giving treatment and follow up and all that, TB is a notifiable diseases, all his record will be taken and the medicine can go to his home. So this is what is needed at the moment, I feel. I have put this slide back, which was done about 7, 8, 10 years back, but then for the need of the, you know, time to contain the disease. Now, the different things have come, how to, you know, the treatment. First is the diagnosis, second is treatment, and third is the control of the disease. So now we have seen the, you know, the drugs. There are, you know, this study has we have done in collaboration with a small place called Jalgaon. From there we have done it. This spray drying technique which has been established. It can be done for the phages, it can be done for the TB drugs, it can be done for the vaccine or any peptide. If it is given, it is dried in a proper fashion and then it can be given as a pulmonary delivery, then you can have a better control of this. This not only controls the disease, but drug resistance should be reduced, and the second thing is in effective treatment, because it doesn't go through all the process of digestion and then all that, and then there may be a beginning of the resistance there. Now, drug resistance, to tell you something about, some of you may be microbiologists here, I can show you one slide. This was a picture about 25 years back where the bacteria was sensitive to most of the drugs. But this is a picture, a recent picture, 4 or 5 years back we are seeing this type of bacteria. They are resistant to all the drugs. What one has to do if some bacteria comes like this? I have seen myself, the community acquired young boy, 7, 8 years has come, and the isolate was resistant to all the things. Now we are the laboratory people, physician is standing in front of it, patient is there. What we have to do? So we have to really look into the alternative things. Otherwise, you know, there is no value for what all we have been doing over the years. Everybody knows that, you know, to develop a drug, it takes about 10 years and $10 million. And for bacteria to develop the resistance, it takes 6 months. All your efforts of, you know, whatever positive efforts, somewhere it may go negative, then it will not come to picture at all. So these are the things. This is a phage. Phage is a virus which eats the bacteria, and they have been found to be specific to the bacteria, whichever bacteria you want to choose and all that. In the system, they go like a missile. Wherever you go, hide and all that, or your email, wherever you put your laptop, it goes in your system. So these go wherever in your body the bacteria is sitting, it finds it out and eats, kills, and then the bacteria is killed. Then what subsequently happens to to this? I will not go much into this, but you can see that if we give the phage, the 100% survival is there. Even if we give the drug, is about 50%, 60% survival, and if we don't give anything, it's 100% death. So we have seen that these phages do produce some degree of antibodies, but they are not, you know, pathogenic. We have seen the histopathological examination in an experimental model is perfectly fine, and phages are viruses that eat bacteria. They can prove alternative as a drug therapy, keeping with the extensive drug resistance in hospital acquired or community acquired this thing. So clinical trials phases have been conducted before the administering. Now the main problem for us is the treatment for TB. TB goes for 2 to 3 years treatment. We have to be accurate in diagnosis. Otherwise, at the end of 3 years, you will see that you have gone, you know, instead of going to, you know, Jaipur, you have gone to Jodhpur, and then you have to come back, and then by the time patient may not be there. So while instituting the therapy, you can take some time but reassess the thing. So these are some of the things which we have to look into the alternative ways. I don't want to irritate with that figure to you, but now non-tuberculous mycobacteria. These are the bacteria which are, they mimic like TB, but they are not. Once the diagnosis of TB is a sputum examination, it comes positive, then you start the treatment. Whereas this non-tuberculous mycobacteria is not sensitive to the TB drugs. You give it for 2 years, 3 years, and the patient worsens.
You're left with 2 minutes.
Thanks. We have got a paper recently from China. They have said that a case of NTM did not respond to, not responding case of TB. Then I checked the details and then said, it's the wrong diagnosis which you have done, and you have put the patient for 3 years on the drug, and then you want to publish the study. I gave a very harsh comment to them. This is a misdiagnosis, you are incompetent, and the patient should sue you for your, and on top of that you are saying that this is, sort of publication. So these are non-tuberculous mycobacteria which look like that. We have done these studies, non-tuberculous mycobacteria sort of studies which were sputum positive, but they were not TB, in about 13, 14 district states we have done this study. And this is a simple study. Within a week you can distinguish. You can start the ATT treatment, but within a week you can, check it and correct it, give what is needed, or continue with that. I will not go much into this. A tubercular meningitis. This is nothing but TB meningitis. This gives very little time for you to, treat the case. You can understand what is happening to the brain. Initially there is nothing, you are, just getting few headache and all that, and then you start getting seizures, and then it becomes difficult. These are the pictures of the brain which are totally. We take cerebrospinal fluid by lumbar puncture and then look into that. These are the bacteria which you can see in that. Culture is very difficult in this because it takes 2 months to grow it, and just if you can give 2 minutes.
Time is over.
Rapid growth is available, but then it still takes about a month or 2. But before that we have to do something to treat the cases. Manto test is still a valid test because it tells whether the person is having TB or not, because the person immediately doesn't go to a specialist where he can say that you have TB meningitis. He may say something, you have just casual headache and all that, unless he comes to a specialized person, a neurologist or so, and then the treatment would start. Otherwise it goes very difficult. These are some of the things which you have looked into, and we have to re-look into. Cutaneous tuberculosis is another thing which is coming up. So I have, specifically avoided the pulmonary TB because I knew that my colleagues would present on that. So this is mainly the extrapulmonary tuberculosis. You can see some of the pictures. These are skin manifestations, and then pathology which tells you about those things, and the treatment. This is the last slide I would like to say. We wanted to characterize these proteins and express into the plants, and then we thought we can give something like treatment to the animals or humans or something like in the long run. We express these proteins 8kd16 and ESAT85 and all that, but the project could not go ahead for various reasons. Funding reasons is the main thing. This is the one we can look into, that spray drying technique which is available, and most of you who are working on tuberculosis can look into that, and this would be very beneficial to avoid the drug resistance and applying the drug at the point of infection. So this will definitely reduce the resistance, because the tablet which you take from here, it goes to your this thing absorbed, and all that, and then circulated, it may or may not, and so on and so forth happening. But point of care, if we give directly, it would reach at the point of the infection. So This is the PhD study of one of my students who has done on POJ, and the drawing study is done in collaboration with Jalgaon University. Thank you so much.
Thank you. So very nice study. You know, all diseases are bad diseases, but these leprosy and tuberculosis, they are horrible diseases. Whereas if person gets leprosy, it infects the body of a patient and mind of public. And that was the reason that Professor Talwar got intrigued about leprosy. So that, and now tuberculosis, of course, is that induces high rate of mortality. So we can take, few quick questions. Yes, please. You can introduce yourself.
Sir.
Its diagnosis and management. Diagnosis, transmission, and management.
Yeah, in zoonotic tuberculosis, there are certain, veterinary centers which have been working on that, and this skin manto test, which we have said, that is also available for cattle to look into. So they give that, the antigen, and there is a swelling of the skin. So based on that, they will, detect that this is a case of TB. And another form of TB in animals is that there is excess of, diarrhea. There's a, yeah. So that is the one which they are. And the treatment, I am not very, aware of those things which they are doing into veterinary sciences, but my experience is limited to, the human infection.
Okay, thank you.
Mycobacterial Jones disease, that's the one which is very prevalent. One thing I would like to say, this podium I have come after 40 years. When I was a student, PhD student, in 1986, I came here to present, and then I was, I became practically where I am sort of thing. And that time there was a, light arrangement also available. If I want to show you some dim slide, I can do it myself. Then I thought, oh my God, where I am. Hats off to you, sir, for making such a, I have not seen such a, unique auditorium even after 40 years. So this is a wonderful planning. Of course, this is one of
even after 40 years, the institute and the auditorium remains young and beautiful.
Yeah, I'm a loud guy.
Yes, sir.
Well, it's a privilege to invite next speaker, Dr. Vinay Nandikuri, Director of CCMB Hyderabad and a staff scientist at this institution. He's very well known for his work on the kinases, their networks, what they do to the mycobacterial biology. I'm thinking he will share many glimpses of his work, which he has sustained over a long period of time.
Thank you.
Dr. Vinay.
Thank you, Dr. Katoch and Dr. Javed. I have given many talks from here, mostly for RAPSAC, and this is the first time I'm actually giving a talk in the honor of, sir, a belated many happy returns of the day. So 99 going on 100, it's something amazing. Here it is. He is our founder director. We all know about it. Congratulations, sir, on your 99th birthday on 2nd October. Somebody actually told me this, and not my own idea. What if there is a podcast interview of Professor Talwar? What would you call it? I couldn't give an answer, but if any of you can, it will be great. Chat GPT. This is something I got online. It kind of fascinated me. This was the original idea, architectural picture of what NII is, and it took a while for me to figure out what is which block. But here is the road and the plan. And this is how RSH looked in those days when it was getting constructed. And this is another picture which I found again in the on web, and the A block, F block. I mean, beautiful, beautiful view. And I spent almost 17 years, and I'm still associated with NII, and I'm on deputation from NII at CCMB. I had the privilege of working in this institute, fantastic institute, and this is where I have done most of my work on tuberculosis, which I promise you I will finish in my time. So many students have worked with me, and many of them have graduated from NII here, and I acknowledge all their work. I'm not going to present most of their work, but nevertheless, these are the people who have worked with me over a period of all these years. This is my current lab at CCMB. I have moved my lab from NII to CCMB, and though, you know, it's pretty much like I'm continuing the work that I have started here. TB as a disease, I'm not going to talk about it. This has already been covered, and its extrapulmonary tuberculosis is also covered. What I started my work when I came to NII was on signaling. I used to work on prostate cancer, and I knew how to work on kinases. And I came here, I started working on kinases in Mycobacterium tuberculosis. And from there, it led me to various other aspects, simply because kinases actually phosphorylate substrates. So when they started looking into what is the impact of post-translational modifications on these substrate and how that impacts the Mycobacterium tuberculosis biology. And in the because of that, I got into various areas, including metabolism, peptidoglycan synthesis, cell division, and and cysteine metabolism, to including all the way up to evolution of drug resistance. But what I'm going to today talk about is one story that we have recently finished, rather than give you an overview of my work for the last 17, 18 years. So it's done by one of my students, Yogita Kapoor, who is currently in Harvard, and it is done in collaborations with Thomas and Neeraj's lab and Debatrin and CCMB. Mycobacterium tuberculosis is a very difficult disease to treat at a molecular level for this following reason. Division is not equal. When one cell divides, it's not like you're going to get two daughter cells of the same size. One of them here is a small cell, the other one is a big cell. In fact, because of that, what happens is when you treat it with drugs, it is not easy to kill because of this heterogeneity in the division. So, and division also has many proteins that are present in E. coli are not present in Mycobacterium tuberculosis, and it's not completely understood despite years of work by various people. So we have been wanted to work on Mycobacterium tuberculosis cell division, and one of the reasons I started working on cell division is because many of these cell division proteins are also substrates of kinases. So phosphorylation modulates how the cell division happens. In fact, if you deplete some of the cell division, phosphorylation, one of the kinases, cells become elongated or bulky and eventually die. So one of the proteins that I'm working on, we were working on, is a protein called Div4A. And this is a protein which is present at the poles and at the septum of the cell, and they have a very typical structure like this. So there is a domain called Div4A, which is essentially this peptide repeated multiple times over. What is interesting about this protein is it doesn't have any activity of whatever, like in any kind of enzymatic activity. All its functionality is to sit at the pole and septum and be a scaffold, something like a ladder which helps in getting other proteins come to the particular place. And so there was a paper there were papers were published well before we started working on it, where you have this particular protein either present in the pole or septum, and if you take them out, it becomes like a round cell. And eventually, after some more time, it dies. So our idea was to figure out how this scaffold is functioning. What is known is this phenotype. What was not known is how exactly it is happening. So that phenotype, when we did in our lab, we generated this mutant in the lab, and you can see this is how it becomes when you start depleting BAC31 or this cell division protein. So we assumed, I mean, we started doing a TEM analysis, which surprisingly nobody had done by then. What we found is when you deplete, you get a cell that looks like this. So if you don't have VAC31, why is it looking like this? We speculated that it is because it is accumulating lipids. So if it is accumulating lipids, then we should be able to stain with a lipid specific dye. And that is what happened. We could stain it with lipid specific dye, and it is indeed accumulating lipids. The question is why a scaffold depletion leads to accumulation of lipids, right? So we did the interactome. I'm trying to make it as brief as I can. We did the interactome and identified that many proteins that are involved in cell division and cell wall synthesis processes are actually interactors of VAC31. These are the top 25, but we got around 100 interactors of VAC31. And these were validated also. The interactions are indeed true. So one of the questions that we had is, we have a cell division protein or a protein that is involved in cell division, and it's a scaffold, but how is it actually sitting at the membrane? Is it directly holding on to the lipids? Nobody had actually looked at it or shown it. So we collaborated with Thomas Piccadille's lab at ICER Pune to look into this. What they do is they do an assay called PlyMap, in which they put in some of the fluorescently tagged lipids, and what happens is, if the binding happens, it will actually show as a fluorescent signal in the SDS page. So after doing this, we run it on an SDS page and look for the fluorescent signal. So you can see that there are multiple different lipids can be generated. Lipids can be used for generating this liposome. For example, PE, okay, PG and Cl is cardiolipin. So all these things were used independently, and what you can see is that while PE and PG do not bind to this particular protein, the cardiolipin very clearly binds. It is maybe looking like why only one particular lipid binds. So obviously because cardiolipin has more negative charge on it. And second aspect is also cardiolipin is known to be located at the poles. So essentially saying that there seems to be a connection between lipid binding and the localization to the poles, and this is pretty much clearly done multiple times, and this is what we get. So it still didn't make any sense. How does it binding to one lipid helps? So this is the structure of VAC31, but for the purpose of biochemistry wise, it is a tetramer with C-terminal regions involved in in stacking, and the N-terminals are relatively free. So we basically generated a deletion mutant of 60 amino acids from the N-terminus, where the VAC31, the D4A domain is being taken out, and we find that the binding goes down. Mind you, this is in vitro. Binding goes down, doesn't get abolished. But what I've found is that N-terminal region most probably is involved in binding, because we see a significant decrease in the intensity, which pulled into the deletion of the N-terminal region. And at that time, we were kind of still, I had this particular hypothesis that it most probably holds on to the lipids and allows the other proteins to load there. But how do you prove it? So there was an assay that was there called basically tethering assay. What you do is you spread lipids on a slide and add the protein. If the protein is involved in bringing lipids together, then you will get droplets become bigger. So first one is without any protein, and it is like the lipids are all there. But if you add VAC31 as a protein, now you see what happens. They're all those lipids start coming together. So there are two things that we have figured out, that basically you have a scaffold which binds to lipids and most probably is helping in bringing lipids together, which means that it is not one end, there's more than one end that is binding. So why does it happen? It's the tetramer. As I said, the N-terminus are free, right? We then went on to show that you need the N-terminal D4A domain for this particular activity. Without the N-terminal domain, which has D4A, this tethering activity is abolished altogether. So what are the biological consequences of not having lipid tethering activity? That was the next question that we had. And what we did is also that cardiolipin is also known to bind, known to localize to the poles of a cell. What happens to the cardiolipin localization? what is the impact of not having lipid binding domain? These are the two questions. Let's start with the cardiolipin. What happens? Its localization. You can see that cardiolipin in a normal cell is localized to the pole, which is well known, nothing new. And you can look at the graph and you can very clearly see it comes poles and septum. Now you take away VAC31, it's all over, which is this one. Now you complement it back, you can actually get it back to the same pole and septum thing. Overexpression also changes the localization, which essentially means that VAC31 levels in the cell are linked to the localization of the cardiolipin in the cell. So which is not a direct correlation, but it is reasonable to say that these are connected to each other. So that's when we actually went to the mutants that we have generated and complemented with the mutants. So what we have shown is that when you take away the N-terminal DIP4A domain, this is a wild type, and when you take away VAC31, you get a cell death. And when you put back the VAC31, cell death is not there. When you put back a VAC31 with a point mutant, cell death is not there. But when we put back a VAC31 which doesn't have N-terminal 60 amino acid, you get cell death. It's a very clearly the N-terminal the DIP4A domain is very much required for the cells to survive. So the question is why, right? So we looked into the cardiolipin localization when you complement it with the N-terminal deletion, and what we see is what we expected hypothetically. Here is the depletion of VAC31, and you see that cardiolipin localization is all over. And I put back the VAC31 back, it's back to pole and septum. And if I put the one which has the point mutant is still at the pole and septum, but when I put back the one which doesn't have the N-terminal, even the cardiolipin localization is abolished. So it took a lot of things, and then what is the role of the C-terminal domain, right? N-terminal domain is very clearly binding to the lipid. We have identified a bunch of interacting partners. What we did is a very classical biochemistry wherein we actually took only the N-terminal domain, or took only the C-terminal domain or the full length, and looked into its interaction with some of the identified partners. We took almost 4 novel partners and 1 known partner and did the experiment. It's like an in vitro experiment where you mix them and eventually pull down and look for the other partner. So here is the first part. Here is the very clearly see that we have taken 3 proteins, and here is the N-terminal region, N-terminal alone, and you don't get any binding with these interacting partners. But when we took the C-terminal, you can see this is a C-terminal, full length is anywhere interacting, you can very clearly see binding. So what we are essentially saying is that you have a situation where a protein N-terminal seems to have a very distinct function, binds to lipids, and the C-terminal has a very distinct function, binds to proteins. So based on that, what we think is it is almost like a local train, which is basically you are holding on to 2 ends, the lipids are holding on to the lipids are held by the N-terminal region, and the C-terminal provides as a landing pad for most of these important proteins, and that's how actually Mycobacterium tuberculosis cell division is regulated by one scaffold protein. So there is, this is one of the things, but you know, when we were working way back into 2017, we were working in cell division in Mycobacterium tuberculosis, and we did a pull down with one of the cell division proteins. What we identified was another novel yet unidentified DIP4A domain containing protein in those days. Later on, some other lab has also identified. So we had this particular intriguing question: are there only 2 DIP4A domain containing proteins in Mycobacteria? So what we can say is that we have identified 2 more proteins.
Dr. Binay,
yeah, no problem. So we have 2 more, we have identified 2 more non-essential DIP4A domain containing proteins, and we have characterized that, which I am not going to present today. But what I am essentially saying is that Mycobacterium has around 4,000 genes. Even today, despite all the work that has been done by many, 46% of it has not yet been characterized. And there is so much more to be done, even at a basic biological level, in addition to understanding pathogenesis and other aspects. What are these 46% of these genes are doing? That is where most of our has been and continues to be. Thank you very much.
Thank you, Dr. Vinay. And yeah, 2 questions are there, yes.
Do you think, and the cells depend on the cellular architecture?
Yeah.
To find any interaction with active myosin or any cell? Because that may change in the cells.
No, but cardiolipin is actually localized to the poles, and this scaffold somehow maintains the negative architecture. It is shown even in bacillus. So the protein somehow holds on to the negative curvature that is present at the pole, and that's how the cellular shape is maintained, this particular proteins. So other than that, no, it doesn't interact with other proteins which are like that. But it is essentially to do with the lipids. It's mostly with the lipids and the proteins which are involved in the cell wall synthesis.
Thank you.
Oh, thank you very much for that very interesting talk. So the question that I have is, have you checked whether this VAC31 forms liquid-liquid phase separation? Because looking at the structure...
I haven't done that, but it's something that we believe it will be, but we have not done that.
Because that seems to explain quite a number of observations that you have shown in your talk.
Possibility, but we have not looked at it, nor any other group has looked at it. That is something that we should be looking at.
Thank you, Doctor. Thank you very much. Thank you. Wonderful. Your talks leaves a lot of scope for future, you know.
So we have a speaker, Dr. Sheetal Gandhotra, and she's a principal investigator at the IGIV New Delhi, and today she's going to talk about the granulomas and the role of foamy macrophages. So over to you, Dr. Sheetal, and you have 20 minutes.
First of all, thank you to the organizers for inviting me here. It's really an honor to be coming here to give a talk on this day. Congratulations NII for your Foundation Day. I am sorry to be standing between lunch and you. These structures are not rasgullas and gulab jamuns, but they're actually lipid droplets. So these beautiful structures are what store free fatty acids and cholesterol esters inside our cells. And the reason why we are interested in these structures is not solely from the point of them being sources of fatty acids, but also sources of proteins that sit on these droplets and carry out several functions, which we are interested in. So before I start, there are a lot of people that I need to thank. These are PhD students, mostly at the top, who have contributed to the work that I'm going to show today, and they're ably contributed by several other people at the lab. So you will see some of their names coming up at the bottom right of my talk as I proceed. So the question that really motivates us is this idea that there has to be something actionable in the granuloma of tuberculosis. And what that actionable point could be are perhaps the battlefield that is created in tuberculosis, which is a battlefield of lipids. So these lipids are stored inside cells, particularly macrophages. So this layer of macrophages just outside the necrotic core are also called foamy macrophages because they don't stain with hematoxylin and eosin, but they do stain with lipid stains like oil red O. And we can basically mimic this in susceptible animal models as well, such as guinea pigs and C3-HEP-FEJ mice, also in marmosets and rabbits. And what's peculiar about these lipid storage organelles is that unlike atherosclerosis, where the lipid is mostly cholesterol ester, here it's mostly the humble lipid triacylglycerol. And if I just take you through the basics of how these structures come into existence, they're made in the bilayer of ER membrane between the two leaflets, and then they're projected outwards towards the cytosol by the action of phospholipids and proteins. And once they're in the cytosol, they can exist as independent entities making intimate interactions with other organelles, and they have several proteins on their surface. So key points to remember is they have a monolayer of phospholipids, core made of neutral lipids like triacylglycerols and sterol esters. They make stable and transient protein interactions, which leads to their dynamic proteome and somewhat cell type specific proteome. So why lipid droplets have caught the fancy of several researchers like me across the world is this very beautiful and compelling data from Frédéric Alter's lab in France, where they showed using electron microscopy that Mycobacterium tuberculosis, which are seen here, can make intimate contacts with these lipid bodies. And these lipid bodies or lipid droplets can be contacted by the bacteria directly or by the compartment in which the bacteria reside. So of course, this makes us think that maybe there is vectorial transfer from the lipid droplet directly to the bacteria. And in fact, that's true. So we can do that by labeling macrophages. So we use THP1 macrophages. They inherently make very nice lipid droplets, mostly consisting of triacylglycerol. So it's relevant. So we can label the lipid droplets using the fluorescent fatty acid BODP FLC12. And when we infect these macrophages after having labeled them with MTB, we can see by microscopy that the bacteria also get labeled by the fluorescent fatty acid. So you can pulse and then chase the fluorescence into the bacteria. However, if you add a lipase inhibitor, you can prevent this, and now the signal is mostly retained in the lipid droplet. So this tells you that there is vectorial transfer of lipid from the host lipid droplets to bacteria. But if you deplete the lipid droplets, it absolutely makes no impact on the growth of the bacteria. So while it's a carbon source, it's not an essential carbon source. So we decided to ask whether lipid droplets are relevant in vivo in infection. And to address that, we use this very specific inhibitor against this enzyme DGAT1, which converts diacylglycerol to triacylglycerol. This inhibitor is called T863, previously very well characterized. And when we treated infected mice with this drug, we could actually lower the lipid content in the granulomas. And then with 2 weeks of treatment, we actually surprisingly found a decrease in bacterial burden. Why I say surprising is because of the previous slide, where in vitro we did not find a difference. What you can also probably appreciate is that here we actually saw a bimodal response, which is 50% of the mice actually did not show any change in CFU, whereas the other 50% showed a 25 to 97% decrease in bacterial burden. And we decided to make use of this heterogeneity in asking what is it about the host response that can lead to decrease in bacterial burden after T860 treatment. So when we did whole lung transcriptome analysis, we found that T860 treatment, which was associating with a decrease in bacterial burden, was also leading to upregulation of genes that are involved in normal lung function, like elastic fiber and muscle contraction, and significantly downregulation of genes involved in the pro-inflammatory response. And right at the top of this pro-inflammatory response was neutrophil degranulation. And just to show some validation, again 50% of the animals showed a much lower infiltration of neutrophils. So this is the myeloperoxidase, which is staining for neutrophils, and you can see much lower neutrophil infiltration in mice that were treated with T860 and showed a decrease in bacterial burden. So this is from the same animal, and you can see much lower bacterial burden compared to the control animal. Why this was very reassuring is because also in our macrophage model, what we had seen is that if we make the macrophages become foamy by loading them with necrotic cells, the cells become pro-inflammatory. And if we inhibit the triglyceride synthesis in these cells, the inflammatory response decreases. So lipid droplets, while serving as a carbon source for bacteria, are not an essential carbon source for bacteria, but at the same time they drive inflammation and pathology in tuberculosis. So now the question that we had appearing in our mind again and again was that when we deplete lipid droplets, we're not just depleting a carbon source. What we're depleting from the cell are organelles on which several proteins can function. So we wanted to know what are these proteins in macrophages that are present on lipid droplets, and how does this lipid droplet proteome change in response to infection. So we did a proteomic analysis. We compared live MTB infected macrophages to heat killed and uninfected. So we took out the lipid droplets using sucrose gradient ultracentrifugation, and this was all done with a virulent strain of Mycobacterium tuberculosis. The proteomic analysis revealed that of the 258 proteins that we identified with very high confidence, there were about 86 proteins that were differentially regulated. So a large amount of the proteome was actually undergoing change. And if we binned these proteins into functional classes, we found that there were two classes of proteins belonging to protein synthesis as well as vesicular trafficking that increased on the lipid droplet. I'm going to talk about one example of the vesicular trafficking that we've been working on, and then move on to protein synthesis. So the protein that we chose to look at was RLAP, which was a protein that was very well known to be localized to the surface of lysosomes, helping the motility of lysosomes. And so we thought, since we don't know what it does on lipid droplets, let's investigate this further. So I'm just going to summarize that in one slide. Through this work, basically what we showed is that this top organelle, which again looks like a laddu, is, sorry about that reference, feeling hungry. That's where the GDP bound state of the protein localizes, and the GTP bound state of the protein localizes on the lysosome, which is probably very active in all of us right now. So the GDP bound state and the GTP bound state can actually interact, and that brings about interaction between the lipid droplet and the lysosome, and then vectorial transfer of lipids from the lipid droplet into the lysosome, where lysosomal acid lipase degrades the lipid into free fatty acids and glycerol. Why this was a very exciting juncture in lipid biology is because this direct transfer of lipid from the lipid droplet to the lysosome was a newly described process, and the key players in this process were not known. So this was the first player to be described in that. What this vectorial transfer of lipid essentially does is degrades the lipid, and therefore when you knock down RLAP, you get an accumulation of lipid droplets which are larger in size. And what we also learned from this work is that this lysosomal lipase dependent lipolysis is actually the most important, or it basically contributes the most to the general lipolysis that is taking place in macrophages. This is both in THP1 and in human monocyte derived macrophages. So I'm now going to switch to the other story that I wanted to talk to you about, which is unpublished. So previously we found that there were several ribosomal proteins that we identified as proteins that were increased on the lipid droplet in response to infection. And we had validated at that point that RPL11, one of the proteins of the ribosome, was increased. Interesting to note that calnexin, which is an ER marker where you find a lot of ribosomes to be stationed, was not increasing. So this wasn't likely to be just because of contamination of the endoplasmic reticulum with the lipid droplet, which can be an easy contaminant. So now we started asking, well, are there active ribosomes on the surface of lipid droplets? And there are several ways by which we've tried to address this. There's one assay that I would like to point out. This might be of interest to others as well. So there is a method called puromycylation. Puromycylation is basically the ability of puromycin to bind to a nascently synthesizing peptide in the ribosome, and that can be covalently attach, of course, to the nascent peptide and can be detected using an antibody. However, this gets released from the ribosome, so you add another antibiotic called emetine, so that puromycylated peptide gets trapped inside the ribosome, and then you can visualize where these active ribosomes are. Of course, when you do this assay in cells, you add a secondary antibody against puromycin, or you take puromycin that has a conjugate fluorophore attached to it. You will find fluorescence everywhere. So you cannot really use co-localization analysis with lipid droplets to say whether there are active ribosomes on the lipid droplet. So we modified this method using proximity ligation assay. So here what we did is, proximity ligation assay is basically a way to figure out if two proteins are in proximity of each other by in a distance of 40 nanometers. So you have antibodies raised in, let's say, rabbit and mouse, two different species. Then you come with secondary antibodies against each of these species. The secondary antibodies are tagged to oligos. Then you add a substrate that both of these oligos can bind to. You ligate this, and then you do a rolling circle model replication, and then you can detect this newly synthesized DNA in the cell by adding a fluorophore. So just to give you a view of what this would look like, if you have two antibodies against the same protein, so here we've used perilipin 2, you find puncta of signal around the lipid droplet. So great, this assay works. Does this work with what we were trying to say, which is, are there active ribosomes on the lipid droplet? To address that, what we did is we took an antibody against puromycin and we took an antibody against perilipin 2 and asked if these would be in proximity. So this is a little hard to visualize. I'm just going to move here. So this is basically a GIF of a Z-stack. So in magenta, you see lipid droplets, and in green, you see the proximity ligation puncta, and you can see that several of these localized to the surface of lipid droplets. So we use this assay to then ask that does translation activity change in response to infection? So this is a busy slide in terms of number of groups here. I'm going to walk you through this. So if you compare the uninfected to the infected, so what I mean by infected here is H37RV infected, which is the strain we used for all our previous experiments. We actually see no difference in the proximity assay activity. But if you look at heat kill, we see a drastic and significant drop, which means that cells are responding to the surface of heat kill bacteria and resulting in a change in how they utilize ribosomes on the lipid droplets. In case of RD1, which is a mutant of MTB, which is closer to BCG in terms of its virulence, you see a decline. These are then, so Erdmann is another laboratory strain which is lineage-wise closer to H37RV. We have two South Indian lineage strains which are less virulent actually than the North Indian, but they showed higher PLA activity or higher ribosomal activity on the surface of lipid droplets compared to the North. And here is LPS, which indeed has a much higher, so if you compare this to either H37RV or to the uninfected, you see much higher activity, ribosomal activity proximal to the lipid droplets, which tells me that lipid droplets are possibly responding to infection by utilizing ribosomes that are present on the surface. What does this look like in terms of the nature of association of ribosomes on lipid droplets? So basically we found 4 different kinds of associations or 3 different kinds of associations of ribosomes with lipid droplets. We saw cases where ribosomes seem to be decorating the surface of lipid droplets uniformly. We found instances where we had sort of aggregates on the surface of the droplet. And then of course what we expected to be as contaminants, which is ribosomes in the context of the ER membrane making close apposition with the lipid droplet. And then of course we had those that had no ribosomes on them. And interestingly what we find is that with H37RV infection, we get an increase in the aggregated nature of ribosomes close to the lipid droplet as at the cost of the ribosome-free lipid droplet. So what this aggregated form could potentially mean is utilization of different kinds of
Sheetal, you are left with 2 minutes.
I'll be finished before that. So I would like to summarize now that biosynthesis of triacylglycerol-rich lipid droplets promotes inflammatory response to infection. We've shown that both for MTB and other groups have shown that for LPS as well. The lipid droplet proteome is dynamic and changes in response to infection. Vesicular trafficking and ribosomal proteins increase in the lipid droplet proteome upon infection with TB. R-late-B, which is a small GTPase, promotes lipid droplet turnover by lysosomes, and this process is likely altered in MTB infection. Ribosomal proteins and active ribosomes are associated with lipid droplets, and that's what leads us into believing that translational activity at the lipid droplet is responsive to infection and likely to be strain dependent. And therefore, our current working hypothesis is that lipid droplets are a depot that orchestrate innate immune responses to infection. This is also a shout out to anybody who is interested, any PhD student who is, or master student who is interested in working with us. We have a lot of interesting ideas to be pursued with the translational activity and also the vesicular trafficking. So if you're interested in working with me, please meet me after my talk. With that, I would like to thank the large list of students who have contributed to this work. My collaborators, specifically I want to give a shout out to Vineet at AIMS for helping us with the EM that I showed you. Abdur Rashid Thiam, with whom we collaborated for the lipid droplet work with R-late-B. And of course the SGWave lab at IGIB, the TB lab. all the funding sources who have generously supported our ideas throughout my career so far, and also the facilities that we have at IGIB, which includes microscopy, mass spec, and BS surgery. And with that, I'd be happy to take any questions.
So this was really a very interesting and intriguing talk by Dr. Sheetal, and of course, very interesting, which you have told about the involvement of lipid droplets and its impact on the response, especially to infections. So it'll be, and people may have a lot of questions. Of course, I also have many questions, but probably I'll give priority to first question.
Me too.
Okay, first question. Let us give to our guest. Please.
Hi.
Short and sweet.
Sorry?
Short and sweet question.
Yes. So the question is, do you find lipid accumulation in neutrophils also in MTB? Because that has been found in both aging neutrophils and atherosclerotic neutrophils.
So we haven't used a neutrophil marker. I am. Same mic. Yeah. Can you hear me? So yeah, so we haven't used myeloperoxidase along with a marker for lipid droplets. The problem with doing that is that the data that I showed with myeloperoxidase, that's using paraffin embedded sections, and that process actually delipidates the after. But yes, we could probably use perilipin, a marker of lipid.
We'll talk later. Thank you.
Yes, Vinay.
Okay, Sheetal, you had the differences in the amount of the protein, like colocalization, when you were doing with more severe strain like Erdmann.
So it's actually the activity of ribosomes.
Yeah, I understand. So have you looked into preparing lipid droplets from various different kinds of infections, or like in Erdmann versus RV, and does it get reflected in the proteome?
I wish somebody would give me money to do that, but that definitely needs to be done.
Okay, Amitabh.
Hello, Sheetal.
Hi.
Nice. I have what you show is not only the mycobacterium, large number of intracellular pathogens rich in lipid bodies possibly to acquire fatty acids. So what you have shown is it's acquiring the SH12. Fine. Then you have shown the role of ARN and taxonomal structure. But what about the interactions of the lipid bodies and the mycobacterium containing phagocytes? That's the way they will get the fatty acids.
Right. So we have seen with salmonella, let's say, and MTB. So salmonella, we know that it would make a specialized vacuole and stay within that vacuole, whereas Mycobacterium tuberculosis actually evades. So it actually comes out of the lysosome as well. So if we, not all. So in
that's a yeast or something is involved in that.
Right.
So then how we are getting the FSC?
So there are lipid droplet lysosome interactions present in, let's say, salmonella and in case of MTB. We haven't looked at transfer of lipids.
TB is also known as Raj Ekshama, that it used to be for rich people, kings, but it's also for the paupers.
So,
yeah.
The lunch is in the playground, and there are a few vehicles if somebody wants to use a vehicle to go to because it's drizzling. But also I would like to make a very important announcement that please try to be here by 3:35 p.m. so that we can start the next session. start the next session. That's the biology of infectious diseases and autoimmunity. And to chair this session, I would like to invite on stage Professor N.K. Mehra, Honorable Emeritus Scientist, ICMR, and Vice President, International Affairs, INSA. Please welcome him with an applause. And joining him on the dais would be Professor Natarajan, Senior Professor from the Ambedkar Center of Biomedical Research. And now I hand it over to you to take the proceedings further.
Is there a new list? Speakers? Okay. We are a bit late, slightly late. But that's not very uncommon here. Should we wait for some more time? People are still taking their jalebi and rabdi, I think. You know? Slowly walking in, because after you take that full meal. Anyway, I think we must start. Thank you very much for this session. This session is on biology of infectious diseases and autoimmunity. Both of these topics are very dear to my heart, at least. I'm very happy to have with me my good old friend, Dr. Natarajan. My other friend could not come for Dr. Malviya. He called me that he's stuck up in the hospital. We have a few speakers here, and I want to, very first speaker is Professor Janko Nigolik. Thank you so much for coming all the way over here from Arizona, beautiful state of US, Arizona. And Professor Janko Nigolik is the director of AG's Consortium for Pandemic-Free Future in the University of Arizona College of Medicine, Phoenix. His title is going to be Immunity to Infection with Aging, What Goes Wrong and What Can Be Fixed. It's a very good title for people like us over here on this side, as well as on the front row.
All right, so thank you so much for the invitation. It's, of course, an honor to be here in the temple that Professor Talwar has built and from which many other temples have been populated. Now, he's not here to hear this, but when I was about 18, 19 years old in medical school, my parents worked in Ethiopia, and I went to visit them, and that's where I bought his book, and I have enjoyed reading it. It further cemented my idea that I'm going to go to basic science after getting my MD degree. So let's talk a little bit about this. So you've heard from Professor Balaram that it's all about survival and reproduction. However, most of us end up living quite a bit after that survival and reproduction. In fact, we survive probably somewhere between 30 and 80 years after we have reproduced, so depending on the exact times. So let's talk a little bit about aging and immunity and how that all works out, because as we go along, having a good immune system certainly helps a great deal in dealing with aging. So my lab has plenty of Indians, more and more by the day. It's soon going to become ethnically clean and totally Indian. My wife is Indian as well, and for the disclosure purposes, I have really enjoyed the jalebis. They were almost as good as the ones in Ghaziabad, where my father-in-law lives and takes me to the live making of the jalebis. So, but today's work has really been done mostly by Sandeep Sonar and Ruchika Bhatt. Ruchika's here in Bhopal, actually working as our bioinformaticist from afar. And Sandeep is fortunately on the ground. And a lot of these stories have been started by other people in the lab before them. So when we think about the adaptive immune system, and that is what I think about most of the time when I'm in the lab, when I'm not doing other things like being an associate dean for research in Phoenix and so forth, I'm focusing on these 2 points where there's clear intersects of immunity and aging. The first one is the most important to me as a challenge, which is how do we survive an acute infection when we're older? And in particular, the infection that we have not experienced before. And so when we look at that, if we look at any infection that has penetrated into the human population as an outbreak that we did not know before, that we did not have some immunity to before, and certainly the case of SARS coronavirus is a good one to go through the numbers, these diseases are very much life-threatening to an aging host, and the numbers are really unmistakable. So there's this log linear vulnerability to infection. For every linear increase in your lifespan, your chance of dying is multiplying logarithmically. So that for this particular last pandemic, for everyone dying between the ages of 18 and 39, we had 270 people dead over the age of 85. But that's not 270%, that is 270-fold. So it's an enormous vulnerability that we need to resolve. So that's one problem. The other problem is that dysregulation of immunity can accelerate many diseases of aging. They may even induce age-related tissue dysfunction. And we don't understand yet how is this all happening, but now we know for sure that there are certain flavors of memory T cells that are capable of aging the whole organism upon adoptive transfer. And those cells arise naturally in both mice and in humans, and they can dysregulate function of different organs. So understanding that part is also going to be really, really important. But we're going to ignore that part for today's talk, and we're going to really focus only on how do we deal with acute diseases in the course, infectious diseases during aging. So this is what the geriatricians would tell you, that there's an increased morbidity and mortality from infection. And I already told you that this susceptibility really goes up. And if you want to know when you're really old, ask your immune system. Already at 45, you will be more vulnerable. At 50 for sure than when you were younger. And poor responses to vaccination used to be universally the case and the problem. But we know now that with the appropriate stimulation of the innate immune system, we can overcome many of the poorer responses to vaccines in older adults. But T cells are very strongly affected, and they're of course strongly affected, we know, because thymus is the first organ to involute. Already by the time we reach puberty, our thymus has hasn't stopped producing, but is producing 90% fewer cells than it used to when we were younger. At the age of between 40 and 50, the thymus is going to drop its production by another tenfold, so that only 1% will be produced. But it will not be dead, which gives us some hope that we might be able to bring components of the immune system back. So what my laboratory has been trying to understand is what are all of the defects in immunity to acute infections with aging, and which are the ones that are most fixable, which are the ones that are primary, as opposed to the other ones that are perhaps compensatory or adaptive. So when we look at how we respond to infection, let's say that this is our coronavirus coming in through the nose or throat, breaching the epithelial barrier, we have an immediate action of the innate immune system. Both the cells and the molecules are coating the virus. And then we have the magical cell, the dendritic cell, that is going to pick up the virus, bring it to the lymph node, present it to the T cells. The virus is going to flow, the free virus and the virus components are going to flow to the B cell zone. They're going to activate B cells. B and T cells will talk to one another. And after a few days, we're going to have a nice, decent protective immune response with some antibodies and T cells that are protecting us. But what goes all wrong with aging here? So some of the bottlenecks that exist at the point of recognition and intake of microbial pathogen, and others certainly exist at the level of the crosstalk between a dendritic cell and T cells. There are fewer T cells that we had before that are specific for any antigen, and the dendritic cells are not nearly as well activated as co-stimulatory and so forth. But now going back to what are the causes of all of these things, and are there cell autonomous problems that we accumulate as defects with aging? You look at a lot of literature, like 40 years of 50 years probably of literature, they would tell you that naive T cells go bad with aging somehow. That they accumulate some age-related defects that are then a barrier to their normal function. And so we went to actually investigate this in greater detail with modern methods and with very stringent ways of purifying these cells. And we found that while the numbers of CD8 T cells with aging are reduced by 60 to 80%, if we take these cells and we compare them on a one-to-one basis to young cells in a young environment, so these are adoptive transfers done in mice, we can find out that they really have no measurable defect that we can find. They work really, really well. Whereas if you take perfectly good, naive adult T cells and you park them in an old body, they will not function at all. They will not function well. They will function on par with the old cells in an old organism. They will not proliferate well. They will not differentiate well. They will not become good defenders of the organism. So then we really wanted to know, well, if we lose most of these cells, can we just get the cell numbers back? And so we tried to do that by thymic rejuvenation. You can do several interventions to reawaken the thymus. Sex steroid blockade is one, provision of certain growth factors is another. Both of them work in a similar way. They give you a transient rejuvenation and growth of mass in the thymus, and the cells come out in the periphery, all of these naive T cells. But we found that this failed to restore the protective function of T cell responses. And so, because the cells that were made in the thymus actually did not go to the lymph nodes where we need them to function and in other secondary lymphoid organs. So there was a problem of you can produce them, but they're not going where they're supposed to go. Now, why are they not going where they're supposed to go and functioning there to defend us was our next question. And so we've described several defects in secondary lymphoid organ homeostasis in response to microbial pathogens, along with other colleagues in the field in both T and B cells. So let's go on that journey a little bit. Before we started this, I had absolutely no idea what all happens in the lymph node, particularly in the non-lymphoid cells. And a lot happens there. There's a whole structure there that is critically important to naive T cell survival and also to activation of T and B cells that is very, very critical to happen in an orderly way. What underlies all of this are the niches and the microenvironments that exist here. You can see it here that B cell zones in green are completely separate from T cell zones in red in the adult lymph node. And underlying all of this is the reticular network of fibroblastic cells, or FRCs, fibroblastic reticular cells, that make these dense connections. You can see here that these cells are interdigitating, connecting to one another. Dendritic cells and T cells crawl around this cell network. B cells do as well in their own zone. And if this network is not around, then there are problems. So these are the molecular interactions that occur here in terms of maintenance of naive T cells. The most important here is provision of interleukin-7 on these conduits and on the extracellular matrix. So if you measure IL-7 elsewhere in the body, in the serum, plasma, and so forth, that is completely meaningless to a naive T cell. A naive T cell has to crawl around these structures, and actually that is where it's only capable of perceiving the IL-7 signal. So if you take two simple molecules, CD31 and GP38, or podoplanid, and you stain all of the stromal cells, so now taking away all of the lymphoid cells, you can find 4 subsets. FRCs are the most numerous. These are the ones that make this network. Lymphatic endothelial cells, really, really important also around, and then blood endothelial cells. And so that is what was known. And so then some brilliant experiments from Shannon Turley's group and also from Sanjeev Luther's group shown that if you eliminate these fibroblastic reticular cells, this is the diphtheria toxin engineered to actually only selectively deplete the fibroblastic reticular cells, you will get rid of these cells over 72 hours almost completely. And in parallel, you will actually also lose T cells as well as B cells. So these cells are absolutely crucial to the maintenance of the cells that we need in those stations where the immune responses are initiated. And as a consequence of all of that, now you have this mess between the T cell and B cell zones that are not anymore delimited from one another. They're actually overlapping, which coincidentally is exactly how an old lymph node looks like. So this is where we came into this picture, and this is where Sandeep Sonar in the lab actually just went to enumerate these stromal cell subsets, and he found that effectively at the end of the mouse life, or when the mouse is qualified for pension, you have the same numbers of these cells as you did in youth, at least by staining with these markers that we've had here. But that the numbers of LEX have drastically diminished, they have crashed by 2/3, and that VEX were fairly normal. So again, that's a relatively crude way of looking at these cell subsets. So what Sandeep then did, he looked at the structure of these fibroblastic reticular cells, and we're only really focusing on the T cell zones here in blue. This is at 3 months in a superficial lymph node and in a deep lymph node, and we're magnifying this and then looking at this network here by red staining. You can see that the network is really alive and well and looking really very, very good when the mouse is 3 months young. Already at 9 months, that structure is lost in superficial lymph nodes, and it's still maintaining deeper lymph nodes. And then finally, when the mouse is bona fide old, that structure is lost from all the lymph nodes and down the line also from the spleen. So there's a problem with structure. So let's then take a deeper look, and the deeper look, of course, is when in doubt, do single-cell RNA-seq. That's what we do these days. And we found out that there's a, beyond the total numbers looking normal, there was a very profound redistribution. Effectively, the two main subsets that are present in the adult were completely depleted in the old. And instead, we found these additional subsets that we didn't quite know how to explain all of this. So we looked deeper and we tried to figure out where is this all happening spatially. And the two subsets of FRCs that were gone with aging were right next normally in the medulla to these LEX cells that were also depleted. So the three subsets that we physically were missing in the old age were all in the same zone and lost from the same zone. It's a perivascular cell zone. We're still trying to understand what is the first initiating event that makes them get depleted. But the key question here, when we think about this, is like, is this a block of differentiation? Is this transdifferentiation maybe of these cells into these cells down the line? And are these cells functionally normal, active, and able to do what they're supposed to do? So one of the clues that Ruchika got from this by doing deeper transcriptional analysis was to find this tuft of CD34 positive cells. CD34 usually means that these cells are precursors. And so now we're doing lineage tracing, because if you look here, all of these cells up here look like they're depleted on this side. And so we're really losing them quite a bit. So we're in the process of doing these experiments. Now we're trying to figure out whether the FRC subsets that are still there with aging are transcriptionally healthy. And so when we did that analysis, we found out that while in youth, these cells are producing extracellular matrix and are looking quite fine, in the old age, they were fighting for survival. They had every stress pathway upregulated here. In order to confirm that, we went to the cellular analysis to look for mitochondrial and metabolic defects in these cells. We found that, just like every fibroblast has been described with aging, our cells also are showing increased mitochondrial mass, which is not good, and decreased mitochondrial membrane potential, which is also not good, and finally an increase in reactive oxygen species, and then FRCs also in cellular oxygen species. So that allowed Sandeep to do these experiments of co-culture of naive T cells and stromal cells to see how are they surviving. The main function of the T cell of stromal cells being to maintain naive T cells. And so when he did that experiment, he could show very clearly that old stroma here in black is maintaining T cells much worse than the adult stroma, and that was independent of the age of T cells. These are live cells, these are apoptotic cells, so of course one is the opposite of the other. That correlated to lower levels of BCL2. We could actually improve this by pre-treating of stroma with global and mitochondrially targeted antioxidants, and actually also importantly with inducers of mitophagy. So urolithin A is an inducer of mitophagy. You can see here that adult cells are maintaining naive T cells much better than the old, but that you can bring that back up by providing urolithin A in a dose-dependent way. So based on all of these experiments, and I'm going to skip this experiment in the interest of time, we did an in vivo experiment with the global antioxidants, so just using N-acetylcysteine, oral gavage every other day, and then providing the challenge of West Nile virus and looking at the immune responses. We could bring back the numbers of CD8 T cells responding to West Nile to the adult levels in the draining lymph nodes, as well as in the blood. Sandeep also showed that differentiation towards cytotoxic phenotype also was completely corrected by this intervention. But now we have to prove this in a lot more specific manner by targeting these specific cell types using nanoparticles and other mechanisms. And I'm going to conclude just by showing you that other leads that are coming from here are that there's a coordinated defect in transporter function. So again, metabolism being very critical to how these cells function. This is likely telling us that a coordinated slowdown of FFC metabolism could be a target for our immunomodulatory interventions down the line. So with all of that, I will just summarize that age-related changes in secondary lymphoid organs as the lymph nodes and spleen substantially erode protective immunity with aging, that all stromal cells exhibit loss of architecture, numerical loss of some cell types, and oxidative and metabolic stress in others. Antioxidant treatment can reverse these problems to a good extent, but we need to figure out whether this is really working at the level of the stroma. And we're continuing our detailed molecular analysis of all of this and hopefully progressing towards some trials in humans that we would love to do. Thank you very much.
Thank you very much, Janko. Thank you very much. We have time for one question from the audience and one will be from me. Anybody from the audience? Let me go with my question. I saw that in your slides you said that certain types of memory T cells are more prone to age. Are you talking in terms of the central memory cells or the peripheral memory cells?
Yes.
And what about the B cells, particularly the B regulatory cells? The B reg cells are very important now. I belong to the field of organ transplantation, so the B regs have become very important for organ transplant, particularly when you age.
So there's no studies that I'm aware of B regulatory cells in aging yet, but they're definitely going to be an important cell subset to study. Naive B cells so far seem to be functioning quite well with aging, and it seems that most of the defects that have been described in affinity maturation and everything else are a consequence of impaired T cell collaboration. In terms of T cell aging and what are the potentially detrimental T cells, it has been hypothesized for a long time that it will be the effector memory cells, but they're not. Surprisingly enough, it's a very odd subset of central memory cells that are marked by granzyme K that Maxim Artyomov and his group have identified as normally arising in both mice and humans, that upon transfer and transplant can age. And interestingly, granzyme K is not a cytotoxic granzyme. It's an enzyme that modifies extracellular matrix.
Just as a follow-up, in your FRC depletion experiments, did you, when you see the architecture getting disrupted, do you also see an imbalance or a change in the naive to memory T cell turnover?
Well, so those were not our experiments, those were other people's experiments, but they basically see a complete disappearance of T cells from the lymph nodes and spleen. So the disturbance is massive.
Thank you very much. Thank you for your very nice talk. Please give him a big hand. Next speaker is Professor Dipankar Nandi. I think everybody in the immunology group knows him. He organized a great conference, immunology conference last year in Bangalore. Sure, all of you were there, or maybe many of you were there. Dipankar is going to talk about interferon gamma-induced responses. Dipankar is a faculty member in the Department of Biochemistry at the Indian Institute of Science, Bangalore. It's already there. Yeah. Thank you.
All right.
Dipankar, go ahead.
Dipankar, you have 20 minutes allotted, but if you finish it in 15, we'll give you 2 mementos.
We have a very strict bellman here.
Right. All right. Thank you so much. Let me start off by thanking the organizers for this opportunity. I will try and finish it as soon as possible. Okay, so first there's an insertion in my title with our studies with NO. Here, NO stands for nitric oxide and not NO as such. So let me start off with, since this is in honor of Professor Talwar, so I thought I would try and make it a little bit personal. So the first time I had a, look at Professor Talwar was when I was a student, a master's student at MS University of Baroda. He had come to give a talk. And the second brush was while we were, looking for a job here in India, and I was giving a talk in NII. The third meeting was at a mid-progress review because we had contributed to some studies on MIP, especially anti-tumor efficacy. And I think it was around this time that we had gone, he had taken me for a tour at the Talwar Research Foundation. So we thank him for encouraging our studies. I think he's particularly fond of MIP. And this was the AFM picture that was generated by a student of mine, Taru. And I think he likes it and he shows it quite often to others. For the students in the audience and the young faculty, I would urge you to read this particular write-up, which is there in the Journal of Biosciences, A Destiny to Fulfill. It's an extensive write-up and it tells a lot about, doing studies doing science in India. And I think it would be informative to all. So certainly impressed with his ability to nurture a large family of scientists and well-wishers and live life large. Okay. And of course, blessed to have so many well-wishers and as seen by this audience. So we wish him several more years of good health and happiness, sir. In 2016, and I had organized a meeting and I was here. And I was very touched by remarks by Raj Reval, who was also the architect of NII and also related to Professor Talwar. So Raj talked about, some of the tough conditions that Professor Talwar faced while growing up as a child and which may have shaped his personality. More importantly, he talked about his affection for Professor Talwar while designing NII. And he felt, when he designed NII, he put in his affection, his special affection for Professor Talwar while designing. So this is a good time to wish NII a happy Foundation Day too. All right, so now I will get on with my talk. So as mentioned, I'm from the Indian Institute of Science, and this was the old biochemistry department, one of the oldest biochemistry departments in this part of the world. Started in 1921, and we moved to a new setting along with other departments in 2012, where we are right now. This is a collage done by students in my lab, and we work on different aspects. So we work on microbial aspects, we work on host responses, we study T-cell biology, inflammatory response. So I will be sticking today to one aspect of this, which is interferon gamma responses. Okay, as most of you know, interferons are important for host protection, and they were discovered as antiviral factors. So in fact, interfere with viral replication. That was the main thing. There are three main types. Type 1 is mainly antiviral. Type 3 are involved in protection at mucosal surfaces. But I'll be focusing mainly on type 2, which was the interferon gamma, which was identified as macrophage activating factor, and it is important for cell-mediated immunity. I think most of this you're aware of. As mentioned, it was originally identified as macrophage activating factor, well known to play important roles in antigen processing and MHC, especially upregulation, components of the antigen processing pathway, and so on. The way interferon gamma works is mainly through the JAK-STAT pathway. That's the canonical pathway. Tyrosine kinases phosphorylate their transcription factor. It goes in, turns on primary responsive genes, especially like STAT, IRF. These in turn will go in and turn on secondary responsive genes. All right. So like many things in immunology, we speak from both sides of our mouth, which means it's protective as well as it's harmful. So it protects us against intracellular infections. It is also harmful when produced in higher amounts under certain conditions like autoimmunity and so on. So the way we started off was, long time back. This is when I was doing my postdoc. We identified a cell line known as H6. It's a hepatoma cell line. And if you give it interferon gamma, it induces a bunch of proteins, and these are proteasome subunits. And we had ways of numbering them. You can see that 2 is absent minus interferon, 2, and this is LMP2, which is present with interferon. Same is true for LMP7 over here, absent here, present here, and then LMP10. And this is how we identified the 3 interferon inducible subunits. So H6 was a wonderful cell line to look at other genes or other responses, some novel responses by which interferon induces host responses over here. So it's a very simple assay. You grow the cells, and then you can do your assays, you process, and so on. Very straightforward. There's a lot of ROS and NO that plays important roles, and I had no idea about both nitric oxide and ROS, but the person over here who catalyzed my interest was Professor Ram Sharma, and these are 2 of my students at that time. Professor Ram Sharma, informed me a lot about simple experiments with both NO and ROS. But the way we started off was very simple. I was starting my lab. We needed an assay for interferon gamma. And at that time, we didn't have money for ELISA plates and all, so we thought we'll come up with an assay. And one of the ways we came up was, in fact, done by these 2 MSc students over here, where we developed a NO-based assay or nitric oxide-based assay to score for interferon. You put in interferon. If there is IFN, it will produce NO. NO gets converted into nitrite and nitrates, and we assay for nitrite using the Grice reagent. A very, very simple assay. At that time when we published, it was so simple that Professor Manjunath, who was the faculty over here, commented that this is the paper, you get something from nothing. So I took it as a compliment, and we moved ahead. Most of the work was initially done by Jyoti Prasanna over here and Bani. We published a lot of papers because to save on time, I'm not going to go into all the details over here. But I'd like to mention this work done by Shravanti, who showed that interferon responses could be NO independent, for example, upregulation of class 1, as well as NO dependent over here, where you see a lot of this. More importantly, she was the one who actually did a lot on the MIP-induced anti-tumor responses, which also Professor Talwar talks about a lot. Okay, with regard to NO, most of you should be aware, but in case you're not, NO got the Nobel Prize in 1998, and Pujjot was the one who identified endothelial relaxation factor. NO is also important for blood pressure regulation, vasodilation, and so on. And then Ignaro identified this as a gas, and that's nitric oxide. And then Murad showed that if you use nitroglycerine, it can cause relaxation of muscle cells also. So there are 3 forms of nitric oxide synthase, which is important: the neuronal, endothelial, and the eye or the immune. And the one that I'll be focusing on is mainly this, or NOS2. Okay, many applications of NO. Again, most of you might be familiar with this. For the aspect of this talk, the observation started off as follows. You take H6 cells, you grow them minus and plus IF and gamma for several hours, and then you take a look at this at the cell culture supernatant. You can see that the cells that were grown with interferon are more yellowish in color. And most of you are aware that in cell culture, you often use phenol red, and under acidic conditions, it turns yellow. So this was something important over here. This could be if the cells had overgrown. But on the other hand, actually what happens is with nitric oxide production, the cell number goes down. So despite lowering of cell number, there is more higher acidification. And this was the observation that really we studied. So, but if you take a look at cell morphology, there's not too much change in cell morphology. So the initial objectives were to understand the mechanisms involved in this acidification, some of the cellular processes, and then try and see if we could try and get some therapeutic benefits out of this model system. I'm going to summarize this part again because of interest of time. What we found is what happens is there's a lot of lactic acid production over here. And this lactic acid production or interferon gamma induced lactate is dependent on nitric oxide, production of reactive oxygen intermediates, and also HIF1 as a key signaling molecule. So HIF1 gets produced, and that's also important. Okay. And the production of these are important in regulating IF and gamma induced NO and ROS production. In our studies, we found that there are some lines that produce NO very quickly with interferon gamma, but some that don't. The ones that don't are resistant to the effects of interferon gamma induced cell growth, drop and so on. And what we could do is using some of our understanding over here, we could affect the growth of these non-NO producing cells using some of the information over here. And this became important over here, and it's shown over here. So this is what happens. There are two cell lines that we use, CD26, B6F10, both of which don't produce NO with IF and gamma. But what we could do is we could have some other molecule that we could use from the signaling cascade as well as lactate to show reduction in cell growth. Why is this important, and why am I emphasizing on this? While we were studying this aspect, we realized that with regard to anti-tumor therapy is often inefficient, especially checkpoint inhibitor therapy. You have about 25% efficacy, right? CARS is much better with about 80% or so on. But interferon gamma plays a very important role. So the important part is, can we make use of this information to try and see if you can better checkpoint inhibitors and so on, which is what the main aspect of this particular work was, and so with implications in cancer immunotherapy. All right. So from now on, I'll move to the second part of the talk, which is a recently published work. So I'll spend a little bit time of it, and the third part again is something that I'll summarize. Okay, so the second part was, can we identify a set of novel genes and cellular responses that are NO dependent? For this process, what we did was we used RNA-seq technology to try and identify novel pathways. That's the important part. Okay? But before we get to novel pathways, is the system working? Are we able to identify ones that are already well known? And indeed, most of these are sort of well known, the transcription factors, the G protein, and so on. And of course, the ones involved in antigen processing and presentation. This was actually the start to my work. So when we take a look at interferon regulated genes with disease, obviously these are ones that were identified. But what was really interesting is we also identified LMP7, currently known as PSMD8. Now it turns out that there is a new set of diseases known as interferonopathies, which is an auto-regulatory or an inflammatory situation where there's a lot of inflammation produced. And this is a disease that clinicians look at and treat, and they usually treat using JAK inhibitor. And one of the genes that are responsible for this is LMP7. So if you remember, we started off with the proteasome subunits, identification of those 3 subunits, and now one of those subunits are actually linked to a particular disease or, constitute part of interferonopathy, right? There's a recent paper in Cell. I would urge students interested in this to take a look at it with its link with multiple sclerosis, where during this, what happens is LMP7 gets increased. It leads to altered cellular metabolism, leading to ferroptosis and neuronal death. So, it's really, the circle is, pretty much complete now, because we started off with that, and now we have these parts. Let's come back to NO. So, yeah, I'll finish it. So, with NO, these ones were not regulated. NOS2 and CYBB, which is NADPH oxidase, a subunit over here, were upregulated. So, so our system was working well. But along with it, the student actually identified genes involved in NAD metabolism, right? Now, NAD metabolism, is an important aspect, and that's something that got us interested, okay? So role of NAD, especially in epigenetics, cellular metabolism is very important, and so we started working on this. These are some assays to show, I'll show it over here, that what happens in a time-dependent manner, the NAD-NADH ratio, NAD is produced. And this has become important because NOS and others, they need NAD for proper working over here. Is it NO dependent? And again, with interferon gamma, it goes up. If you block NOS, then it's reduced. So again, showing that it's NO dependent. All right. So we used inhibitors to block the pathway to functionally test. There are different ways you make NAD. We use a blocker over here, and we use something that will enhance NAD. So both ways, right? So you increase as well as you inhibit, and then ask what happens. I'm going to summarize again this part over here to tell you that NAD levels increase with interferon gamma. They do so in a NO-dependent manner. And again, what is important is these genes are expressed in multiple organs in humans, and and in fact, what we found is that several interferon gamma regulated genes are also highly expressed in EBV transformed lymphocytes. Now, which I think is an interesting observation, and something, the roles of these interferon gamma genes in this needs to be studied further. These genes are also associated with phosphorylase deficiency, obesity, and gestational diabetes. So the observation that NAD plays an important role over here is, certainly something important, and we need to look into it further. What is interesting is this is actually a part of the vitamin B3 or niacin, as it's known. It's a nutritional supplement and may improve mitochondrial function, regulate DNA repair, boost metabolic functions, and so on. So these are the people who did the work. Avik did the work. Sai is an undergraduate student who helped with a lot of the bioinformatics, and Sreyashi did the work with the NAD part, showing functionally that it's okay. The last part of it, I'm going to quickly summarize. What we did was to screen using our model system. We wondered if we could screen for molecules, novel molecules that can affect NO production and result in any possible translational effects. So this study was done along a collaboration with Deepak Saini, who gave us the LOPAC library, which has all these different pharmacological agents, and these were tested over here. Okay, I'm going to quickly go over this. So using the library, we identified NOS inhibitors, we identified steroidal inhibitors. So this told us that it works. Steroids obviously inhibit, so we didn't work ahead with these. We went ahead with the novel hits over here. We identified 4 different ones. And then went ahead with pentamidine and auranofin, which showed major effects. I'm going to summarize this again, this part of it. So we developed a screen, a high throughput screen. We screened and we identified these to play important roles. These are playing basically anti-inflammatory roles. And previously, no one had shown that they affect NO production. So that is the important part over here. So there's a publication on this, as well as we have applied for patent over it. And so this is Avik, who again did the work, and these are all his screening plates. This was done when lab cleanup was organized, and we decided to take a photo of him with all his plates over here. And this is at the Immunocon that was Immunocon 50, which was organized by AIMS. I'd like to thank everyone. It takes a village to raise a kid and more to run a laboratory. I'd like to thank all the people over here. And so this is January 2020. We didn't know what was coming. COVID was on its way. And this is over here. And so obviously it's the same location, but people have changed. And I thank you for your attention. Thank you.
Thank you very much. Thank you very much, Dipankar. We have time for just one question. Your lecture
went too fast. No, Dipankar, just one. Yeah. Do you have any animal model where you tested?
Yeah, actually, I didn't show it for the sake of time. Both DSS, we used that both in DSS. In the last part, we used it in the DSS model of colitis, as well as we have a salmonella model for systemic. Yes, that's correct. That's correct. Yeah. Otherwise, it's very difficult to get papers through with just in vitro data. Okay. All right. Thank you so much. Thank you.
So my co-chair had to leave. All up to me. The next speaker is Professor Manjula Kalia. She's on. There she is. Professor Kalia, this talk is going to be on the sting. Oh, there you are.
Professor Mehra has to leave early.
Dr. Manjula, and there she is. So the title is going to be The Sting in Virus-Induced Neuroinflammation. Professor Manjula Kalia is an associate professor at the Regional Center for Biotechnology in Faridabad. Research focuses on virology, immunology, and cell biology. With particular interest in flaviviruses like the JEV and dengue. Dr. Manjula, you have 15 minutes. In the interest of time, there has been a change in the speakers. A few speakers like Rahul and Ayub and Ashish has been shifted tomorrow.
So I will, thank you very much. I will go very fast and finish well in time. Apart from my journey, I think probably everybody would know that I'm also an NII alumni. And I would like to take the last couple of minutes of my talk to, reflect on the good old times. But I thank Professor Mohanty and all the organizers. I'm really happy to be here today and to present my work. So I work at RCB, and the focus of my group is the Japanese encephalitis virus. Now, this is a neurotropic virus and is endemic in several regions of Southeast Asia, and has a significant global burden as well as an Indian burden with a huge morbidity and mortality, specifically in the pediatric population. The virus is maintained in an enzoonotic cycle with the Culex mosquitoes being the vectors of the virus, and it is maintained in birds and amplified in pigs. Now, JE is endemic in several districts of India, and epidemics usually coincide with the rainy seasons when the vector density is high. And the clinical presentation is grouped under the term acute encephalitis syndromes. And though in recent times with vaccinations and a global One Health approach, there's been significant decrease in the number of cases in some of the very endemic districts like Gorakhpur. However, we still see news popping up again and again on the outbreaks of JE in India, as well as now it's become a global concern where we see now it's very well spread in Australia as well. Now, how does the virus establish infection? Whenever an individual is bitten by an infected mosquito, the virus will initially replicate in the keratinocytes and in the epidermal cells, and ultimately a viremia is established. Now, in most individuals, this viremia is sufficient to clear the virus. There is a robust innate immune response with the involvement of dendritic cells and macrophages. But in a few individuals, for reasons we still don't completely understand, the virus is going to breach the blood-brain barrier. And once it has invaded the central nervous system, it's going to infect neuronal cells, and neuronal cells support very good replication of the virus, which results in direct neuronal cell death, and as well as huge neuroinflammation, which is mediated by activation of several astrocytes and microglial cells in the brain. So the breach of the blood-brain barrier and neuroinflammation and neuronal cell death are the key pathogenic drivers of the disease. Now, the virus life cycle basically starts with an endocytic uptake of the virus, and being an RNA virus, it replicates in very close proximity of the ER-derived membranes, and the egress of the virus takes place through the secretory pathway. Now, the virus can actually be assayed by looking at virus replication complexes, by assaying for the viral RNA, as well as for plaque assays. And my group is engaged in several aspects of the host pathogen interactions of flaviviruses. And for today's talk, I'm going to focus on one very interesting aspect of the host innate immune response, that is the role of the sting molecule. Now, this study basically originated with a huge proteome-wide study of virus-infected cells. And our global proteome analysis showed that several innate immune responses get activated in infected cells, and Dr. Dipankar has already given a nice introduction about the interferons. We see activation of pathogen recognition receptors, TLR3, TLR7, RIG-I, MPA5, the activation of type 1 interferon through the JAK-STAT signaling, and also secretion of several pro-inflammatory cytokines and other immune stimulatory genes. Interestingly, the virus infection is going to down-modulate certain cellular pathways, lipid metabolism, transporters, focal adhesion, etc. Now, from our global proteomic study, a few very interesting molecules popped up which are not conventionally known to get activated in RNA virus infections. And these were TLR2, CGAS, and STING. And for today's talk, I'm going to focus on our work on the role of STING in the JE infection. Now, the CGAS STING pathway has gained a lot of popularity in recent times, and this is primarily a DNA sensing innate immune pathway. This basically gets activated whenever you have double-stranded DNA present in the cytosol. The origin can be a bacterial DNA, viral DNA. It can be because of micronuclei or DNA damage or damage to the microporia. And this pathway basically initiates with a molecule called CGAS, which will produce a secondary messenger called CGAM, which ultimately binds to the STING molecule. Now, STING stands for the stimulator of interferon gene. This STING on activation will dimerize, will recruit the protein kinase TBK1, resulting in IRF3 phosphorylation and activation of type 1 interferon and pro-inflammatory cytokines. So this pathway is very well characterized in the context of DNA virus infection. However, there are now studies which are coming up and which are implicating the role of STING in RNA virus infections as well. So the first observation that we made was that JV infection results in very robust activation of STING. You can see in virus-infected cells, there is huge upregulation of the phosphorylated form of STING as well as upregulation of CGAS. And the second very interesting observation that we made was that both the STING as well as the phosphorylated form of the protein very strongly co-localize to virus replication complexes. So in the immunofluorescence images here, you can see that we have stained for the viral NS1. And this NS1 protein marks the virus replication complexes, and there is a remarkable overlap of the STING and the phosphorylated form of the protein along with NS1. Now, how was STING getting activated? Like I said, that there is usually a DNA damage which activates the STING pathway. So we did several studies and we ruled out a role for C-gas or DNA damage in the activation of STING. Instead, what we observed was that in the context of JAV infection, STING activation takes place through the interferon signaling pathway. So in this particular set of experiments, we have used a MAV knockout mouse embryonic fibroblast. Now, MAV stands for the mitochondrial antiviral signaling protein, which becomes a very important hub to actually activate the innate immune responses through the RIG-I pathway. And MAV actually localizes through mitochondria and to peroxisomes and is very critical, a critical node in the type 1 interferon activation. So you can see in the MAV knockout met, we don't see any interferon beta production. And because these cells are deficient for interferon beta, virus can replicate to very high levels here, as can be seen from the viral RNA levels and the titers. And interestingly, in the MAV deficient cells, we don't see any activation of C-gas or STING, either at the transcriptional level and nor at the protein level, suggesting that you need to have activation through MAV for upregulation of STING. The other model that we checked was the 81-9 mouse. This is an interferon alpha beta gamma receptor knockout mice, where again they are completely deficient for interferon signaling. And here again we can see that virus can replicate to very high titers. And this experiment has been done in the EMDMs derived from these mice. But we see absolutely no activation of C-gas or STING, again suggesting that these particular proteins are interferon stimulating genes in the context of JE infection. Now, is STING playing an antiviral role? It is. And we observed that through siRNA-mediated depletion of STING in several cell types, in neuronal cells, epithelial cells, microglial cells, we see that virus can replicate to very high levels. If we also use a very potent and specific inhibitor of STING, we can see that again the virus replication increases. And this observation was also validated in BMDMs, which were derived from the STING golden ticket mouse, golden ticket BMDM. This mouse model is a null missense mutation, carries a null missense mutant allele for STING. So there is no functional protein produced here. And again, you can see that the virus can replicate to very high levels, suggesting that STING is playing a critical viral role in virus infection. Now, typically the role of STING is to amplify the interferon response. But interestingly, in the case of JEV, we see that STING does not contribute at all to interferon beta production. In this STING golden ticket BMDM, we see very robust activation of phospho-PBK1, which is the kinase for the interferon pathway, phospho-IRF3, and the interferon beta levels are very high, both transcriptionally as well as secretory form, suggesting that STING is not contributing to interferon beta production. Now, a very important kind of primordial function of STING is actually autophagy induction, and to see if the antiviral role of STING was being mediated through autophagy, we've used another model system, which is the ATG5 knockout system. And this ATG5 is a very critical gene for autophagy modulation. And you can see that also in the ATG5 knockout system, again, we see virus replication going very high in the STING, siRNA STING repeated conditions, as well as high interferon beta levels. Again, suggesting that STING is not playing an dependent antiviral role. 5 minutes. And ultimately, we did transcriptomic analysis of the string BMDM, and we see a very robust inflammatory, drop in the inflammatory response. And interestingly, the STING knockout animals show very poor levels of the innate, the pro-inflammatory cytokines, TNF alpha, RANDs, IL-1 beta, IL-6. And these cells per se are not deficient for inflammatory cytokine production, but they just appear to be very, very deficient in the case of the virus infection. In accordance with these observations, again, we see that these STING knockout animals will succumb very early to the disease. They have very high viremia. And if we look at the markers of the inflammasome, wherein we look at the protein levels of the NLRP3 inflammasome and the caspases. So the pyroprotic pathway of inflammation basically involves the formation of the NLRP3 inflammasome. And there are several caspases which are involved here, caspase 1, caspase 4, 5, and 11, which cleave a very important molecule called gas-dermin D. And this cleaved gas-dermin D can actually oligomerize to form a pore, and they will ultimately release pro-inflammatory cytokines. So we see that virus infection activates the non-canonical inflammasome through caspase 11 as well as the Gas-Germin D. And in the knockout animal model, we see that there is absolutely no upregulation of the NLRP3 inflammasome, no activation of caspase 11 or free Gas-Germin D, again suggesting that STING is mediating the inflammatory response in infection. And very interestingly, we also see that STING is essential to recruit both NLRP3 as well as Gas-Germin D on the virus replication complex. So you can see here in the infected cells, both phosphorylated STING and NLRP3 show a very good co-localization, and this is lost under conditions of STING. So I will summarize my work here to basically inform you that C-gas STING activation is dependent upon type 1 interferon signaling in virus infection. STING has an antiviral role which is mediated through the activation of the inflammasomes, and the virus replication complex is basically serving as a hub for the recruitment of innate immune effectors. That is STING, PBK1, Gas-Germin D, and NLRP3. And current work we are looking at various mutants of STING and to see how it influences the recruitment of the NLRP3 influx. So I end here. I would like to acknowledge this work was done by PhD students Simran Chabra and Ruben Patel. I'd like to acknowledge Santosh and Krishnan for the golden ticket mice models and the MALS knockout cell lines, and my lab members RCB and AMR serve for funding. And I just want to take 2 minutes now. Okay, thank you. So I want to really acknowledge all my NII mentors. So I started here as a PhD student in 1993, and a lot of people know Professor Talwar for SPG, but I would like to inform them that he was also very instrumental in my PhD project, which was genetic polymorphisms of Indian wild mice population. So at that time, it was a huge CEPI-PRA project, which was driven by Professor Kazanev in Institute Pasteur and Professor Talwar from here. And we had a complete wild mice facility was established here. That was my PhD project. I also want to really acknowledge my mentor, Dr. Rajesh Anand, who was heading the animal house at that time. Late Dr. Ramesh Tuwal, again a deep friend and a mentor. Satyajit and Vinita were very instrumental. Satyajit was my SAT committee member. Vinita, of course, I did a lot of work in her lab. A special thanks goes to Dr. Syed Hussain, Dr. Lalit Garg, and Dr. Sher Ali, because a lot of my PhD experiments were done with reagents from their lab. I'm not sure if they knew that, but I had very good friends in their
With
lab.
their knowledge or without their knowledge?
So I'm not sure, because in NII, everything would get done in after hours. You know, I think the labs were really vibrant after dinner, and we could work through the night, and anything and everything could get done very easily. Of course, Dr. Chandrama Shaha and Dr. Dajjendra Salunke have been wonderful mentors. I have to deeply acknowledge Dr. Suvanshu Gati because he brought me to RCB. All the staff of the small animal facility of NII, of course, RSH was just a wonderful place. We spent wonderful moments here. I think those were the best 5 years of my life. I made very valuable friends, all my friends in RSH. And I want to share a throwback picture. This is exactly 30 years ago. And this is one early Monday morning where we have come back after a trip from Missouri over the weekend. And those were just wonderful times. And I'm just so happy to be here. Thank you.
So Manjula, the reason I asked is when you were a student, I was a postdoc in Dr. Hasnain's lab. So I know the drill.
Okay, right? Yeah.
So with Nasreen, someone. So one burning question, if it cannot wait till tea. Okay. Yes, sir. Followed by Amit and then
nice work. I'll be a protagonist of traditional knowledge systems, aware of work, Word Prathan. Have you ever heard of it? Particularly in Japanese institute, Gorakhpur district, which is a endemic area. This work was done by King George's Mental University team.
Yes.
And immunological studies were done at the Sanjay Gandhi BGI. They claim to, and I've seen some of the studies, that while there were a fairly large number of deaths in the earlier periods, that cluster of villages where encephalitis, Japanese encephalitis was very active, it's almost gone there.
Absolutely. It's a huge success story.
It's a huge success story. But why does the modern Western biomedicine not interact with working on such funding? You're working at the molecular level in the animal models.
I have collaboration with
Why
King
I'm
George.
just complete just one. You're talking of holistic immunology started with holistic understanding. As with Professor Talwar, I had this discussion often as a PhD student. But slowly immunology has gone to reductionist, to molecular, to even atomic and so on. Like what Balaram did in the morning. So
Absolutely.
Short answer.
I agree with what he said. Yeah, I completely agree. Yeah. Amit, one question from Amit for old time's sake. Yeah.
Getting back to atomic and cytokine talks, I am now perturbed by my contemporaries, my juniors, totally ignoring the process of cytokine signaling. In previous, the reductionist assumption of nascent oxygen binding the H2O, giving HNO3 to reduce the pH, is something that is, I mean, not holistic, but likely, and Sting not working because of the process of cytokine signaling, then equally prevalent, let's say, mission that I am very perturbed to see today.
I have no answer to that.
That's Amit's point.
Yes. Yeah. Yes. Yeah, I agree. Yeah, thank you.
So our next speaker is going to be Ashish. He was in and out of the program a couple of times and we finally made it. So like we have those 2 rounds of interviews for any faculty position or PhD students. So Ashish, 15 minutes.
Yeah, okay. Certainly.
Same goes for you also. You finish it in 10.
Okay, let me try. First of all, it's very difficult for me to express how I'm feeling right now, because this stage is something, this hall, the auditorium is something very special. Prof sitting there, ma'am sitting there, it just can't be possible. What I'm going to do is talk about something which is not directly related to immunological aspects, directly related to immunology right now, but which could have significant impacts on host-cell-site interaction and hence affect the immunological mechanisms which could be tweaked to prevent a parasitic disease like this. What I'm going to talk about today, again, in our very limited way, we have tried to do the first experiment ever on using nanopore sequencing on field-derived parasites. Just 2 samples. One, Plasmodium falciparum, another from Plasmodium vivax, both from patients showing hepatitis lung. So what exactly were the thoughts which went through us when we started off? Why did we do this? The reason was very simple. We have in the past been talking about various things like natural antisense transcripts from field parasites. We are talking about regulation. We are talking about transcriptome upregulation, downregulation, steady-state processes, all from patient-derived samples. That's a specialty of my group. We work with patient-derived samples. Now, the question which has always been with us is, as yet we are not really clear of the genome or transcriptome regulation at all levels of these parasites, and very little is understood as to what is happening inside the genome host. We wanted to see if there was an additional layer of regulation possible in the transcriptome by methylation signals in the transcript. So, what we did was something fairly simple. Before that, for those who are not into malaria, what we focused on, it's a vector-borne disease transmitted by anopheline mosquitoes. And what we are focusing on now is this part, the asexual blood stain in the host, in this case, the human host. Now, transcriptomics, as we call it, is a new layer in regulation. Nobody has looked at it at the level of the parasite in the host. Nobody has at all looked at it in vivax. And there are just a few falciparum culture studies using other techniques which have been published. Just two. Now, we know that there is 5-MC and 6-MA modification in RNA. Now, why I'm mentioning this is, what we did was we took a fairly simple strategy. We did all our sample processing, extracted the RNA, confirmed that it was caused by monoinfections of Fifax or falciparum through our in-house PCRs. Then we went in for Oxford nanopore-based direct RNA sequencing. Once the data was available, we did an alternative splicing analysis using tools like Claire and Squanty-3, and then looked at the methylation events using tools like M6A-NET, which is for M6A, and CHUI, which is both for M6A and M5C. For M6A, we took the concordant calls through M6A-NET and CHUI. Now, these are two samples that we consider. You can all see the relevant clinical information given. Both of them are hepatic dysfunction cases. No other infection was found in them. There was no comorbidity. This, for all practical purposes, was from falciparum or malaria. No. So this is, we checked this, all these transcript information, against the genomes of the parasite. And what we found was that approximately 50% of the transcriptome, or 50% of the genome, was expressed as transcriptome in these two samples, in each individual case. Subsequently, we looked at the process reads and number of isoforms expressed in PBC and GFC data sets, segregated along sense, antisense, and intersense. So what we are talking about here is this PBC stands for P-Vivax complicated, GFC stands for Lagonium falciparum complicated, and what we are looking at here are different splicing formats, starting from full splice match, genic intron, intergenic, fusion element, antisense, etc. So why are we interested in this? If you look at it under these circumstances, you can already see you are getting fusion events. That is transcript from two genes used together. We are ignoring intergenic right now for this particular talk. If you look at, we are ignoring incomplete splice match, but novel in catalog, novel not in catalog, one in PFC, novel in catalog splices, quite a few. Now, then, when we look at these alternative splicing events together, we find a distinct pattern emerging. Okay? Amongst the different splicing events which we have been able to detect from each of these samples. Now, when we look at the methylation events based on the transcript, we find, to our surprise, we found that M6A and M5C are found on 308 transcripts together in PV complicated, and in 582 transcripts together in PF complicated. As to the best of our knowledge, this is the first study which has shown the presence of these two modifications on same transcript. And that opens the whole new paradigm, because what we conventionally believe in other models is that when you have 5MC, that's a protective RNA protective signal. 6MA mostly is thought to be degraded, depending on the location. What happens when both these methylation signals are present on the same transcript? We don't know at this point. How does one attempt to analyze what would be the possible protein outcome? Now, speeding up, we have seen that along the transcripts, the methylation signals appear to be in the sense transcript for both 5MC and 6MA, tilted towards the 3' end of the CDS, less at the 5'. That is, the 5' UTR has very little methylation, 3' UTR, and CDS has more of methylation. These are the gene ontology enrichments, which I won't go through right now for lack of time, because I think we are running out of time quickly. 5 minutes? Okay. Yeah, and what we see is, if you look at the molecular function of anti-cells, so now we have two categories. One is the sense transcript, the other are the anti-sense. Now, what do we mean by anti-sense transcript in this one? We are talking about the natural antisense transcripts, okay, which are directly against the cell. I'm right now not going to go into implications of regulation of these, but I'm just mentioning that we have seen that some of these MACs are also methylated. Okay? And this is a distribution which shows that in PFC, we have 58 transcripts which have methylation signals in the CDS 5' UTR and 3' UTR, and 18 transcripts. This is for M5C and this is for M6A. Whereas 5X doesn't have any transcript which maps across all the regions in the RNA. Okay? I'm sorry. It doesn't have any transcript which shows methylation patterns across the transcript in all cases. Now, so then comes the next point. You have alternative splicing. You're seeing alternative splicing events, and I'll just show a couple of alternative splicing events, which includes methylation. One is intron retention. These are supposed to go into degradative pathway. Look at this, phosphorylation 6 putative from PVP01 base. You see alternative splices 1, 2, 3, 4, 1, 3, 2, 4, along with methylation signals, both 5M6A and M5C. Now we find this fascination. Then we look at autologs across the two data points. When we look at the autologs, we find methylated sense transcripts 43 for M6A and 27 for M5. We see methylation M6A for falciparum and pivapine, which are supposed to be in the degradative, taking it to the degradative pathway. We find ETRAM2 showing M6A modification. Now, this is possible only when you're looking at it in real life ex vivo conditions. Now, so what I would like to say is what we feel consciously inside. What we would love to do, and for which we have written projects which still haven't come, is do all this on a larger sample size, track it down to the protein level in the clinical condition with ex vivo sample, and see which of these alternative splice events, which of these isoforms are actually giving rise to the protein. Otherwise, some of these are touted and started. You're working with the main protein. If you have an alternative splice phenomenon, and that is being expressed in the host, your all effort may be in vain. So I will not go in detail to all this, just to go down that stress. And NATs are also methylated, including indicating methylation of non-coding RNAs. Isoforms of methylated traces are detected. This is all that I mentioned is mentioned in this paper. The QR code is given in the recent paper. I want to particularly express my thanks to Priyanka and Sukriti, Priyanka and Sukriti. This is my entire team at present. And these are the two persons who led this paper, one working on Palsam, the other on Vivac. ICMR for funding. This is a collaborative effort. Ishan and Pallavi are, Dr. Ishan Gupta is from IIT Delhi, Parma is from CSI Pilani, Dinesh Gupta ji, everybody knows, from ICT. Sanjay Kochar and Dhanpat Kochar are clinicians from Bikani. With that, I will end the talk, and thank you very much for a patient listening.
One question? No? Ashish, thank you so much for finishing in time. So we move to the last speaker of the session, Dr. Nimesh Gupta, who is here at NII, and he's going to talk about gut immune axis in shaping vaccine immunity. Nimesh, 15 minutes is what you have? Yep.
Can you hear me? Can you hear me? Yes. Perfect. A very good evening to all of you. It's a great honor and privilege to be here on podium on the 39th Foundation Day. And I congratulate all past and present members and all my colleagues. We are celebrating this day of scientific excellence and the shared purposes. So, as we look ahead, I'll share a glimpse of our work that we are trying to, understand the human immune system and how that understanding is actually guiding the rational development of vaccines and also strengthening the efforts towards public health. So what we do is we are actually interested in understanding how a long-lasting and a broadly protective immunity is developed. While we try to understand that, we focus on one of the very unique interactions in the secondary lymphoid organ, which is between the T cells and the B cells. It's called T cell B cell collaboration. And a very uniquely specialized subset of T cells is called follicular T helper cells, which drives the development of germinal centers, which are the basis for the development of the breadth as well as the durability of protective immunity. And while we look into this aspect on the CFS subset, the overall idea and the global aim is to harness the positive attributes of these cells for the development of the rational design of vaccines. We utilize some of the models here, because our approach is to, have top-down approaches where we learn from the human and then utilize different models to do the mechanistic investigations. So we use the human infection and vaccination models basically from the most common viruses like Japanese encephalitis and dengue. And in addition to our, the fundamental questions, we immediately translate the knowledge that we develop, while we are studying the human immune system, utilize those, a very antigen-specific and high-throughput T-cell assays to also put up on this new platform that we have established, which is called Human Immune Monitoring and T-cell Assay Platform. We are actually working with the industries and helping them to evaluate their vaccines and the biotherapeutics. And some of them actually have even helped to develop some of the novel T-cell assays so that we can actually, evaluate their candidates at a very early stage of development and give them the confidence to take it further with particular, understanding on the advanced immunological outcomes. Some of these studies which we actually put up from our team, which provided the evidence on a very key role of this T-cell B-cell collaboration, came out in Nature Microbiology on the inactivated COVID-19 vaccines. And very recently, we also showed that how this T-cell B-cell collaboration is very unique towards the development of humoral immunity in dengue, and why we have a very suboptimal and poor quality of antibodies, which are the caveat for the dengue vaccine development. And we are very proud that with this platform, when the vaccine efficacy studies were not available, we were able to help the vaccine manufacturers in the country to do a phase 2 trial on intranasal COVID-19 vaccine, which got approved for UL in 2020 September. So for today's discussion, what will I do is that I will just share a glimpse of the program that we are actually developing in the domain of Japanese colitis. I'll tell you why we chose this model, what's so unique about it to address our question on T-cell B-cell collaboration. And I think Manjula has done a great job in introducing what Japanese colitis, it's a mosquito-borne zoonotic disease. And we are, including India, we have over 100 countries which are actually suffered with this. And humans are the dead end source. So still we are on a safer side that it is not transmitting from human to human, unlike its related family members like dengue, Zika, and so on. Something which I would like to put in the more focus is when the individuals are infected with the Japanese colitis virus, they develop severe disease, 30% of them die. Around 30% or 50%, 30 to 50% of them recover, but they recover with the neurological sequelae. Around 30% of them recovered successfully. Those who recovered successfully, they do not get the infection again. At the same time, another condition in the same demography that exists is the vaccination in India, which is a live attenuated vaccine in the government's national immunization program, which gives protection for initially 1 year to maximum 3 years. So there is a successful recovery from natural infection, which is for lifelong. However, there is a waned immunity in case of the vaccination with a live attenuated vaccine, no adjuvant there, right? So this provides us a unique opportunity to understand what happens here when you recover successfully as compared to here, where you have the virus, it is just attenuated, and it gives you an immunity which wanes down very quickly. So we utilize this particular model to address our question, and we set up a cohort in the northeast part of India, which is actually highly endemic for dengue, and also has a mass vaccination program going on for adult immunization with a single dose of live attenuated vaccine. And then we compared these two cohorts in terms of understanding the T-cell B-cell collaboration, and looked at a very higher granularity in terms of the T-cell responses at antigen-specific level and their durability in terms of their breadth to protection against the virus. So we did here the single dose immunization, followed these individuals for almost 3 years, did the multiple sampling. At the same time, the individuals who were infected, admitted, we followed them until 3 months to 6 months initially to decide who are recovered successfully, who recovered with the sequelae, and then again followed them up longitudinally, looked at the multiple aspects of their immune responses at a very antigen-specific level, recorded that information, and what we found was two important messages. One, not everyone respond to the single dose vaccine in the seronegative individuals. Almost 20% of them do not make the response. And those who recovered successfully have a very unique traits of their T-cell responses as compared to the responders to the vaccine. So we looked into these two groups and showed that both at the transcriptomic and the metabolic level, as well as we also looked into the traits of the T cells, particularly the TFH cells, which are showing a different and unique trait between recovery and the responder group. So for today, what will I do is I will just show you briefly some data which we are looking into this particular group. Why and how these 20% of the individuals are not responding to this single dose immunization. So these 18-20% of individuals do make IgM. They have a very inefficient generation of the T cell and the activated T cells, and they have a very poor compartment of TFH cell responses as compared to the seroconverter group, those who also make the memory B cells and the IgG positive memory, which is not the case with the non-seroconverters. And we did more analysis here in terms of understanding the functions of TFH, because we have some of the unique assays in the lab which can tell us whether the TFH cells are capable of providing optimal health to B cells or not. What we found that there is a very functional deficit at the TFH level, which might be providing an impaired health to the B cell, and thus there is a failure in the classic. So that might be leading to the overall non-seroconversion to the vaccine. So we did some of the global transcriptomics analysis, and we got some hints on two of the aspects. There is a unique enrichment of the TCR in the responder group, and there was a perturbation of the metabolic pathway in the responder group. So what we decided with Bhushan, that he took a challenge to then examine, is there a metabolic basis of this TFH deficit, right? TFH are not functional, leading to a non-poor seroconversion. Why it is happening and how it is happening. So what he did, he took help with Neil Bhavesh at the ITGEV, utilized the NMR-based approach, and looked into the overall metabolite response in the samples from the different groups at the different time points. What he found, if you look into the seroconverters, there is a fantastic separation and a very enriched metabolic response after 7 days of the vaccination as compared to the basal level. We do not see any much perturbation in the metabolic response in the non-seroconverters. He found some of the very uniquely enriched metabolites, and he then looked into the overall correlation with the immune parameters and found that some of these metabolites do have a very strong correlation with these metabolites with these immune parameters like neutralizing antibodies. But because there was no response in the non-seroconverters, we then looked into the baseline level, and what we found is a very unique enrichment of some of the metabolites which are actually showing a very high enrichment in the non-seroconverters. And these metabolites are at a very low level in the seroconverters. So then we decided to choose some of these metabolites and looked into the details. One of the metabolites was a sodium butyrate. To our excitement, we found that this has some relation with what the food habits are in the Northeast, mostly for the complex polysaccharides and the resistant starch. And to our surprise, it is also available in the prebiotics and probiotics, and it's just available in a very low cost on Amazon, and everyone is probably using it just to have a better gut. But more important to that, they are actually promoting these metabolites very widely and globally, that it can actually support the immune system and also support the cellular function. So we looked into the literature. We didn't find any study which talks about the role of the sodium butyrate on the protective immunity, while there are a lot of studies on the inflammatory aspect which have been talked about. So we then explored and decided to look into this particular metabolite. So I have less time, so I will not go into the details. I'll just give you the outcome of this study. So very simple experiments. We chose the drinking water and the sodium butyrate to see what happens to the humoral immunity in the mouse model. And to our surprise, unlike sodium acetate or propionate, which is also the short chain fatty acid, sodium butyrate can have a drastic reduction on the antibody responses to the antigen-specific immunization. And we then looked into more details. We then utilized a setup of the human TFH differentiation, because there was a link with the GC-derived antibody responses. So we thought, okay, let's see if it has any direct impact on TFH cells. We found yes, it can actually specifically suppress the differentiation of TFH cells and can also dampen its function. And that was not happening with the sodium acetate or the propionate. We utilized multiple models. I will not go into the details, but we looked into the TFH and the GC axis. Does it influence that overall dermal center response, like what we have seen in case of the vaccination? Yes, it does affect the TFH, differentiation antibody responses, not only in the antigen immunization, but also in the virus infection. This is an influenza model that we use here. And what we found in a very specific influenza-specific settings, you can see a dampening TFH cells as well as the suppression of the germinal center responses, including the antibodies. So we found that this particular molecule has the capacity to suppress the TFH as well as to suppress the germinal center responses. The last question we asked on this particular metabolite, while it suppresses, how does it matter? What is the physiological relevance of it? You know, some suppression, whether it is leading to an impact on the protective threshold or not. So we utilize one of the new models that we have developed in the lab, which is a Japanese fly disease infection model, where the immune competent mice are challenged with the virus, and then they show all clinical manifestations like human, and they succumb to the infection. And what we found is the mice, those who are treated with sodium butyrate, actually succumb to the infection. And this was due to both the reduction in the quantity and quality of antibodies, as well as the failure in controlling the spread of the virus. So just to summarize, I will not go into the details. We are also looking into why it is happening, how this is actually doing it. Two quick answers. It works by inhibiting HDAC activity in the TFH, and it actually utilizes a free fatty acid receptor 3 to do these interactions, which if you work as an agonist, you can rescue the overall effect of the sodium butyrate. Just to summarize that, what we found through all those human-centered studies, that gut-derived metabolites like sodium butyrate can have a systemic impact on the germinal center reaction by influencing and impacting directly the TFH differentiation, leading to an impaired humoral immunity and a possibility of a poor humoral immunity generation to vaccine or infection. So with this, I just would like to thank my team members who have contributed to developing these programs in the lab. Bhushan was the one who actually led this particular metabolic analysis. We worked with multiple collaborators across the globe. This work was done with collaboration with AIMS, Dr. Siraj in RMRT Assam, and also our collaborators at La Jolla. And thanks to our funders. And thank you so much for your time and attention.
Quick question, Professor Nikolovic.
Hi, Nimesh.
Yeah, hi.
So this, both in your challenge experiments with the whole virus and also the vaccines attenuated, right? You do get CD8 immunity. What happens to that with butyrate?
So yeah, so
you said that the control of the brain titers is lower, and in our hands with West Nile at least, that CD8s are quite essential to that.
So we have previously shown that in case of JEV, it's a CD4 which is actually more prominently controlling the infection. CD8s do contribute, but are not indispensable. So while we didn't look into the CD8 response in case of butyrate, we found that if we deplete CD4s, we can actually deplete the protective response against the virus.
That I totally agree with you, but I think in our hands it was actually cytolytic CD4s that were also really important. So that's what I'm asking you about.
There is a possibility. See, our of course focus was on looking into the T cell B cell collaboration and the TFH aspects. But I think when this whole T cell B cell collaboration is impacted due to the TFH and impaired B cell outcome, there is a possibility that you also have the impact on the CD8 responses. Needs to be explored. That further.
Thank you.
Very interesting presentation. The one quick question, in your Japanese encephalitis animal model, how did you give the sodium butyrate? Did you give them orally, or did you inject them?
No, it's orally. It's in water. It's in drinking water.
And is there a dose-dependent phenomenon?
Yes, there is a dose-dependent phenomenon. So it's a lower doses like 50 micromolar or something do not have much impact. 100 to 200, yes. If you go further ahead, it becomes toxic. So we always choose between 100 or 200 micromolars of the dose.
Thank you.
Last question by Professor Sinha.
Yeah. 20% vaccine non-responder is a pretty high figure when you go to the field.
Yes.
So are there non-responders to vaccines in general? And this, because your experiments of the metabolites are really not in the antigen. I mean, they have an antigen-specific system.
Yeah.
But the effects don't seem to be in that sense antigen-specific.
Yes.
So so these 20% represent a class of, because all of them would be getting vaccines through government program.
Right.
So what is any epidemiology or any in fact translational work which would indicate that these are generalized effects or specific?
I think that's a very good point in terms of generalizing whether these individuals, these are adults, these are all adults of the FABS, the class year 2, and it's very difficult to generalize whether they are not responding to all. We looked into the tetanus responses. So they do make antibodies to tetanus, suggesting that they are not having a completely impaired immune system, that they are not immune compromised in terms of responding to a vaccine. It's possible that what we have found, it's a very, very specific and a context dependent phenomena, that particularly at that particular juncture of the vaccination, they didn't respond to this particular vaccine. This response to this vaccine needs both coordination between innate and adaptive much more than the adjuvanted vaccines, because it replicates very poorly in the peripheral cells.
Non-adjuvanted vaccine
Exactly.
as opposed to the others.
Exactly.
That's the main point.
Exactly. So, but particularly because it's 20%, it's a big number.
It's a huge.
Right, and it has not been reported before because no one was looking into a group as a seronegative separately. Everyone looks into the whole population, where 50% of them have an existing memory, even if they don't have antibodies. So that's where number looks very high, but I think we are probably coming to more answers to why these individuals are not responding to the vaccine and what is that context dependent phenomena which might be happening in this particular population. Yeah, Radni, say that again. No, we didn't look into it. So yeah, we can look at it. Thank you. Thank you so much. Thank you.
I would like to now please request Dr. Sehgal to join us on stage and present a token of our gratitude to our chair, Professor Natarajan.
Talked about serendipity, so I just got the chair because one chair could make it. So I do something, end up doing something else.
And it's not the end of this session because we have 2 talks from this session tomorrow, and I would like a very special shout out to actually Dr. Ayub Kadri and Dr. Rahul Pal, who have very graciously allowed us to reschedule their talk to save some time today, to manage time today. So we now can proceed for tea, a quick tea, and we'll be back here sharp at 5:15 to start the panel discussion. And the tea is being served at 3 venues to make it easier for all of us. So it's the Rock Garden and both the rooms on this side and Seminar 3. Yeah, we'll see each other at 5:15 sharp.
dementia, all of these are now handled through information. So thank you very much. This was my opening remark, and Dr. Talwar was not here. I started with my regards to you, sir. When I was simple assistant professor in PGI, you included me in RAPSAC, and since then I was in the RAPSAC continuously till I grew old. So thank you very much.
Professor Talwar, I was saying that as we celebrate nearly 4 decades of this alma mater of ours where we have done our PhD, cutting edge research, innovation and public service, going hand in hand with your visionary leadership and your legacy that many of us, each one of us is aware of, and your pioneering work that laid the foundation for immunology research in India. This afternoon, this evening, this panel that we have set up here is the forward-looking panel that looks ahead to the next chapter of your legacy. And so therefore, we are now in a position to be taking opening remarks from our panelists. And we have just taken Professor Ganguly's remarks. I would like to now turn to Dr. Renu Swaroop, the former Secretary, Department of Biotechnology, and one of India's strongest champions.
5 years in the government, I can't overlook Hayati.
I know, but he insisted upon it. May I just
thank you so much, and
give your
very
remarks.
good evening to all of you, and our regards to Professor Talwar. It's such a pleasure to be here today, sir, and if I can say this on behalf of everyone, I think all of us are here mainly to wish you and congratulate you. So our wishes. Gitanjali said this is a forward-looking panel, which is to look at how do we take this legacy forward. Tough task. Because the legacy which has been created itself is such a strong legacy that to take it ahead, you really need to think out of box and put in a lot of effort to be able to take it there. This institute, and for people like me, as Professor Ganguly said, I joined DBT in 1989 when the Department of Biotechnology had just been founded, but the first thought for any institute for us was NII. You know, and I was just talking outside that the first child is always the special child. This was and continues to be the special child for DBT. And also, immunology research meant NII. That was really what we looked at. I think if you look at where NII began, and if I may say that today, in the last couple of years or the last decade or so, we've been all talking about translational research, product development, innovation. I think this institute, steered by Professor Talwar and his very, very strong team, many of them were around here, many of them may have moved out, exhibited that even before we knew what translational research was. You know, looking at how basic science moves towards applied science, how basic science gets applied in society, that really happened at this institute. And as I said, I don't think many departments, many organizations were even talking about translational research. We didn't have the words of an innovation ecosystem that existed in the late 80s and early 90s. It's only later. It was globally, but within the country it had not come. So this is the foundation that was laid for this to move. Moving forward, I think what over the last couple of years immunology research has exhibited. In fact, today was, I think for all those in this institute must be very pleased, and it's probably the next step that we need to look at is the Nobel Prize for immunology research or T-cell regulators. That's really what the basic strength of this institute is. So maybe we're going to look at Nobel Prizes coming from this institute. Now, that's really what our next step has to be. But I think moving forward, what strength, and we could spend the whole day even talking about how each group that has worked at this institute, each area of research, each significant milestone that they have brought in has given us results which have impacted society, whether it's the vac, or whether it's infectious disease models, or it's models that they've worked out on systems biology or computational biology. Each of them have got today. We've also seen it, and in fact, COVID was a good example again, where we saw a lot of work coming out from this institute, which had direct impact, not just, of course, on the medical countermeasure development, but on the understanding of the disease, how its management is going to be. And I think that really is what we need to move ahead. So it's not an incremental change that can now move ahead. It has to be an exponential leap that we move forward. And that is what we would like to see in terms of the research, in terms of the strength. NII has the competence to do it. It has the conviction to do it. It has the required collaborations to do it. So this is an institute which is definitely our institute of the future. Immunology research has already exhibited its strength, and I think we have to now see how collectively, and I couldn't attend the sessions this morning. I see Dr. Balram here, which I'm sure he gave a wonderful Foundation Day address. Each of those areas which have been talked about have potential for moving ahead. More will come around. Thank you.
Thank you, ma'am. Our next panelist is the former Principal Scientific Advisor to the Government of India, a man who has long championed far-sighted thinking in science and the courage to invest in ambitious frontier areas. From gene editing to synthetic biology to AI-driven discovery converging to advanced therapies, his leadership continues to shape India's scientific vision. Professor Vijayaraghavan, may I invite you now to share your views on the long-term frontiers in immunology and how India can build enduring scientific leadership in such areas.
Thank you very much, Gichandri. Can you hear me? Yeah. And it's really wonderful to hear Dr. Ganguly and Dr. Renu Swarup about their perspectives. I'll address the points which Gichandri mentioned, but let me start by saying how I came upon to first meet Professor Talwar and how that really transformed my trajectory. It was in 1977, right after the emergency. I was living then on tea, cigarettes, and marks, two of which are very dangerous to one's health. And I therefore got tuberculosis in a very bad state. And I was recovering. A friend of mine in Kanpur told me to come and see Krishna Prasad, KP, who was working in the biochemistry department. And I said, KP, I've got a few months to recover. I'm going abroad for my PhD. What should I do? And KP pointed to someone coming down the hall and said, go and talk to him. And that was Professor Talwar, who I didn't know anything about. So I told him about myself. And next to Professor Talwar was Mrs. Bhatia. And Professor Talwar said, isko SRO bana do. You know, and so I then got a job right that day, and my job was then to go and collect first trimester urine from the OPD and use ultrafiltration, as I was a chemical engineer, to concentrate it and give it to Wahi for the radioimmunoassay. So it was a sort of a reverse engineering for me, learning immunology and biology by actually getting my hands wet. But, you know, there were two things which impressed me. The biochemistry department was terrific. There were an extraordinary set of people there. And Professor Talwar had the ability to plug them into roles depending on what their talent was. So there were some people who were really good thinkers and who did nothing else, and they were left to do that, and others who are much more suited to taking on big mission projects, and they did that. And, you know, of those people, then I see Dinkar here. I don't know. Dinkar was working with IR at that time, you know. So Nilab Shastri, Dinkar, Vijayaraghavan, my namesake, Several others were there. It's a wonderful time. The other component which was really wonderful about that department at that time was the library. So you could, while in between your experiments, well, I ruined a Coulter counter, I remember, and I was not punished for that. So I was very grateful. Very expensive setup. I went to the library, and I was very impressed by two things in the library. One was entire volumes of the journal Reproduction, which had all papers from the Talwar lab on vaccines, on anti-pregnancy vaccines. So I was impressed that so much could come out from a lab with such intensity. But I also saw a conference proceeding there. And in that conference proceeding on reproduction, there was one article by someone who was not associated with the topic at all. And that was an article by Obaid Siddiqui. So Professor Talwar had invited Obaid for a talk. And this was a very good thing to do because, as you heard this morning, all life sciences is connected by evolution. And to get a neurobiologist and neurogeneticist to a talk on reproduction is a very wise thing to do. Anyway, I read that article, and instead of staying on at biochemistry at that time or going to Zurich for a PhD, I wrote to Obaid and decided to do my PhD in neurogenetics. So I owe this, my transition into Drosophila neurobiology, to a series of accidents facilitated by Professor Talwar and his speed of action. Now, one of the great features, to answer Geetanjali's question, what do we do going forward? There are two kinds of approaches, and my concern is that we need to push a bottoms-up approach, which was characterized by Professor Talwar's intensity much more than the top-down ones. The top-down ones are driven by governments which say, where are we lagging here, or where are the opportunities there, what do we need to do, and so on. And that results in institutions, big mission programs, funding ideas, and so on. Now, there are two kinds of scientists, those who look at these kinds of missions and say, how can I use this mission to grab something so that I can get some resources to do what I'm already doing? And there are others who say, not only do they grab opportunities to drive the mission, but they actually frame missions. And one of my concerns about the Department of Biotechnology's institutions is we have a National Institute of this, a National Institute of that, for stem cells, for plant research, for brain research, for immunology. Each of these have scientists who total the number of a small department anywhere in the world, right? This number by itself, no matter how well supported, cannot internally drive a mission. But this number can lead missions and transform these missions nationally. So there's a great opportunity for our institutions to do that. What Rajesh Gokhale has done is to unite all these institutions under one flag, and therefore there's a much more potential for seamless interaction. That interaction affords an opportunity. Extraordinary changes in technology which drive the generation of data but have challenges in their analysis is an opportunity. And extraordinary challenges which require us to bridge the genotype-phenotype gap, because our analysis of cellular properties, of phenotypes, of human behavior in different contexts, environmental causes, all of these have resulted in an enormous deluge of data. And potentially, the immune system and its response to various conditions, both inherited and external, is one of the greatest scientific opportunities in India. And therefore, I would urge the NII and the Department of Biotechnology to take a human immunome mission to understand, just as you're looking at genetics, we need to understand the context of how the immune system develops right from its earliest stages to maturity. And that fundamental knowledge is absolutely vital now, not only for our health, but also for a fundamental understanding. So I'd like to stop here to say, thank you, Professor Talwar, for your vision, your courage, and your ease of functioning. That really stimulated people to do what they wanted to do. Thank you very much. And give me my first job.
Yes, absolutely. Thank you so much. I'm sure we all will thank our panelists for setting such a powerful foundation for this panel discussion. We've heard their vision, we've heard ecosystems, regulation, community translation, a lot of essential pieces in the science to policy journey. Let's now build on these ideas with an interactive discussion. In this second round, I will put a question to one of the panelists. Others can chip in with a small comment later, and we can also have comments from the audience. So I'd like to begin. I think I'd like to begin with you, Dr. Swaroop, because through Dr. Renu Swaroop's vision, through her vision, DBT and BIRAC nurtured startups, incubators, and public-private partnerships in India that accelerated the path from research to products to policy. So, Dr. Swaroop, may I please invite you to reflect upon how India's biotech innovations foster more inclusive pipeline for translation and for impact?
Thank you, Gitanjali.
Yeah, I think it's
as I said, immunology research at this institute itself exhibited the power of innovation, the power of collaboration, the power of public-private partnership much before we even created an ecosystem for it. In the last couple of years, today there have been policy enablers, both in terms of creating an ecosystem, and providing the investments for it, providing the required connections and collaborations which facilitate that to move ahead. But I think what's important for us to keep in mind is there has to be a very fine balance between basic, applied, and translational research. Many a times when we look at the end products that we are developing, which obviously come out into society, and we've seen it in our innovation ecosystem that we have built through the large number of startups today, the incubators, the clusters that we have. There is many times a tendency to move towards one end of that where you feel that the impact is more visible. And I won't say the gestation period is short, because in biotechnology, the gestation period for any translational research is long. But on the other hand, it's very important to keep the foundation of basic sciences well grounded into the system. And I think this institute is unique for being able to do that. Whatever we may do in creating this translational pipeline, in integrating it within the larger cluster concept that we have, one thing for sure this institute has to do is to keep its identity in terms of its strength and competence in basic sciences, and making sure that that then moves through that value chain. Again, the cluster concept that we have in DBT, which was set up and now we see it in other places, is clearly a concept which can then take this immunology research to where we would want to see it moving. When we talk about innovation or we talk about areas of research, and we've seen that in many organizations, the moment you say, where is innovation research happening? The next step is, can we have an incubator in our institute? You don't need incubators in every institute. You don't need an accelerator in every institute to be able to drive that translational innovation culture within that institute. And I think that is what NII has to do moving forward. Strengthen its immunology research in areas where we've all been talking about, been mentioned as well, but stay connected with the other end of that value chain. And that comes through these clusters. If you talk about any of our global clusters, which we used to talk about earlier, whether it's Stanford, whether it's Boston, you see at the heart of it is the research or academic institute which brings in that strength. And I think that connecting with those components of it is what is important. It's not necessary that every researcher within the institute or every PhD student within the or maybe a large percentage of them move out to these startups. But yes, that culture of innovation needs to move so that they can actually make use of what enablers exist within the system. When Professor Talwar started the first vaccine research at NII, there was no policy framework which facilitated that. We didn't have a policy which allowed funding to go to industry. These were collaborations which were built on the strength of the research which got started here. There was no existing framework which allowed or which drove what technology transfer was or what intellectual property management was. And I think all that has evolved over these years. And that probably is the strength that we have to see how immunology research can move forward. Immunology research, the potential of immunology research that we have, there will be definitely innovations which will move into this larger space of moving out of the basic research, moving towards applied research. And I think that cluster concept is what we need to now promote to be able to see how we can take it forward. Professor Vijayaraghavan just mentioned about how all the institutes are connected. The NCR cluster was the first cluster, and we have the strength of the NCR cluster with NII, with THSTI, with the RCB, with NBRC, with all the health related, and now of course even the hospitals which are a part of it. So I think that is where immunology research can actually make a difference, but with each institute maintaining its identity, making sure that they make the best use of that innovation ecosystem which is built up. After all, we have a robust innovation ecosystem, but yet ensuring that each player has its own role in that value chain, and everyone does not sort of move to one and we sort of lose the balance with which we will have to take this forward. And that also holds true for the investments and the funding which comes in. We have seen many a time that the funding gets sort of skewed towards one side, which is again not the appropriate thing to be done, whether it's from within the government systems or it's international funding, etc. And as it's been said, innovation and science culture cannot be driven by what the funders want. It has to be the excellence of the idea that actually draws or attracts the funding to be able to see that you take it ahead. And probably innovation is something that we'll see it happening as it moves on.
Thank you, ma'am. Thank you very much for your comments. I think the next question is to you, Professor Ganguly, in following up from Dr. Swaroop's answer about making science and policy interactions productive. You are aware, we all are aware, that no translation can happen in isolation. It needs governance. Yet we often hear about friction between scientists, bureaucrats, and policymakers. The question that I have to you is, in your wide experiences, what models of coordination within and between the DBT, ICMR, Niti Aayog, or academia have worked best to ensure productive translation without red tape? I mean, maybe I'm asking, do we need to trust scientists more again, or regulate less, or what?
Actually, one thing happened during Dr. Khan's time, because normally we smiled a lot, we said we will work together, but no agency actually worked together genuinely. This happened during Dr. Khan's time, but he was a friend. So
but all of you have managed it. It's very successful. We need to know.
Managed it, but genuinely it started happening. It then continued during Dr. Vijayaraghavan, Dr. Renu Saru. Dr. Bhan brought that transformation. So we should genuinely work together, and that is something which is important. But in my opinion, that if you do science, somebody will take it forward. Like Dr. Talwar was also chemist. I am talking about today that it's very important that to be an immunologist, you should be a chemist. So he was also a chemist, and he developed that leprosy vaccine when everybody was opposing it. And it also worked on partially on tuberculosis. But the Bakulesh Khamar is sitting somewhere here. this same thing, exactly same thing, is now licensed by DCGI for squamous cell lung cancer, NCLSC, is known as Mycilex. It is being given to lung cancer patients. Cadila has made it. It has also been licensed with the Control General of India as Sepsivac. This is the only drug against sepsis. So this is something, again, Dr. Talwar, you did that. And we have another chemist sitting in the back, Dr. Balram. And Dr. Balram, you started working on the polymyxin peptides, and then Dr. Mathan and you actually did a PhD thesis for a boy, his name was Sunil David. And Sunil David used those peptides for action on the septicemia. Septicemia has got no drugs. So as you know that if you go on doing science, you don't get dementia. I have remembered every name and every event which has happened. So what I will say that go on doing your science. Somebody will translate it somewhere. You might also translate it, but somebody will translate. And I am tempting because Dr. Vinod Paul has come.
Yes.
And one of the major themes which he will be talking about, perhaps will be one of the things in immunology, which from the time memorial had gone into public health. That was the vaccine, right from Dr. Robert Koch, Louis Pasteur, you just name it. And this was very simple: kill the organism, or extract the toxin, or attenuate the organism and make a vaccine. But this is a science with the immunology, with multidisciplines. Now every platform is turning into vaccine. And there are so many new vaccines with new platforms which have come up in India, not only in COVID, but in the rabies, in the quadrivalent influenza vaccine. You just name it, and we have made it. So India, the vaccine research, which is really multi-centric, where you need material scientists, you need chemists, you need biologists, you need immunologists, you need engineers. This has gone into a tremendous transformative change. So I will make a plea that just go on doing the science. If you have done a good science, somebody is going to translate it into action. So I will stop here. Dr.
Thank
Paul
you
has
so
come
much.
already.
Thank you so much, Professor Ganguly. हम बाकियों से पूछ चुके हैं, Dr. Paul, and so for the audience, I would like to just say that we now have Dr. Vinod Paul, member Niti Aayog, with us. Dr. Paul has guided India's health systems policy from maternal and child health to vaccination programs and pandemic preparedness. And he has been a driving force in translating evidence into national scale action. Dr. Paul, could you, for us and for our audience today, share your perspective on how immunology innovations can be integrated into India's major national programs, such as the vaccination programs, family health initiatives, and preparedness frameworks, ensuring that science truly informs strategy and delivery?
Thank you very much. Most respected Dr. G.P. Talwar sir, distinguished this on the dais. There is so much of immunology in this room that I will take another aspect. That's what I bring to. That's what I would like to bring to the gathering here. I am a student of Dr. G.P. Talwar. मैं उधर से ही आता हूँ, बाकी इम्यूनोलॉजी आप उनसे पूछिए. अब वैसे भी नोबेल प्राइज मिल गया एक बार और. Such a big thing to happen. So let me bring that perspective. I am his student. So it goes back to 1972, 1st August 1972, I joined AIMS. And I'm sure the very first day we were exposed to respected Dr. G.P. Talwar. और वो माहौल उस टाइम पे के फैकल्टी ने स्टूडेंट्स को बहुत इम्प्रेस करती थी, बड़ी अच्छी जगह पे आप आ गए, बहुत इम्प्रेस करते और तरह तरह से वो डॉक्टर केसवानी अपने तरीके, सर आप ही के साथ उन्होंने ज्वाइन किया था. You were the two founding faculty joined perhaps on the same day, first two appointees at the All India Institute of Medical Sciences, handpicked by Rajkumari Amrit Kaur ji and installed as professor. 30 साल पे सर इनको प्रोफेसर बना लिया, हमें तो बनने नहीं देते प्रोफेसर. 30 साल पे ये लोग प्रोफेसर बन गए सर, इतने हिस्ट्री है ये, सब इम्प्रेस करते हैं. डॉक्टर तलवार वाज वेरी डिफरेंट, क्यों से अलग है, तो जोश, स्पॉन्टेनिटी, स्माइल, कपड़े अलग. मैं सोचा ये बंदा अलग है कुछ. Even though there was tough competition, the others equally impressive and unique personalities, but he different. One impression that you get was at his department, third floor, most vibrant floor in the entire academic side. Hospital को छोड़ दो, most vibrant. Lights का भी वहाँ off होती नहीं थी. Huge PhD program. Activity all through 24/7. And corridor, which is a busy corridor, up and down, and lots of people, and he's the live wire out there. Why I'm saying this is, and that's probably one message for the audience. The All India Institute of Medical Sciences at that time in particular was very focused on science, basic sciences, pre-clinical sciences, paraclinical sciences, of course clinical sciences, but then pre-para, biophysics, biochemistry, pharmacology, physiology, anatomy, and then microbiology. Very strong, so visible. Their presence was like, very, very special. And we as students would think this is the big one. This part is big. Unfortunately, sir, over the time, Dr. Ganguly sir, over the period of time, that clinical activities overwhelm the ethos of things, and I feel so sorry for this. And I very often take the opportunity to remind us that ultimately a medical university must have all pervasive sciences, and particularly the basic sciences, because they are the basis as well as the frontier of health sciences, clinical sciences. Foundation as well as the frontier. दोनों F जो आते हैं वो हमारे basic science से आते हैं. And in terms of the overall size of activities at AIMS, because this has shrunk for good reason. Good reason. I think it needs to be repositioned, reimagined. B, to say that that's how the ecosystem was built by the founding fathers of our institute. तो बहुत visible वो एक presence थी. So the buzz would be कि यहाँ गोविंद खुराना भी आते हैं और वो भी आ ही सकते थे, उनको भी फैकल्टी बनाना था, उनके दोस्त हैं, बड़े हम इम्प्रेस होते थे. But then let me now say teaching capability. That's my most profound impression about it. Fascinating. I mean, unmatched among, and believe me on this, clinical teaching, other people, but the classroom teaching, basic concepts in particular, and perhaps all branches included, most fascinating. And what I'm saying now is actually etched in my memory. This lecture on DNA. I think we had goosebumps because it was relatively recent. वो हैरान हो गए हम लोग सुन के, and he took this class not in the lecture theater. It was in a seminar room by chemistry for whatever reason. And he was going up and down the steps in the class. बहुत बड़ा room नहीं था, 50 की fact, हमारा batch ही 50 का था. And we had goosebumps. I couldn't believe that please believe that that lecture is etched here. And the profound and the love of subject that he brought in, the passion, I think the joy of teaching, that frontier, which was relatively nascent at that time, was exhilarating, and I just cannot forget it. He was also, of course, live wire on the campus. Cine Club sub-coordinate karte the wo. That aspect was very profound, and that gave us a sense of connect with the faculty, connect with the the ecosystem, a nurturing ecosystem, a Guru Shishya Parampara, as has been enshrined in our act actually. Words are there. तो उस माहौल को जिन्होंने दिया है उनमें डॉक्टर तलवार का बहुत ही बड़ा योगदान है. Amazing. I mean, he was special. He was to be looked upon. He was to be seen. He was to be wished. He was to be adored. But he was our true hero as a teacher, friend, philosopher, guide, and role model. Sir, I salute you fully. अभी पूरा नहीं. Dr. Ramalinga Swami would mention his work in Things Institute Day, his work on anti-pregnancy vaccine it had to happen to us. We knew कि ऐसा कुछ करेंगे, इतना बड़ा इन्होंने करना ही. And his words, अगर नोबेल प्राइज मिलने वाला है किसी को तो सर को मिलने वाला है. ये मैंने डॉक्टर करण सिंह से सुना पूरी मीटिंग में. इतना इन्होंने काम उस लेवल पे. आई एम सो प्राउड दैट these words, I heard these words in the auditorium all the other days to my conferences. When he moved to NII, I must say, sir, we missed you, although we had passed the stage of those subjects. I thought it was a loss. क्या करूं? इतने अच्छे लोगों को और कहाँ जाना चाहिए? But then we know what you did, sir, in your subsequent period of your contribution. You created this great institution. You're so proud. Your legacy was destined to be bigger than what you would have achieved in the All India Institute of Medical Sciences. But then we have watched your journey in awe, in admiration. I mean, thank you so much, sir.
नहीं कुछ पूछेंगे नहीं. I think you really put a showstopper there with your comments, Dr. Paul. And what we are hearing, sir, is that you are the power of connection between research and policy, between discovery and delivery, between institutions and the society. And with that, with the fact that we are completely tight on time, and Banya has been staring at me, I would like to still say that as we honor Professor Talwar today and are reminded that institutions like the National Institute of Immunology are at their best when discovery, policy, and public health work hand in hand, may I just ask each one of the panelists to give a single statement or a closing statement of where you see the Institute's frontiers going in the future, for each one of you. All right, Professor Ganguly, one sentence from you as a closing statement for translation.
If you really want to translate, you should love art, you should love architecture, you should love music, you should love food. Only then you will be a good scientist. All these are embodied in Dr. Talwar. And if you see this picture of NII,
Yes.
proves it.
Thank you, sir. Renu ma'am, what's the single most urgent policy frontier in immunology in India?
Whether it's immunology or anything, I will only say take forward what Professor Talwar has said.
Okay.
And this is something which is important for any research that you do. Nothing is impossible. If you want to do something, so it's like, it's not even sky is the limit. I think you always thought of beyond the skies as the limit. And I think that's really what we need to think today. Because you cannot just think of one aspect. So I think take the learning that we have as an example sitting before us. For everyone in this room, nothing is impossible.
Thank you.
Dream big and
do bigger. Yeah. Professor Vijay Raghavan.
Thank you. For science in general, there are two ways of doing really exciting science. One is to take the most important problems which have been defined anywhere in the world and do them well. And the other is look around you and work on problems which will be recognized as the most important. And I think Professor Talwar did both. And that kind of ability to do both, take cutting edge problems as well as look around and be inspired to look at solutions, is what we need more of. Thank you very much.
For NII, I would say you should strengthen, enlarge, expand your bridges with clinicians, public health people, and industry. I see that you are in it's a splendid isolation. Don't take it literally, but I'm saying that this campus should have lots of clinicians, public health people, industries walking in and out. I think that needs to be strengthened. I may be wrong. I've not seen systematically, but in the wake of COVID-19, looking upon you did your job, but then because I was handling for hand one-to-one, and I think there is a huge room for strengthening those bonds, clinicians, with public health, people, experts, with industry, and with startups, and also create technologies. By the way, हम दोनों ये बात करते थे उस टाइम, तो इन्होंने कहा नहीं. Thank you.
Let us take what we've heard today in this room and turn it into action so that India's immunology research not only leads the world, but reaches every hand that needs it. With that, we would like to close this panel and thank all of you. I know that we've not taken Q&A from the audience, but the panelists are here. They're going to be with us for dinner, and you can ask them and grill them and talk to them after this. Thank you very much for having us on this. Thank you, each one of you.
Thank you. I thank the panelist and our moderator, and I request you to please wait on stage for a few minutes as Dr. Mohanty joins us to present a token of our gratitude and appreciation for all of you for your insightful discussion today. Professor N.K. Ganguly, thank you, sir, for joining us. Dr. Renu Swaroop, thank you, ma'am, for joining us. Professor Vijay Raghavan, thank you very much, sir, for discussing the points that you brought forward and for joining us. Dr. V.K. Paul, thank you for joining us. And Dr. Gitanjali Yadav, thank you very much for leading this. And with that, we end the scientific discussions of today's sessions. And we will very soon at 7 have a special program for all of you, a Santur recital by Pandit Abhay Rustam Saporiji. So till then, you're free to explore the beautiful campus of NII and have a little walk. And then please come back at 7.